Hi, I'm Amy Prowal, President and Research Director of PolyBio Research Foundation, and I want to welcome you to our Fall 2024 Symposium. Now, first, I want to let you know that you can post questions throughout the symposium to relevant speakers if you have questions about their talks in the chat, and we'll do our best to answer those questions over the course of the symposium in the chat. Now, I want to give you a background on PolyBio. We're a nonprofit research organization building a collaborative network of scientists and doctors dedicated to studying how viral, bacterial, fungal, and other pathogens contribute to a wide range of chronic diseases. These include long COVID and ME/CFS, but also conditions such as Alzheimer's disease, cardiovascular disease, and even parts of what are sometimes deemed healthy human aging. Now, data from our projects are channeled into treatment innovation and clinical trials of drugs that might help get people better. In other words, we're always working to take our academic knowledge gained from projects and figuring out how to best apply it to actual diagnostics or treatments in the real world. Now, we work with the design team to build this module, and it facilitates knowledge of our research and therapeutic projects, technologies, and findings across international institutions. So, for example, here is one of the many projects we both helped to conceptualize and to support. It's called Delineating Vascular Coagulation in Live Immune Cell Dysfunction in Chronic Illness. Now, what the team's doing is using advanced technologies that capture the activity of live neutrophil immune cells and other parameters of the immune response in patients with long COVID and pre-COVID ME/CFS. They're also running a targeted panel of cardiovascular disease and blood vessel markers, plus measuring clotlet and platelet activation measures. Now, the study is run by my PolyBio co-founder, Mike Van Weyenburg, who you'll hear from later today, and it's based at Harvard Medical School. There's also a group of pediatric long COVID patients, children in the study. With that arm of the study, an analysis led by Dr. Lael Jonker, who you'll also hear from later today. There's even a nurse that goes directly to the homes of patients in the study to collect their blood, which means that the most underserved and severe patients also can participate in the research. And thanks to a new likely grant, we should be able to ship samples from the study to the J. Craig Venter Institute in San Diego, California. And there, an incredible collaborative network will work on the samples further. They include Gene's Tan's lab, who will analyze a wider range of parameters of the immune system in samples, including looking at dysfunctional antibody responses. And you'll also hear from Gene talk later today. Now, here's a view of all of our projects. And we're truly proud that we have groups working on projects at many of the top institutions in the world. These include the Karolinska Institute in Sweden. They include the University of Pennsylvania. They include the Yale School of Medicine. They include Institut Pasteur in France. They include Cardiff University in the United Kingdom, and actually multiple institutions that could not even fit on this page. Now, importantly, our projects are designed to span the full range of the human body in terms of working to understand disease mechanisms, including parts of the long COVID disease process. For example, it's really important to understand these body systems because the virus, bacterial, fungal, and even parasite pathogens that can contribute to complex chronic illness processes infect tissue across the entire human body. They infect the brain and the spinal cord, and thus we must analyze many body systems, often in tandem, to understand a full picture of chronic disease. Thus, we have groups working to study and understand dysfunction of the autonomic nervous system. We have many teams working to better understand how infections and other processes can dysregulate the central nervous system. For example, you'll hear from Tim Henrik from UCSF speak later today about a project using advanced multimodal PET imaging to measure T cell activation throughout the bodies and brains of patients with long COVID, and now some patients with pre-COVID ME/CFS. You'll also hear Mike Van Weyenburg talk about how a team at Harvard that he's working with is using advanced 7 Tesla imaging to measure cerebral spinal fluid flow and a range of other parameters across the central nervous system in patients with craniocervical instability. We also have teams working to characterize human microbiome activity and composition across different body sites. We have teams working on the immune response in long COVID, but also related diagnoses. We also have teams working on the cardiovascular system, which is truly important in the context of SARS-CoV-2 infection, because the SARS-CoV-2 virus has a profoundly detrimental impact on the blood vessels. So, for example, Chiara Gianarelli's team at New York University is collecting blood tissue samples from the blood vessels of patients with heart disease. And she's analyzing those samples to see if SARS-CoV-2 and its potential persistence in macrophage foam cells that form part of arterial plaque, plus other inflammatory processes, may contribute to a higher risk of cardiovascular disease, especially in people repeatedly infected with the SARS-CoV-2 virus over time. We also have groups working on the respiratory system, teams working on the gastrointestinal system, which is definitely important in the context of long COVID, because the lining of the gut, the tissue of the gut, seems like a primary site of SARS-CoV-2 persistence over time in at least some patients with long COVID, and also a potential site of enterovirus RNA persistence in patients with pre-COVID ME/CFS. There's also groups working on the endocrine and reproductive systems, and that's truly important because it's increasingly understood that sex hormones control, to a large degree, how the immune response is able to handle infection, which is a topic that Peter Broden from the Karolinska Institute will talk about more later today. Now, many teams have worked hard to collect tissue samples via biopsy or surgical procedures from patients with long COVID or related conditions. These samples include gut tissue, lung tissue, lymph node tissue, blood vessel tissue, bone marrow, tonsil tissue, and even endometrial tissue. For example, you'll hear Dr. Nadia Rowan from UCSF talk later today about a project that's collecting endometrial tissue in order to best understand if SARS-CoV-2 persistence or immune dysregulation in the female reproductive tract may contribute to the higher incidence of long COVID in women. Now, there's also sample sharing between sites. So, for example, we support the UCSF LINK cohort, which started in 2020, tracking patients with long COVID and collecting their samples over time. And you'll hear Michael Peluso talk more about LINK later today. Now, LINK serves as a clinical and sample core that can send samples and is sending samples to other teams in the consortium for analysis. So, for example, you'll hear Isan Safiq talk about a project where a group of Sterling professors she's working with at Yale, including Akiko Iwasaki, and you'll hear from both Isan and Akiko later today, are working on a project using a mouse model to study mechanisms of SARS-CoV-2 RNA persistence over time. They're even giving therapeutics to the mice to see which therapeutics might disrupt or best break up persistent RNA. And what's important is that the LINK team at UCSF is sending them more samples for analysis so that they can validate their findings in mice in human tissue sent from the LINK team, which will greatly add to the overall scope and knowledge gained from the study over time. In other words, many or most of our projects are designed to be iterative. They start with components, for example, components of biology and long COVID, but are moving into related other diagnoses when feasible. So, for example, here is a clinical trial of, let me find it, repurposed HIV antivirals in long COVID, repurposed HIV antivirals. And this is a trial that we're running at CORE, the Cohen Center for Recovery from Complex Chronic Illness, which is a clinic at Mount Sinai, working to innovate treatment for patients with long COVID and related complex chronic conditions. I serve as scientific director of CORE, along with my amazing colleague, David Petrino, who's the NASH family director. Now, the way this trial came about is that in 2021, Mike Van Elziger and I wrote a paper on biological factors that likely contribute to the long COVID disease process. These included SARS-CoV-2 persistence in tissue over time. It also included the reactivation of other bacterial or viral pathogens in some patients with long COVID, including the Epstein-Barr virus. And We also delineated the possibility that SARS-CoV-2 infection might kick up the activity of human endogenous retroviruses, which are elements of the genome that Akiko Iwasaki will talk about more later. Now, as we understood these parameters, David Petrino and Akiko Iwasaki actually began to collect blood from patients with long COVID, and they ran a huge amount of analysis on these samples, uncovering many biological abnormalities, which they published in the journal Nature. Among them was the fact that some patients with long COVID did indeed show signs of Epstein-Barr virus reactivation. This was encouraging. So David set out to find teams that were using therapeutics in other spaces to be able to try to help people manage Epstein-Barr virus. And what he learned was Michael Levy and a group of scientists at Harvard Medical School were using an HIV antiviral, Truvada, or related antivirals, in an effort to control Epstein-Barr virus activity in patients with multiple sclerosis. And that's because, at least in the lab, Truvada has been shown to have some activity against Epstein-Barr virus, in some cases SARS-CoV-2, and even in some cases those human endogenous retroviruses that you'll hear more about later. So we thought, okay, let's work with Michael Levy's team, and they were kind enough to help us build a clinical trial protocol of Truvada in patients with long COVID. Now, at the same time, we heard from the patient community that Maraviroc, another affordable HIV antiviral, was helping some patients with long COVID. So we added that into the trial along with Truvada. As we designed the study, importantly, we designed it so that blood samples are collected from patients undergoing the interventions at the beginning of the trial, at the middle of the trial, and at the end of the trial. And what we're doing is we're sending those samples to Akiko Iwasaki's lab at Yale, where she's analyzing them for a wide range of infectious, immune, hormonal, and other parameters. And at the end of the trial, that allows us to crunch the data. And if someone or patients, certain patients, optimally respond to either Truvada or Maraviroc, we can understand their biological characteristics. We can understand what metrics most correlate with their potential response to the drug. What that allows us to do is then build a potentially tighter, better phenotyped phase 3 trial that could go on to help patients with these drugs, or to add a new arm of patients into the trial, potentially with pre-COVID ME/CFS, or with tick-borne or vector-borne illness, once we best understand the characteristics of responders to the therapeutics and also the side effects that might occur in patients who are given the medications. Thank you.
Okay, so today we'll speak a little bit about our recent results regarding the role of a molecule which is called THEMIS in the modulation of NK cell activity during SARS-CoV-2 infection. So the story starts 1 year ago when we show, in fact, that the virus was able to persist up to 80 months in alveolar macrophages in a non-human primate model of SARS-CoV-2 infection. Behind the fact that the virus could persist in macrophages, we also show that interferon gamma could modulate the viral replication by inhibiting viral transcription in macrophages which were treated with interferon gamma. Moreover, we also show that interferon gamma treatment of macrophages increased at the surface of macrophages a non-classical molecule called MHCE. And this molecule is of interest due to the fact that, in fact, this molecule is recognized by inhibitory receptor expressed at the surface of NK cell, which is named NKG2A. In this context, when NK cell will recognize infected cells expressing MHCE, NKG2A will inhibit the capacity of NK cell to degranulate. So in the paper, we decided to test this hypothesis, and we compare the capacity of NK cell to kill target cell expressing MHCE load with a different kind of peptide. We use two peptide controls, the VL9 peptide, which is a peptide which is expressed by CMV viruses, and HSP60 peptide, which is in fact came from a protein, a cellular protein. The VL9 peptide is known to strongly inhibit NK cell. As you can see, when a target cell are low with this peptide, we have around 75% of inhibition. At the opposite, the HSP60 peptide is known to not inhibit NK cell, and you can see that we have no inhibition.
Recording in progress.
When NK cell are when they are used to kill the target cells. Interestingly, when we use a peptide derived from the single peptide of the SARS-CoV-2 glycoprotein, we found, in fact, an inhibition of NK cell cytotoxic activity. More surprisingly, when we segregate the monkey according to the status, meaning that, in fact, when we separate monkey which have recovered from the infection, meaning that they have we are not able to detect any viruses after 80 months of convalescence. And when we compare with monkey in which we were able to detect some viruses after 80 months of convalescence, you can see that, in fact, the monkey for which no viruses could be detected were not inhibited when they are put in culture against the target cell load with spike peptides. So we hypothesize, in fact, that there is a mechanism which leads to inhibit the inhibition of the leads by NKG2A, meaning that there is potentially a molecule which could inhibit the signal transduced by NKG2A. So we decided for this study to investigate which could be the molecular or contextual mechanisms which could explain the relief of inhibition in NK cells. So I will not go into detail why we choose this protein, but we decided to focus on THEMIS. So THEMIS was described now 15 years ago. It's a thymocyte express molecule involved in selection. It plays a great role in CD8 and CD4 cells. In fact, THEMIS is essential for the development of thymocyte by regulating the positive and negative selection of T cells. In fact, it modulates the strength of signal of the TCR. How it works, in fact, THEMIS is able to segregate an adaptator molecule which is involved in the transduction of the signals led by the TCR, and this molecule is called SHP1 or 2. By interacting with this molecule, THEMIS, in fact, modulates the signal delivered by the TCR. Interestingly, SHP1 is also involved in the transduction of the signal of the inhibitory receptor expressed by NK cell, and especially by NKG2A. So we hypothesize that THEMIS could play the same role in NK cell as it plays for the TCR in CD8 and CD4 T cells. So we decided to investigate if THEMIS could be modulated during SARS-CoV-2 persistence. So we go back to our monkey and we decided to follow up THEMIS expression in both blood and in bone marrow. We decided to focus on bone marrow because THEMIS is involved in the generation of CD8 and CD4 T cells. So bone marrow could be a good site to investigate the role of THEMIS in the generation of NK cells. As you can see, we are able to detect a high level of THEMIS in both B cells and T cells, validating the fact that the antibodies that we use could detect these molecules in monkeys. And we were surprised to see that we were able to see two levels of expression of THEMIS, as you can see on the histogram on the left part of the picture. We quantify the frequency of THEMIS positive cells in each subset, and we can find that THEMIS was highly expressed on B cell and T cells. And we are surprised to see that, in fact, THEMIS was downregulated in NK cells isolated from chronically infected monkeys, whereas it seems to be upregulated in a monkey which have recovered from the SARS-CoV-2 persistence. So we also look in the bone marrow, and we observe exactly the same picture. Indeed, THEMIS seems to be downregulated on NK cell in the bone marrow, but in chronic infected monkey, and is still recovered in monkey which have recovered from the infections. As I mentioned previously, there is two levels of THEMIS in NK cells, so we decided to investigate this aspect, and you can see that, in fact, by using NKG2A and THEMIS, we are able to define different kind of NK cell subsets, expressing or not THEMIS and NKG2A. And we were able to show, in fact, that in a convalescent monkey, which have still a chronic virus in their lungs, we found, in fact, a decrease of NK cell expressing both NKG2A and high level of THEMIS, where we found upregulation. an increase of the frequency of NK cell expressing a low level of THEMIS and NKG2A, suggesting that, in fact, those monkeys could not respond or will be inhibited by the MEGE molecule when it's expressed at the surface of the macrophages. So we also look in the macrophage, in the bone marrow, and we in fact found exactly the same picture. THEMIS seems to be downregulated. THEMIS high expression seems to be downregulated on NK cells in chronic infected monkeys, whereas still we found the same level that in healthy control in monkeys which have eliminated the viruses. So we decided to investigate which could explain the difference in the THEMIS level expression in NK cells, and we decided to isolate NK cells from the monkeys. So we take blood, we isolate PBMC, and we put PBMC in culture with different cytokines known to be involved in NK cell regulation. So here you have a list of the cytokines that we have tested. There is cytokine which is involved in inflammation and other which are involved in NK cell maturation. We also decided to test TGF-beta, which is known to block NK cell activity, and fatty acids. Fatty acids are known to participate to the regulation of NK cells. So we first, after 24-hour culture, we decided first to estimate the impact of the different cytokine of THEMIS expression of both B cell and T cells. And you can see that we have a very low effect of the different cytokine on THEMIS expression of B and T cells. Now we first look of the role of the impact of IL-15, IL-21, and IL-7 on THEMIS expression of NK cells, and we do not see any differences between cells which are stimulated or not with the cytokine when we with control with NK cells which are not stimulated. Interestingly, we found in the case when we stimulate NK cell with IL-12, IL-18, and IL-1 beta, a strong decrease of THEMIS expression on NK cells when we compare with the same NK cell, but not stimulated by the different cytokine. Interestingly, both of those cytokines are expressed by macrophages, suggesting in fact that when macrophages start to produce or are inflammatory, they in a certain way participate to the downregulation of THEMIS expression, rendering difficult the capability, perturbating the capacity of NK cells to kill infected cells. So we show that THEMIS was downregulated in chronic infected monkeys. So we next decided to investigate when it could be occur in humans. And if it occurs. So for that, we decided to perform a pilot study in a human cohort, which is called cohort 5. It's a cohort of the Institute Pasteurs. The patient was longitudinally follow-up. They were included in the study immediately when they start to have symptoms. So the inclusion was 2 to 4 days after the onset of the symptoms. All the patient the strain was in each case sequenced to be sure of the strain which have infected the patient. And we have a group control. So I will show result obtained from 10 patients, but we are doing more patients currently. And we follow the patient at the inclusion 1 month after and 6 months after the acute phase. So based on this, we decided to use a panel of cytometry to define different NK cell subsets. We are able to find 15 NK clusters. We use different markers to define different levels of NK cell maturation. So we use, as you can see on the bottom, we use a CD56, which represents a non-mature NK cell, and NKG2C, which represents the more mature NK cells. We also include this panel NKG2A and THEMIS. So when we focus only on the acute phase of the infection, and we compare the distribution of the different clusters between healthy control and SARS-CoV-2 infected patient, we don't see so much differences, within the exception of one cluster. It's in orange on the left part of the UMAP. You will see better after 1 month. 1 month after, we found, I don't see, just a second. Okay, so we found a cluster which seems to increase during 1 month after the onset of infections when we compare between the healthy control and the SARS-CoV-2, and 6 months after, this cluster is maintained in patients which were infected by SARS-CoV-2. Interestingly, this cluster express a high level of NKG2A and a high level of THEMIS, suggesting that in fact, in those patients, there is an induction during the acute phase of this NK cell able to recognize and kill the infected cells. So we measure the frequency of this population in our patient, and you can clearly see an increase of this cluster 1 month after the onset of the infection, and start to decrease only 6 months after the acute phase. So to conclude, we decided to investigate the role of THEMIS. THEMIS seems to be involved in the regulation of NK cells. It play a role in CD8 T cell and CD4 T cells. We investigate the capacity of THEMIS to regulate NK cell development. We were mainly interested in the fact that THEMIS could play a role in the regulation of NKG2A, which could be a killer molecule to the induction of adaptive or memory NK cells. We are able to show, in fact, that THEMIS could contribute to the control of infected cells, especially through NKG2A activity. And we think that it could be important due to the fact that THEMIS could influence NK cell response against SARS-CoV-2. It could participate to the regulation of the chronic inflammation. In the case of if THEMIS is mainly downregulated, it could be involved in the immune dysfunction, especially regarding NK cells. And by dysregulating NK cells, it could participate to the persistence of the symptom if the virus is not eliminated. I will stop here and would like to thank all the people which finance all our founders, all the members of the team, especially Michela, Beatrice, all the persons which are in charge of the monkey, and all the patients for their help in our work. Thanks. If you have any question or comment, I don't know.
Great, Nicolas. Thanks so much. I mean, understanding natural killer cell activity is important for diagnostics and therapeutics in this space, so it's really huge. I'll move to our next speaker, who's actually going to be David Price. David is the chair of infection and immunity at Cardiff University School of Medicine, and he'll be talking about infectious immune and microbiome signals in the long COVID lug. Thanks, David.
Thanks, Amy. Okay, so thanks, Amy, for the kind invitation to present today. What I'd like to do is just give you a recap on our first study, which was conducted using peripheral blood samples from patients with long COVID who were infected with a documented episode of initiating SARS-CoV-2 infection early in the pandemic, and healthy controls who were infected at the same time but did not go on to develop long COVID. And then I'd like to tell you how that led us to look in the lung for clues to the pathogenesis of long COVID and give you an update on where we are with that study. So the peripheral blood study started in 2020, very early after the pandemic started. And I'm just going to show you 3 slides to summarize the key findings of what was actually a huge amount of work over many patients. So the first thing we did was to use high-dimensional flow cytometry to just get an overall picture of the circulating immune system by looking at immune cell lineages, and that's shown in the heat map on the top left. So we're looking here at things like lymphocytes, basophils, dendritic cells, monocytes, and comparing the frequencies of these cells in the blood with across healthy convalescent controls and patients with long COVID. And essentially, we saw very little difference between these 2 groups. Similarly, when we looked at the frequencies of antigen-specific T cell responses against SARS-CoV-2 and against some other viruses that are sometimes reactivated in patients with long COVID, such as CMV and EBV, we found no quantitative differences in antiviral reactivity between healthy convalescent individuals and patients with long COVID. When we applied a high-dimensional phenotypic panel to look at really how these cells will look rather than just how many of them there are, and this is shown at the bottom left here, we found no phenotypic differences in T-cell responses targeting the spike protein of SARS-CoV-2, and we think that's because both patients and healthy convalescent controls were subsequently vaccinated, either before or after they're initiating infection, so they're exposed to the spike antigen in the vaccine as well as in the original infection. But what we did see was that there were signs of what we call T-cell exhaustion in the non-spike compartment. So these are T-cells targeting other proteins of the SARS-CoV-2 virus, particularly PD-1 and TIM3, which are known to be essentially inhibitory molecules that develop to indicate that the response has been exposed repeatedly to antigen. So we took this to mean that, that there was potentially ongoing antigen exposure in people with long COVID, consistent with persistent SARS-CoV-2 infection. More strikingly, when we looked at antibody titers against SARS-CoV-2, in terms of the total antibody titer, this is shown on the top left, we saw no differences. But what we did notice is that there was a significant difference shown on the bottom left here in neutralizing activity. And this is actually, I haven't updated this graph. This has been updated across many more patients now, and it's even more significant. So essentially, patients with long COVID are less able to neutralize the virus despite having similar amounts of total spike-specific antibodies. And following on from the last talk, we saw no differences in a process known as antibody-dependent cellular cytotoxicity, where the antibodies essentially prime cells to be killed by NK cells. We then did targeted proteomics using the Olink platform, and the two key take-home messages here are first on the top left. If you just do simple volcano plots across 4 distinct panels looking at cardiovascular, neurological, cancer and inflammatory markers, and this totals 3,072 proteins, you can see that large numbers of proteins are upregulated in patients with long COVID. This really, I think, reinforces the fact that long COVID is a systemic disease. There's clearly something important going on. And when we did a deeper dive into those data, we found that many proteins spanning things like apoptosis, cell death, and proliferation were upregulated in people with long COVID. The update here since the last symposium is that we've now identified key markers that associate with the symptom of breathlessness. If you actually break down the cohort into people with distinct symptomatology, and those are things like TNF inflammatory molecules and thromboxane A2, which is a marker of platelet activation, suggesting there's some lung pathology going on. And the third key finding really is that complement, which is an innate system that mediates antimicrobial activity, was activated very prominently in people with long COVID. And that actually was predictive with an area under the curve of around almost 0.9, predictive of people having long COVID. And again, we think that is because the cascade is activated by persistent virus. So from that study, just to summarize, no differences in circulating immune cell lineages and no differences in circulating virus-specific T-cell immunity, but some evidence of phenotypic differences suggesting ongoing viral replication somewhere in the body. And we think that might be enabled by suboptimal antibody-mediated neutralization in people with long COVID. We found substantial perturbation of the plasma proteome, which suggests a covert pathology that's not obviously associated with widespread viral replication, but nonetheless drives systemic disease. And what we inferred from these findings was that ongoing tissue damage was associated with local reservoirs of SARS-CoV-2, and these could be a mechanistic driver of long COVID. And that's what led us to look in the lung as really the site of initial infection with SARS-CoV-2, and therefore a good candidate, I guess, for looking for cellular and molecular origins of long COVID. And this is the study that was funded by and still funded by PolyBio. This is just an outline of the study, which I shall skip. But essentially, we sampled, and I list it here actually. We're sampling peripheral blood again, plasma, serum, and saliva, but we're also sampling bronchoalveolar lavage fluid, where we wash the lungs and collect the fluid. And we're taking transbronchial biopsies, about 4 to 6 transbronchial biopsies per patient to actually get lung and alveolar tissue. And what we planned to do was to do metagenomics, metatranscriptomics, and single nuclei RNA sequencing in collaboration with colleagues at the JCVI to see if we could do a broad scan to get some idea about the pathogenesis of long COVID at the site of initial infection. The update here is that we've completed our initial quota of recruits. So we have 15 patients who do not have long COVID, and 15 people who don't have long COVID, and 15 people who do have long COVID, which was our initial target recruitment range. In terms of updates, this is a project that has a very, very long lead in, given that it takes a while to get the ethics and to recruit the samples, schedule people for surgeries, and process and collect the samples and send them to the JCVI. So we're currently awaiting data on most of these studies. The metagenomics is, the microbiome studies are exciting because we know very little about the lung microbiome in health or disease. And this is being run by Chris Dupont at the JCVI. And the single nuclei RNA sequencing effort, also run by Chris, is in process. And that's required quite a bit of optimization with the samples that we've sent over to date. Last time I spoke, some preliminary samples were being mapped to the lung atlas. But the good news and the update for this symposium is that Mark and Chris have done a phenomenal job in figuring out how to isolate sufficient numbers of nuclei from almost all of the samples we sent to enable single nuclei RNA sequencing. What that means is that sample collection processing at our end is compatible with the experimental pipelines at the JCVI. And that, I think, was probably my biggest worry about the study, was that we would encounter some general technical failure that would negate all the work we've done so far. But this is a really exciting update from our perspective. We expect a slew of data by the time we get to the next symposium. And in line with the fact that everything seems to be working so well, what we're doing now is extending our ethics here so that we can do another set of sample collections for spatial transcriptomics, hoping we can update by the next symposium, and Chris may actually have some updates today, but we'll wait and see. Acknowledgements, everyone in my lab, everyone at the JCVI, everyone at the University Hospital of Llandoch, where we collect the samples, which is just down the road. Our collaborators at the Karolinska Institute, who are also linked into this lung study, are doing a fantastic job with various platforms, including Olink. And the people at the Karolinska Hospital are also collecting samples, lung epithelial brushings to complement the transbronchial biopsy studies. Special thanks to Amy and Mike at PolyBio for funding this study to make it possible, and to everyone who participated and actually was incredibly brave to agree to undergo bronchoscopy for sample collection. That's just a phenomenal effort, and we're extremely grateful. And I have to say, that's not something I think I would be brave enough to do. So many thanks to them, and thanks to everyone for listening.
Great, David. Thank you. That was an excellent overview. And as you know, we meet every week to go over the progress on this study and others. And it's been so cool to see it coming along piece by piece. So it's really exciting. All right.
Thanks.
Well, next speaker will be Resia Pretorius. She's a distinguished professor in the Physiological Science Department at Stellenbosch University in South Africa. She'll be talking about microclots, bacterial biofilm, and NETs, optimizing methods for biomarker identification.
Thank you very much for this opportunity to give a short update on what we have been doing in our laboratory. I just wish to acknowledge my key collaborators and students that were involved in the work presented today. And just to name a few, Prof. Doug Cal, my long-standing collaborator from University of Liverpool, and his postdoc, Justine Crixie, my Stellenbosch team, Chantal Venter, the blood lab manager, and a few of my dedicated students, Massimo Nunes, Simone Turner, Tom Usher, and Nell Thompson, and Maxine Waters. Our lab's focus is to understand which biomarkers are of importance in long COVID. We wish to also understand the presence of the various symptoms and then understand which biomarkers are indicative of which symptoms. Eventually, we would want to see the clinical interventions and customize based on the combination of the symptoms, but also the reliable biomarkers for specific long COVID phenotypes. Thus, our reasoning is that in clinical trials, we need measurable biomarkers before and after interventions. And we believe we have a good candidate and with some added benefit, and that is microclotting. It also entraps various different inflammatory molecules, making it a relatively easy and quantifiable method to look at entrapped molecules of interest. In our research for novel biomarkers, we have then focused over the past months on various markers that might be of interest and that might be associated with microclots. I will be discussing now tonight just a few, including NETs and myeloperoxidase, entrapped and structurally associated with microclots. The work was done together with Professor Alain Thierry. Fibrinogen is key component inside our microclots, biofilm associated with microclots, Syndican-1, which is an endothelial marker, and then also just pre-printed our work on extracted stroke clots with Doug Cal and his postdoc, Justine Crixie, and a team of neurologists. So if we look at what we have recently found in the collaboration with Professor Alain Thierry from INSERM, he's interested in NETs, formation and neutrophil elastase, myeloperoxidase, and circulating DNA in long COVID versus control samples. We confirmed that NETs are entrapped and structurally associated with microclots in individuals that has got long COVID. We used, in this case here at the top A and B, we used the cytostain, which is the red stain, and which is very similar to Huot's. And Cyto and Hus are very well-known DNA stains. Also, theoflavin T for amyloid protein were used in these analyses, and myeloperoxidase and MPO antibody, confirming that NETs and the various molecules associated with NETs, MPO, etc., are structurally entrapped in some microclots at least. Persisting virus presence could be the reason for a constant production of spike protein. This will result in increased spike protein in circulation and a constant spewing out of the spike protein, and also dysregulation of many other inflammatory molecules. Spike and these molecules do not only have the capacity to bind to circulating fibrinogen proteins, but we hypothesized that it might also have the possibility to make the fibrinogen protein itself amyloid. Over the last few months, we have added spike protein to purified fibrinogen and used a fibrinogen alpha chain antibody and our marker, also theoflavin T. And in this figure here, you could see that there is definitely fibrinogen that can be detected in the presence of spike. The fibrinogen becomes amyloid, as can be seen with the green signal. Here you can also see fibrinogen analysis where we added LPS. Again, the purified soluble fibrinogen turned into an insoluble fibrinogen clot with areas that show amyloid protein changes. Note the green areas in the clot on the far right, embedded in the fibrinogen orange areas, which is demarcating the alpha chain of the fibrinogen molecule. Here is another example of the molecular content of microclots, and at this time from a long COVID participant. The blue areas are that of NPO. The net itself was not marked in this specific example, but one could imagine that it expands out into from the microclot into the topmost areas to reach the blue signal of the NPO. Unfortunately, we couldn't add 4 markers together in our system. Here is some more data from long COVID microclots showing co-localization of molecules of interest, and again confirming that the microclots contain fibrinogen as demarcated by the red signal from the fibrinogen alpha chain antibody inside the clot and in association with the amyloid protein areas demarcated by the green signal from thuroflavin T. We also then, as I mentioned previously, interested in biofilm presence, and this was one of the key studies that were funded by PolyBio. Although we anticipated that biofilm would not necessarily be prominent and a marker in all microclots, it may give us important information of the specific drivers of microclot formation in a subset of patients that in long COVID is in a specific phenotype where bacterial reactivation might be of importance. As we know, biofilm contributes to clinical infections linked to chronic disease progression. And the bacterial clusters form protective self-produced matrices that adhere to surfaces and resist immune attacks. The matrix can also shield the bacteria and perhaps also persistent virus. So therefore, and as we know that fibrinogen can be associated with these biofilm areas, we decided to look at this specific marker. It proved to be quite a challenge to optimize the methods, and we've been busy now for nearly 2 years. Thus, we embarked on a titration series to determine the optimal marker concentration. We also got access to a Thermo Fisher spectral viewer, and that provided a good overview of the spillover between fluorophores, and thus the ability to view these fluorophores separately and in combination with ones that we would like to use together. It thus allowed us to prevent spillover and to separate the signal better. Here you can see the microclots stained with biofilm. This confirmed the presence of biofilm matrices in plasma and possibly then associated with microclots. And then we continued to stain the plasma for the biofilm, and we saw co-localization also with the amyloid areas, the THT signal, the green one, and that was quite nice for us. And also we could see co-localization with the alpha chain antibody of fibrinogen. So here is an example where you can see the alpha chain fibrinogen also in association with THT and biofilm. As I mentioned, it's not in all microclots, but it's good to understand that that is a marker in a subset of microclots. Then we followed this up with imaging flow cytometry. On the left, you can see the images captured via the imaging flow cytometry that showcase the typical appearance of biofilm. We see channel 1 and channel 9 as the bright field channels, channel 3 as the fluorescence intensity of the biofilm, and 6 as side scatter. And the figure on the right illustrates the concatenated files of the biofilm titration series, with T1 showing the highest antibody concentration and T8 showing the lowest antibody concentration. So now we know which antibody concentration works the best. Then we also went on and looked at Syndican, which is a very important endothelial marker, damage marker. And we believe that Syndican might be a good candidate in long COVID as well. We did indeed find Syndican in our samples, in our long COVID samples. And here is an example where we use Syndican antibody with THT and with alpha chain antibody. Also showing that Syndican is entrapped in a subset of microclots. We again used the Thermo Fisher spectral viewer. We again used a titration series, and at last we again have the flow cytometry with the various channels 1 and 9 for the bright field, channel 3 for Syndican, and then the side scatter laser as well that you could see that shows the specific side scatter complexity of the objects passing through. So with that said, the last few slides that I want to show, last 2 slides, I'm excited to share our latest preprint results done by Doug Kell's lab with his postdoc Justine Crixie and a whole collaborated team of neurologists, including Melanie Walker and Aladenku Senkar from the Department of Neurology from the Walton Center at the University Hospital in Liverpool. And we looked at clots extracted from stroke patients. Now, The reason why we wanted to do that, we wanted to confirm that microclots can indeed go into macroclots, develop into macroclots. And here you can see THT signal, suggesting that microclots, which also show the THT signal, might go together and eventually group and form these macroclots that can be extracted, and that macroclots in stroke patients indeed mostly contain amyloid protein areas. So that's a recap from what we have done the past few months. I just wish to absolutely dedicate this last slide to PolyBio. PolyBio has supported Massimo Nunes when he started his PhD. He's now completed his PhD in 2024. Together with Bolvi Research Foundation and Kernels, PolyBio supported our imaging flow cytometer. In 2022, PolyBio supported my visit together with the funding from Mount Sinai to travel to Yale and to Mount Sinai and to Harvard to look at the microclot methods. And then obviously the biofilm research support was from PolyBio, Tom Usher, that started last year, and now Anel Thompson that took over. Tom has just completed his master's partially on biofilm work. So a real shout out for PolyBio. Without you, we wouldn't have been able to continue our research. Thank you.
Amazing, Resia. Every single part of that has been worth it and so productive. So especially when you came to visit our sites at Harvard, at Yale, that was just one of the better parts of just beginning a lot of collaboration. So thank you for walking through that. All right, we have Morgane Bomsel now. She is a senior researcher in HIV entry and the Laboratory of Mucosal Immunity at the Cochrane Institute in Paris, France. And she'll be talking about SARS-CoV-2 persistence and impact on long COVID megakaryocytes and platelets. Okay.
So, thank you. As you know, long COVID symptoms can be stratified in three categories: cardiovascular, neurological, and metabolic symptoms. And we have decided to, among the proposed mechanism, we have decided to focus on viral persistence. Ah, il marche pas. Okay. Indeed, the SARS-CoV-2 RNA and protein have been characterized in tissue from some of the long COVID patient, from 25 to 60% of the patient from the literature. And as an additional sign of viral persistence, spike has been detected in the circulation of long COVID patient, but also with a very large variety of frequency ranging from 5 to 60%. In any case, only viral components have been detected in this study, and whether it corresponds to infectious virus remains unknown. So our interest in megakaryocytes comes from our previous study in severe acute COVID patients, in which we could found that megakaryocytes were infected with SARS-CoV-2 in the bone marrow and also in the lung. This infected megakaryocyte replicates the virus, and during thrombopoiesis, the virus is inserted in some of the platelets produced, and we found that the virus inside this platelet was infectious. Infected platelets were mostly detected in non-survivors, as you can see here, but also in some of the survivors. And recently, we questioned this survivor having SARS-CoV-2 infected platelets several years after the acute infection, and found that this individual reported symptoms compatible with long COVID. We therefore hypothesize that SARS-CoV-2 persists in megakaryocytes and platelets forming a replication-competent reservoir which could contribute to long COVID symptoms. So to initiate this study, Dominique Salmon our collaborator sampled long COVID patient in Institut Fournier in France, together with Association Espoir. She already sampled more than 50 long COVID patient blood, and also sampled blood from COVID-resolved individual with no declared long COVID symptoms. So we first started to look at the presence of viral component in megakaryocyte, and as you can see here, CD41 positive megakaryocyte contain spike as detected in immunofluorescence, but also viral positive RNA as detected by in situ hybridization. This component can be found in roughly at least 2/3 of the patient, long COVID patient, but not in COVID recovered individual. This megakaryocyte also contain double-strand RNA indicative of viral replication in this megakaryocyte. We also use another technique to get a better quantification of the infected megakaryocyte, which is a fish flow cytometry. And indeed, again, we could find that at least 2/3 of the long COVID individual contain infected megakaryocyte to a reasonable extent. Furthermore, the frequency of total megakaryocyte within PBMCs in long COVID patient was much higher than that in COVID recovered or healthy donor, which is indicative of emergency megakaryopoiesis, as we have already shown in acute COVID individual, infected individual in which had SARS-CoV-2 infected megakaryocyte. So this results suggest that megakaryocyte can form or may form SARS-CoV-2 reservoir in long COVID patient, which may produce platelets sheltering infectious SARS-CoV-2. We therefore then look at platelets in this individual using the same technology. And as you can see here, for instance, the platelets, here that's the surface of the platelets contained inside platelets and exclusively inside the spike, but also positive SARS-CoV-2 RNA, as you can see here, and also replicative negative strand of the RNA, suggesting that the virus is not only inside the platelets, but may replicate inside platelets. As a quantitative level, long COVID platelets, platelets from long COVID contain infectious platelets, and in a frequency a bit less than 1%. This number can look very small or very limited. However, knowing the amount of platelets within the blood, it corresponds roughly to 10 to the 8 to 10 to the 9 infected platelets per liter of this long COVID individual, in contrast to COVID recovered individual. We then ask the question, is this viral component detected in platelets correspond to infectious virus? Therefore, platelets were stimulated to release the virus, and the viral resisate was added to reporter viral cell for 48 hours to allow infection of reporter cells. Infection of reporter cells were then detected by flow cytometry and looking for spike and RNA. And as you can see here, platelets from long COVID patient, or at least 2/3 of platelet sample could indeed infect viral cells who contain infectious virus, and the estimated amount of infectious viruses per million of platelets is around 30. We then found that platelets from a long COVID patient indeed form microclots, unlike blood or platelets from COVID recovered or healthy donor. And interestingly, in our case, this platelets aggregate do not seem to be induced by spike, because we can't find spike outside of the platelets in these aggregates. Finally, we used a specific SARS-CoV-2 specific spike, SARS-CoV-2 specific ELISA to detect spike in the plasma of this individual. And as you can see here, only 20% of the long COVID patient harbor significant amount of spike in their plasma. So to conclude, these initial studies indicate that indeed in long COVID patients, SARS-CoV-2 persists in megakaryocytes that form a viral reservoir. This megakaryocyte replicate the virus, which is included in platelets formed during thrombopoiesis. These infected platelets are likely activated and could explain the microclot aggregates we find in the blood of this patient and contribute to vascular symptoms from this patient. The infectious virus and probably the spike found in some of the individual could contribute to tissue damage and dysfunction, and also immune dysregulation reported in long COVID. And finally, I showed last year in the symposium that serotonin and its metabolites are decreased in long COVID individual, likely due to TPH1 deficiency. This defect in serotonin in platelets could contribute to dysfunctional neuronal signaling observed in long COVID. So we have now to validate this fully this hypothesis, and for that we will continue our biological analysis and compare or correlate, try to correlate this biological data with the clinical information clinicians are collecting in parallel. So I want to finish by acknowledging Fefan Hayes, a student in the lab who did most of the biological analysis shown here today. And also Association Espoir, and of course Dominique Salmon, with whom I collaborate for this project financed by PolyBio, who starts now for us. And also Professor Jacques Calbert, biochemist, Faroudi Boufassa, epidemiologist, and Elisabeth Kramer-Bordet, who are hematologists with whom I initiate this project. So thank you.
Great, Morgane. That was an incredible overview and such important work. With that in mind, we're going to go, for the sake of time, into the next talk. Hopefully we're answering questions in the chat when we can. And the next talk is relevant to your work. It's Johan Van Weyenburg from the Resia Institute for Medical Research in Belgium. He's talking about digital transcriptomics to capture SARS-CoV-2 RNA persistence in blood, plus a patient-led Paxlovid trial. Thanks, Johan.
Thanks, Amy, for the kind invitation. I'll start sharing. Okay, so in short, yes, we will be talking about blood transcriptomics, because we believe, first of all, they can show us viral persistence, and it also might lead to what we urgently need, that is long COVID biomarkers and probably also therapeutic targets. And all the data I'm going to share with you today is from a real world long COVID cohort in Belgium. So of course, I don't need to introduce long COVID to this audience. Several people already spoke about biomarkers that do come in different flavors. And I believe as a whole, everybody is right on the biomarker front. We do need them in several types, and I think all our data are in fact complementary. So, but I think the first and most pressing societal and clinical need is to have a diagnostic biomarker so that we can say, so this is a real case, this is a control. And ideally that would then be based on viral persistence. And of course, the question is, we can do several biopsies. You can do CSF, you can do a bronchial lavage, you can do intestinal biopsies, but I think for clinical purposes, and especially if you want to have a biomarker that you can frequently test for relapse remission before and after treatment, it would have to be non-invasive, such as blood or saliva. So that's the first one we need, but then of course we do need biomarkers to guide treatment, whether that is antiviral, anti-inflammatory, or anticoagulant, in line with all the data we have seen and will be seeing today. But then I want to draw attention that we also do need, when we have trials, have these predictive biomarkers that actually help us predict clinical evolution of the patients, whether it's by imaging or by these so-called patient-reported outcome measures. And I do believe so for all of them, they would have to be preferably non-invasive and ideally affordable. So why transcriptomics? Again, omics have been used by several research groups, and they all have their qualities. But I do think transcriptome has the advantage of being highly dynamic, and it also gives us a snapshot of the ongoing host-viral interplay, because both the host and the virus express RNA molecules, and they are also tissue-specific, in contrast to, of course, your DNA, which is fixed in every single cell. Results of these digital transcriptomics, we've seen volcano plots before. They look always quite busy because they actually summarize here 800 different RNA molecules. Or if you can see my arrow, hopefully I'll take you through it. So in this axis, we see the fold change. So everything you see on this side is higher in long COVID patients. Everything on the left side is higher in the controls. And up and down just means that it's more significant or less significant, statistically speaking. As you can see, statistically speaking, this Fin mRNA was the strongest in long COVID patients, and we'll get back to that molecule later. But of course, we want to pay attention to all these red dots because these are the viral targets. Several of these viral RNAs, nucleocapsid or 7A or 3A. We also see the protease, which is a target of Paxlovid. We also see the antisense RNA we spoke about earlier as a marker of viral replication. But we also find several host proteins associated, such as the receptors, ACE2, TMPRSS2, and proteases. And from the immunological point of view, in green, we see several markers of memory B cells, and especially we do find they express more IgE, which is then linked to the allergies and asthmas that are related very often by patients with a sudden onset after their long COVID diagnosis. And then last but not least, just following up on Morgane gave a perfect introduction, there are several platelet genes that are also highly expressed, and they in fact correlate significantly to the viral RNA levels we see, which provide a link to coagulation, so microclots, and definitely the viral reservoir just presented by Morgane in the previous talk. So zooming in on these viral RNAs, these are the results summarized of 48 long COVID patients with exactly matched controls. Matching means age, sex, but also time after acute COVID. I think that's really important. Both the patients and the controls were sampled more than 2 years on average after their acute COVID episode. They were also matched for vaccine status and number of comorbidities. As you can see, really macroscopically in red, see that all of these RNAs are actually much higher in the patients as compared to the controls. And the highest one was in fact the antisense RNA, again hinting at replication. So we found it in roughly 65% of the long COVID patients. That again resonates with the previous talk, like roughly 2/3 of the patients will present this, and this is in the whole blood. quite surprising, in fact, was that spike levels on the whole were lower, than in fact not significantly different, which is good because then, of course, we can nicely discriminate between vaccination and real viral infection. So the matched controls, they do have some levels. It is quantitatively and qualitatively much lower. And if you just quantify here the total viral load just by summing all of these viral RNAs, you see that there is just 1 in 12, so 8% of the controls that pass this cutoff. So this means that we in fact have a candidate diagnostic biomarker for long COVID. If we only use the antisense RNA, we get a nice predictive ROC curve. If we do the same for the Fin molecule, which I said was the most significant immune marker, we also get nice prediction. But if we combine both of them, we get in fact a combo biomarker that can predict with over 90% sensitivity and specificity, meaning we can drop the false positives and false negative to less than 10%. So another biomarker that I think would be clinically relevant for the patients, I think also to avoid the stigma that's been linked to especially the mental health question, the reported anxiety and depression. So we're talking a lot about these COOP charts, which are a very handy and visual way of measuring patient-reported outcomes. And for now, I would like to focus on this one. So zooming on the one that's called feelings, actually summarizing on a visual scale very rapidly from 1 to 5. Patients take usually less than 2 minutes to fill out these forms, so it's handy for both the clinician and the patients. So this meaning in the last 2 weeks, did you feel bothered by emotional problems such as feeling anxious or depressed? And as you see, lower scores mean that patients do better, higher scores mean that patients do worse. When we do actually plot the patients in the worst scales, we see that these patients had much higher levels, again, of the SARS-CoV-2 antisense RNA. We also found that at lower levels of immunometabolism scores, which is just a summarized score from several genes that make up the metabolic activity, again, in the whole blood. So we believe this is a different flavor of biomarker that can help us definitely relieve the stigma, because in fact patients are relieved when they get these results. They say, wow, it's not in our head, we actually see something real happening in our blood. But of course, the important question is, can we target this? And that's the next thing I would like to talk about. This is patient-led design clinical trial, which we simply called a grassroots clinical trial, in which we used Paxlovid. And we have to say, of course, this is off-label use. This was not an academic or a pharmaceutical-sponsored trial, in keeping with this short communication we wrote before with our clinician, Mark Jamul, and patient representatives. So in fact, we're proposing clinician-patient pairs as central to this new type of doing very fast clinical trials. It's a small trial. We only have up to 20 patients now. I think what was interesting is that we have a personalized list of long COVID symptoms for each individual patient on a visual scale. It was online and cloud-based, so we have real-time info, including the identification of adverse events. So primary outcome was, in fact, the average % of drop in these personalized symptoms. Secondary outcomes, again, the patient-reported COOP charts and our transcriptomic biomarkers. So this is just an example of a heat map of what a patient actually reports over the time of day. So each column is 1 day. This is before treatment. And as you can see, patient just choose from drop-down menu all of the symptoms that they have at the start, as fatigue, post-exertional malaise, insomnia, headaches, memory troubles, vertigo, etc. So you can rate this from a scale from 0, absent, to 6, extremely strong. As you can see, this patient is a non-responder. Over time, there were some fluctuations, but roughly symptoms remained around 100%. There was no increase. This is, in fact, as you can see, a responder. So as you can see at the start, several symptoms are scoring red to orange. As you can see, it gets a lot more green over time. And in fact, at the last day of treatment, there was more than 80% drop in these symptoms. This was in fact a very good responder. So when we plot all of the patients together that we have outcomes for, we see as a whole, roughly 1/3 of clinical improvement. So 1/3 of drop in these personalized symptoms. And you can see this was true at the last day, but also if we take just the mean over the entire treatment course. So secondary outcomes, again, back to the Coop chart, and now we're taking just the Coop chart total. So all of these fitness, social activities, feelings, etc. And when we plot these, you can see that patients that opted for Paxlovid, it was an opt-in, in fact, had higher clinical scores at the beginning, so we're doing worse. But there was a significant drop in these group chart-related outcomes. And as a control, we used the rest of the cohort, which had only anticoagulant therapy, so almost 50 patients. And there we can see there was strong variation, but there's no significant decrease in symptoms using the same scale. And both groups are currently still being followed up. We have more than 1,000 person months of follow-up for now. We'll keep going. Then going as a last part to the biomarkers, what did we learn from this small trial? In fact, we did see some light antiviral activity. We saw a decrease in several viral RNAs, nucleic capsid, ORF7, ORF3, the protease, but to our surprise, we didn't see any difference in the antisense RNA. So there might still be replicating virus, and of course, that might help explain why we see Paxlovid rebounds, either in acute COVID, but also long COVID patients often report that. So first of all, can we do more grassroots trials, and especially can we maybe try combination therapy, try different antivirals, maybe use a combination with anti-inflammatory or anticoagulants? I think that might get us forward. So I'll go quickly to the conclusion. So I believe what digital transcriptomics can show is first of all, that we can document systemic viral persistence in the blood up to 2 years after acute COVID, which is a possible diagnostic long COVID biomarker, but maybe also biomarker for anxiety and depression. And of course, all of these need to be validated independent cohorts. So this is an open invitation to all the colleagues worldwide. So if you can, please share the samples with us. We'd love to validate. And of course, this goes both ways. We would love to share our samples with you to help validate your favorite biomarkers. I believe also what the grassroots trial has shown us is that maybe these personalized clinical scores are a bit more sensitive than usually very long questionnaires or clinical scales. They need to be validated, of course. This goes for the clinician and patient scales, but I do think these very rapid Coop charts are time effective, less energy consuming, definitely. Having been a patient myself, filling out these questions can be really tiring, and of course can allow patients sometimes to drop out or being lost to follow-up. So I think that's also an important item to consider. Quickly acknowledging all the people that actually helped us realize this work. First of all, big shout out to Mark Chamul, the clinician of our team who's coordinating all these efforts, the patients, controls, and Belgian Patient Associations, especially Helena and Tommaso here, the colleagues from our institute, especially Soraya Maria, first author in several of the papers and preprints, and the colleagues abroad, so from Paris, Dominique, Lisa, Peter from Karolinska, Jean-Laurent from Paris as well, and of course, last but not least, PolyBio for the very generous funding. And thank you for your attention.
Great. Thank you, Johan. Really cool results. I'll move to our next speaker, which is Dr. Marcus Buggert. He's an assistant professor in the Department of Medicine at the Karolinska Institute, and he'll be talking about the impact of SARS-CoV-2 on long COVID gut and lymphoid pathology.
Right. Thank you. Thank you, Amy. Can you see it?
Yes.
Perfect. All right. Thank you. First of all, thank you very much for inviting me to give this talk. Like Amy said, now what I'm going to do is talk a little bit about some ongoing projects that we have funded through PolyBio, particularly LymphPASC, which is a project on exploring lymphoid immune activation in the PASC in long COVID patients. But I'm also going to, towards the end, go a little bit through some results also when it comes to biomarkers and specific symptoms which are linked to systemic biomarkers. So I'm not going to do a big introduction when it comes to what long COVID is. Instead, what I thought I'm going to do is to show this picture. And essentially what this picture is illustrating is the fact that viruses in general, they affect multiple tissues in the body. And SARS-CoV-2 is, of course, no exception for that. And although most people associate SARS-CoV-2 with respiratory disease, it has become very clear that the virus can affect a variety of tissues beyond the respiratory tract. This includes, of course, gastrointestinal tract due to high ACE2 levels, but also the cardiovascular system, nervous system, lymphoid tissues, etc. And this broad presence suggests that a virus might have both immediate but also long-lasting impacts on various organs. And to fully understand if and how persistent SARS-CoV-2 contributes to pathology in long COVID, it's crucial to investigate specific tissues. And throughout these years, what we've been doing is to collect samples from the lung, from cerebrospinal fluid, etc. But now I'm going to focus a little bit on a study that we're conducting and the plans that we're going to conduct for LymphPASC, where the idea is to focus on detecting persistent viral antigens in the GI tract and upper respiratory tissues, with a focus on tonsils to explore how these sites might contribute to ongoing immune activation symptoms in PASC. And the goal essentially is then to determine if persistent SARS-CoV-2 viral antigens or other local cues in the GI tract and tonsil drive systemic immune activation and contribute to chronic symptoms observed in long COVID. Okay, so essentially the idea is quite similar between these two different names. The first aim is investigating, but there are two different sites which we're looking at. The first one is to investigate the persistence of SARS-CoV-2 in gastrointestinal cell subsets. So that will include both looking into immune and non-immune cell subsets and its association with transcriptional, microbial, and inflammatory changes. And then number two is to determine if SARS-CoV-2 antigens in lymphoid tissues, and specifically tonsils, we will have a large collection of these samples lead to sustained T-cell activation. So the hypothesis for the first one is that persistent SARS-CoV-2 in the GI tract contributes to local cellular changes and systemic inflammation. And to this end, what we have done is to collect samples, or it's ongoing still, but and I'll show a little bit so far what we have done. But essentially what we do is that we collect biopsies. So that's 6 different biopsies per site. So we collect them from sigmoid colon, cecum, and terminal ileum of patients with long COVID, 15 of those, and then 6 age-matched healthy controls, which with previous COVID, but without any symptoms. And essentially what we have been developing is a system which to some degree has been used from the HIV arena, and that is that we can use now single cell sequencing and imaging techniques, but particularly single cell sequencing for identifying SARS-CoV-2 reservoir in GI cell subsets by sequencing one single cell at a time. And essentially, the idea is illustrated here in HIV infection, where we and other colleagues, this is data from Joshi Ho's lab, where essentially they were able to pinpoint here in red different cell subsets that are infected with the virus. And the idea and what we're doing right now is to do the same thing for SARS-CoV-2 as well throughout the different sites of the gastrointestinal tract, and both looking at immune and non-immune cell subsets. So essentially, that's the first idea, is to use these new type of technologies to identify specific GI cells that harbor SARS-CoV-2 RNA and protein probably in the future. This is going to be done by 10x, but also imaging at protein level. And then we also, depending on what we find, we will also have the chance with these type of technologies to analyze transcriptional changes in immune and epithelial cells, but also fibroblasts and other type of immune subsets that or non-immune subsets that are present in the gut. We'll also assess if SARS-CoV-2 specific CD8+ T cell cytotoxicity is changed in the GI tissues. And the reason for why we're interested about that is that we have data indicating that when you look specifically for CD8+ T cells in tissues, and particularly in the gastrointestinal tract, these cells are going to harbor lower levels of cytotoxicity in comparison to many other tissues, and particularly in the blood. And that means that even if you have SARS-CoV-2 specific cells there and you have persistent virus, they may not be able to actually eradicate these viruses. And so far, without showing any data, the data is suggesting the same when it comes to the long COVID and SARS-CoV-2 as well, that these kind of dogmas sustain the same. And finally, what we're doing is that we're looking into microbiome and barrier functions to examine microbial shifts by 16S and epithelial barrier integrity. So far, what we have done is to collect tissue samples from 23 out of 30 donors. We have conducted fresh facts and 10x genomics analysis on all collected samples. And sequencing has begun a first set of analysis will be conducted very soon, with the idea of generating probably one of the biggest, if not the biggest, resource for single cell analysis on long COVID versus healthy donors within the next month or so. So that's aim one. Aim two, it's more or less the same in nature, but the difference here is that we're looking into tonsils. And the idea here is to look into persistent SARS-CoV-2 antigens in tonsils and understand if that potentially can trigger ongoing T cell activation. And the idea behind this is that Tim and others has been showing in the past that potentially tonsils are shining up for increased T cell activation, but there's also studies on that showing that kids particularly can have persistent SARS-CoV-2 in tonsils for a long time. So what we have in my group is a big cohort of tonsil samples from I would say it's probably over now 220 donors. And what we can do is that we're dividing them up into 3 different groups. So we can divide them up into individuals with prior SARS-CoV-2 infection, prior SARS-CoV-2 just vaccination without having the infection, and those without any infection or vaccination history. And then what we have done is that we are analyzing SARS-CoV-2 RNA in tonsils, and we visualize the virus with multiplex imaging, such as with Maxima instruments, which will allow us to basically look into a little bit similar with 10x, but with imaging instead, to get a map of exactly which cells that may harbor the virus here as well. Furthermore, if we find virus, the idea is to include MSC class 1 and 2 tetramer stainings to detect localized immune activation in SARS-CoV-2 specific T cells from tonsils in the blood. And the reason for why we think this is important is that we have been able to show that before, that particularly within tonsils, you actually have more antigen-specific cells. So again, against SARS-CoV-2. So again, this sort of implies that if you're just looking to blood, you might miss some of these changes. And the reason for that is that you have these local resident cells recognizing SARS-CoV-2. And the idea is to look at activation signatures in those and try to link it to SARS-CoV-2 persistence. To simulate also this entire scenario, what we're doing is to, and what we have done in my lab is that we have we're assessing right now if persistent virus can drive SARS-CoV-2 T cell activation and proliferation to more mimic in vivo conditions. And finally, what we're trying to do is to link it also to specific T cell receptor clones. So essentially the idea here is that during multisystem inflammatory syndrome, you have an expansion, potentially superangiogenic effect. And what we're trying to do now is to link it and see if we can find it both in tonsils, but also systemically as a proof of concept for that this might be a driver potentially of long COVID symptoms. So the status of this is that, like I said before, we have already collected the tissue samples from all the donors, and we have conducted FACS analysis on most of these individuals to identify SARS-CoV-2 and other virus-specific T cell populations to look at their activation profiles, and we also started viral quantifications. And basically, the idea with this study or these studies is that they will provide data to determine whether persistent spike antigens and tissues contribute to ongoing local and/or systemic T cell activation, potentially fueling long COVID symptoms. So finally, I'm not going to be worse than other people. I will show some data as well, and not only show things that we will do, but I thought it was good for you to get a grasp on some of the things that we're planning on doing as well. But I also wanted to show you something which is related to biomarkers as well. So one of the ideas which we had from the beginning of time when we recruited long COVID patients were to understand which distinct long COVID symptoms are most closely associated with systemic protein alterations. So in order to answer that question very shortly, what we did, where we were working together with David Price, we have included 150 donors with or without long COVID, both from UK and Sweden, and we used this very sensitive assay where we can look into over 3,000 protein assays for exploratory proteomics from Olink. And it can measure everything from markers of inflammation, neurology, cancer, metabolic, cardiovascular features, etc. And to make a long story short, essentially by adjusting our p-values and looking into the symptom that seems to be best correlated, if one would put it like that, to a distinct or a highly elevated plasma protein profile is specifically patients who have severe breathlessness. So these are different type of volcanoplots, which are showing that multiple markers for cardiometabolic, inflammatory, oncology, even neurology symptoms, independent on what to look at, for these different tiers of markers, you can identify that particular breathlessness is highly correlated to a distinct protein profile. And some people might say, does that matter at all? But what's really interesting is that many of these proteins are actually linked to public databases, proteins that are usually elevated in collapse of lung, lymphoedema, shortness of breath, etc. And finally, these specific to understand some type of etiology behind it, what we've been able to do is to run both GSEA and pathway analysis. And essentially what these are showing is that we have different pathways which are involved in TNS and FAST pathways that's linked to cell death, but also chronic inflammation. We have pathways involved in cell growth and survival. Other ones were involved cell migration and cytoskeletal dynamics, but particularly one that is standing out a bit for us is the fact that many of these pathways are also linked to atherosclerosis and particularly platelet activation. So that will summarize particularly this last part by saying that there's limited signs of significant immune perturbations in individuals with long COVID. That was something that we have shown before. But what we can see through ultrasensitive proteomics is it demonstrates clear signs of increased cell proliferation, inflammation, platelet activation, and apoptosis. And the reason of this is still unknown. So with that, I'd like to thank my group members, particularly Millie and Hautau, who's doing a lot of the work in the lab particularly on the first end of the analysis. Other people from Karolinska University Hospital, patient health volunteers, and of course, PolyBio. Thank you very much.
Yeah, thank you, Marcus. All the methods that you're using together so intelligently. It's really cool to see and to overview. So thank you. I'll introduce our next speaker, who is Michela Locci. She's assistant professor in the Department of Microbiology at the Perelman School of Medicine at the University of Pennsylvania, and she'll be talking about lymph node immune responses in long COVID.
Well, thanks so much for having me. It's a pleasure to be here today and to give you an update on this project that I already presented in the past symposia. And so I think we have nice updates because the project is progressing nicely. So starting with some introduction. So my lab studies a particular type of adaptive immune responses called germinal centers. And so they take place in lymphoid tissues, like in lymph nodes. And there, the B cells that are specific for a pathogen like SARS-CoV-2, in response to the pathogen, expand and also undergo selection for the cells that are really high affinity, so high quality. They can then differentiate into plasma cells and memory B cells. So now plasma cells secrete antibodies, some can be binding, some neutralizing, and so they're important for not only preventing infection, but also for once the infection is established to clear the virus, help clear the virus. And memory B cells also, once recognizing the antigen again, they can quickly differentiate into plasma cells and so contribute to the wave of antibody produced. So are germinal center responses alter in long COVID? We don't really know. So before this study, there is no previous publication addressing directly this question, but there are indications in the literature suggesting that some alteration in humoral responses, so specifically in antibodies, are present in some cohort, as described by different groups, including groups that are part of this consortium. And so this led us to make the hypothesis that perhaps in healthy convalescents that are infected and then go on and recover, germinal center responses are normal, and this is associated to efficient clearance of SARS-CoV-2. Whereas potentially in long COVID, germinal center responses can be either quantitatively reduced or their quality may be affected, overall leading to antibodies that are lesser quality and that are not capable of contributing to the clearance of the virus. And so how do we test our hypothesis? So we heard before from Marcus about the importance of studying cells in tissue, and germinal centers are no different. So they are present in tissue and need to be studied there. So an approach that we have deployed in the past to study germinal center responses to vaccination is minimally invasive and is called fine needle aspirate. So in the case of vaccination, the vaccine is injected in the deltoid muscle, and so the draining lymph node that we are targeting is the axillary node. And basically, you visualize this lymph node with an ultrasound approach. And then, as you can see in this video, we enter with a fine needle and we target the cortex of the lymph node multiple times. And this is sufficient, applying some light aspiration to recover enough cells for downstream applications. So now, what I've shown you in the previous slide is a study where we targeted the axillary node. In the setting of long COVID, we are trying to target cervical L2 lymph nodes, which are located in the neck, because this lymph node drain the oropharynx, which is a site where SARS-CoV-2 can be enriched. This has never been done before, so the application of FNA is to study longitudinally germinal center responses to a viral infection, but we were really excited to try. And so we began initially collecting FNA sample from our PEN cohort, including healthy convalescent and long COVID. And initially we recruited with the help of Ben Abramoff at PEN running the long COVID clinic. And also key was the help from PolyBio in advertising and recruiting more patients. Later on, a few months ago, we started collaborate more actively with Michael Peluso also, and we are receiving currently FNA samples obtained at UCSF. And so this has been also a huge help. So we are certifying these FNA samples into two major time points. So we certify them into T1, which is any time between 3 weeks to 6 months post-infection, and T2, which is any time between 6 months to roughly a year post-infection. We have also later sample, but for the sake of today's presentation, I'm not going to talk about those. Briefly skimming through the clinical manifestation of the donors of our cohort, which are predominantly female donors. No surprise here. So there are manifestations are highly heterogeneous, and something that I want to highlight is that our cohort seems to be a reach for a potentially neurological manifestation. Oh, sorry. As you can see, there are many patients presenting with brain fog or concentration or memory loss. And also pretty much most of them present with fatigue. So now I presented already some of these data, the previous symposium. Now we were able to add several data points, but basically the way we have begun this analysis is by flow cytometry, analyzing first sample and try to answer the simple question, are there germinal center B cells that are specific for SARS-CoV-2 in the cervical lymph nodes? And the answer is yes. We can track these cells by flow cytometry using fluorescently labeled tetrameric probes. As you can see here, we use a combination of spike and RBD or spike and S2, and we can classify GCB cells as SARS-CoV-2 specific. So now, what did we find? So in healthy convalescent, as expected, you can observe nice GCB cells early on that then decline over time, and they're predominantly gone by 6 months, so by T2. And that's expected because at that time in healthy convalescent, the virus is cleared. But a completely different scenario emerged in long COVID, where now you can see that the germinal center responses targeting SARS-CoV-2 are severely hindered early on. And then if anything, they seem to try to catch up later on, but they're still very, very low in comparison to the peak of the response in the healthy convalescent. So severely hindered germinal centers. What does this mean in terms of the downstream responses? So looking at serology, we find that binding antibodies targeting SARS-CoV-2, they seem to be slightly lower in long COVID early on. And then what's really clear is that their durability is reduced in comparison to the healthy convalescent. And trying to assess better the quality of these antibodies, we look at their capacity to neutralize SARS-CoV-2 pseudoviruses in vitro. And here you can see data from a pseudovirus containing the D614G mutation, so one of the early type, but similar data are present also when using a more recent strain. And you can see that at both time points in the long COVID group, there is a substantial reduction in the amount of neutralizing antibodies. And interestingly enough, this data shows that essentially, there is a nice correlation in the healthy convalescent between the amount of neutralizing antibody produced and the size of the germinal center response. So the more antibody, the bigger the germinal center response is targeting SARS-CoV-2. And this correlation is not present at all in the long COVID group. So we also were able to do site-seq analysis in these FNA samples, including single-cell transcriptome, including probes to test the specificity of these cells, and coupled with BCR sequencing. And we have done that in 10 long COVID and 10 convalescent, equally split between T1 and T2. And briefly looking at the transcriptomic, you can see three major groups of B cells. So there is a small cluster of plasma cells, a larger subset of memory B cells, and GCB cells. Briefly, the GCB cells, if you look at the differences between the two groups, you can appreciate, especially at time point 1, what I already showed you by flow cytometry. So these analyses nicely recapitulate the fact that there is a substantial reduction in germinal center B cells early on in long COVID in comparison to convalescent. There are also transcriptional abnormalities that, for sake of time, I don't have time to show you, but what I really want to show you is some BCR data, because by looking at the sequence of the B cell receptor, we can essentially establish the clonal size, so clonal expansion of certain clonotypes of B cells. And what's very clear from these analyses is that both that you look at total GCB cells or that you focus on the SARS-CoV-2 specific GCB cells, you can see that all time points, there is a significant reduction in the clonal size of the GCB cells in long COVID in comparison to convalescent. So we think this means that it's a reduced clonal expansion in long COVID, probably due to altered selection process in the germinal center. And so now we're digging more into the mechanism behind this observation. I think I missed a slide. Sorry, I think I missed a slide on B cells. I had also a slide on memory B cells showing abnormality in transcriptome as well. Also there, specifically, there is a very strong interferon signature. And yeah, so in conclusion, these are the major findings that we have so far from this study. So major alteration in germinal center responses, a strong reduction combined with reduced quantity and quality of SARS-CoV-2 targeting antibodies, and with alteration in the signature of transcriptional signature of memory B cells. We still have a lot of work to do. For instance, we need to understand whether the GCB cells have a reduced affinity for SARS-CoV-2, and whether, importantly, there is an association between these altered germinal centers and the persistence of the virus. Last but not least, a huge thank to PolyBio for making this study possible, and to all the study participants that generously agreed to enroll in our work. This study was led by Hannah Sharp in my lab, and she was helped by Joy Chu for some analysis. We received help from Amelia Spolano and the Wister for the BCR analysis, and we are collaborating with John Ware's group for a transcriptomic analysis. Binding antibodies were measured by the group of Scott Hensley, and I already acknowledge Michael and Ben for helping us recruiting and obtaining FNA samples. And I'm gonna stop here.
Great, Michela. It's really cool to see the progress since the last symposium. And as always, just analyzing the lymph node samples is so innovative. So it's really cool to see the progress. Yes. So I'll introduce the next speaker, who is Akiko Iwasaki. She's a Sterling Professor of Immunobiology and Molecular, Cellular, and Developmental Biology at Yale University. She'll be talking about probing the role of endogenous and latent viruses in long COVID.
Okay, wonderful. So I'm delighted to be here today at the 2024 Fall Symposium for PolyBio. So today I'll be talking about our current work on probing the role of endogenous and latent viruses. So we are all here to pursue how we might be able to detect and treat long COVID. And there are 4 possible root causes that we are pursuing. And some of these have been already described. But today what I'd like to focus on is the possibility that latent viruses that reactivate due to acute COVID infection is responsible or contributing to the diseases within long COVID. And so in terms of latent viruses, there are many viruses that we live with, as well as there are viruses that have integrated into our genome over the ancestral integration. And those are the kinds of viruses that we're going to be focusing on. And in terms of latent viruses, we've shown with other groups that Epstein-Barr virus reactivation is linked to long COVID. About this, Jim Heath's team earlier on showed that EBV viremia is one of the 4 predictive factors for developing long COVID. And Michael Peluso's team, as well as our team, have shown that there is antibody reactivity to lytic EBV antigens that is indicative of recent reactivation of EBV in people with long COVID. But EBV is just one of many virome that we live with. The most sort of insidious or prevalent virus that we live with is the endogenous retroviruses. So all of us humans carry endogenous retroviruses as well as other retroelements in our genome. And EBV is very prevalent. 95% of adults carry latent forms of EBV. So we wanted to understand what are the impact of these human virome on diseases like lung. Here we are looking at our genome composition, and only 1.8% of our genome encodes for protein sequences that most of us study. However, over 40% of our genome is occupied by endogenous retroelements, such as the LTR retrotransposons. These are the remnants of retroviruses that have integrated in our ancestors, as well as this line 1, which is a non-LTR retrotransposons that still have active transcriptase, as well as ability to transpose itself from one location to another. And this is still activated even in humans. So retroviruses can integrate into our genome, and this is an irreversible process. So once the reverse transcriptase converts the RNA of the retrovirus into DNA, and that DNA gets integrated into our gene. And if that happens in germline cells, there's no way for us to get rid of it if it's vertically transmitted to the offsprings. And so we've been studying the role of these endogenous retroviruses. In humans, there are no active replicating endogenous retroviruses, but we contain upwards of 8% of our genome expressing a variety of antigens from these endogenous retroviruses. So a few years ago, we started to develop tools, molecular tools, to be able to map RNA sequencing reads to various endogenous retrovirus loci within our genome by making a platform called ERVMap. And this enabled us to analyze a genome-wide transcription of human endogenous retroviruses, as well as identify antibodies that target some of these antigens that are encoded by endogenous retroviruses. And we indeed identified pathological immune responses envelope of one of these endogenous retroviruses. This work was led by a former postdoc, Maria Tokuyama, with many of the collaborators that are listed here. So using the ERVMap technology, Maria sequenced a deep RNA sequencing of PBMC isolated from lupus patients. And so the lupus patients here are indicated in red dots. Healthy controls are in black dots. And you can see that, in fact, we found over 100 different endogenous retroviral loci that were elevated in PBMC from lupus patients. And what we found was that this particular endogenous retrovirus called K10, this is highly upregulated in people with lupus. So Maria cloned the envelope that is encoded by this locus and tested for antibody reactivity from people with lupus versus healthy controls. And even though the antibody binding envelope was seen in lupus patients, healthy controls also had reactivity to endogenous retroviral envelope. And we've tested all the different isotypes, and they're comparably detecting these envelope. So this is interesting that there's something that we are continuously making antibodies to these envelope proteins. However, antibodies have two different regions. One is the variable region that most of us care about, which is the antibody binding site, antigen binding site of the antibody. So this variable region specifies the reactivity of a particular antibody. However, there's also this region called the FC region of an antibody that determines its function by binding to receptors that are expressed by a variety of immune cell types. And in fact, effector function of antibody is dictated by the kinds of FC that they contain. So not only can antibodies bind to the pathogen and neutralize them, but it can also engage phagocytes to induce uptake of pathogens, as well as NK cells to kill infected cells through antibody recognition. And also by fixing complement, it can induce not only the lysis of the microbe, but also phagocytosis of the microbe by opsonizing them and neutrophil activation. So we wanted to understand whether these the phagocytosis inducing capability of antibody is different in people with lupus and healthy control. And indeed, what we found was that antibody isolated from lupus patients, when conjugated to endogenous retroviral envelope proteins, elicit this increase in antibody-dependent neutralized phagocytosis, as well as this neutrophil extracellular net formation that is only seen with IgG from lupus patients. And this is also quantitated here. So these results led to our understanding that lupus patients make antibodies against the envelope of endogenous retroviruses that can engage phagocytes such as neutrophils to potentially leading to the pathogenesis of lupus. So what we're doing right now with help by PolyBio is to collaborate with a company called Ceromics that specializes in the FC effector function analysis of antibodies. So we're now collaborating to understand whether antibodies specific to EBV or endogenous retroviruses can engage pathological impact when bound to this cognate antigen with their help, both biochemically as well as looking at cell activation by these antibodies. And as Amy already introduced, we're also very happy to collaborate with PolyBio supported clinical trial on Truvada and Maraviroc, which are HIV drugs that have been used in many patients with HIV. And we are now repurposing these drugs because Truvada, for example, it's an RT inhibitor, and it has been shown to interfere with also EBV replication. And it also has inhibitory activity against the RT of endogenous retro elements, particularly line 1. Truvada is also in clinical trial to prevent ERV-K activation in ALS and EBV reactivation in MS. And Maraviroc is a CCR5 inhibitor, which blocks inflammatory cell recruitment to different tissues. So we've already started enrolling patients into these 3 arms, the placebo control, Truvada, and Maraviroc. And we will be treating patients for 3 months and measure before, after, and follow-up time points, variety of patient-reported outcomes, as well as deep immune profiling. And from our team, study lead is Claire Laxon. So I want to end here by acknowledging this amazing teams that I have the pleasure of working with. Particularly, this project is supported by PolyBio and led by Claire, Simone, Sasha, and Bornali from our team, and David Petrino on the Truvada trial, as well as Amy and Mike on the various different projects that we're working with. So thank you so much.
Great. Thank you, Akiko. It is definitely cool to work with you on those projects, especially the trial. So we'll see how it goes. I'm very excited. All right. And then I'll introduce our next speaker, who is John Wary. He is the Richard and Barbara Schifflin, President's Distinguished Professor and Department Chair of Systems Pharmacology and Translational Therapeutics at the University of Pennsylvania. He'll be talking about T cells as biosensors of viral persistence in long COVID.
Okay, great, thanks. Yeah, it's super fun to be here. We're very excited about these projects and also very excited about working with the PolyBio group and this wonderful extended collaborative group of people around the country. So I want to tell you a little bit of the work we've been doing, looking at long COVID and trying to understand whether we can actually use the immune system to help tell us what's happening. I think it's requisite that everybody shows this slide or some version of this for these talks. The area that we want to focus on is partially overlapping with what Akiko introduced. And among the various potential mechanisms underlying long COVID, we were very interested to understand or begin to test the hypothesis around persisting virus or viral pathogenesis as one of the drivers. We have a long history of investigating and trying to understand the relationship between persisting viruses and the immune system, and thought we could apply some of that knowledge to starting to investigate the potential role of microbial cofactors or factors in long COVID. So this is all work done by Mark Painter, who's been a great lead on these projects. What he's really done is try to use antigen-specific or virus-specific T cells as biosensors to tell us whether there's persisting viruses, persisting SARS-CoV-2, or persisting change in other viruses in long COVID patients. So the system he set up, let me spend just a minute or two on this slide, is to develop HLA tetramer reagents to track individual virus-specific specificities over time. So each of these HLA molecules is bound to a peptide, either from SARS-CoV-2 or other viruses. That molecule then is multiplexed, basically tetramerized, with a streptavidin with a fluorochrome. For each of these, we include a different peptide. In this case, I'm just illustrating peptides from the SARS-CoV-2 spike protein. And then when you complex these, you can detect rare antigen-specific cells by flow cytometry. Here, and in what I'm going to show you, we always use double labeling. We use a tetramer on 2 different fluorochromes, so you can get rid of some of this background that you see in the single positive events here. After vaccination, people have a nice T cell response of antigen-specific T cells in the red bubble here that recognize spike. But after vaccination alone, you see no antigen-specific cells for the other proteins from SARS-CoV-2. However, when individuals experience a breakthrough infection, now they have an amplification of the spike response because these cells are getting boosted and presumably helping to provide some protective immunity, but you do see priming now of T cells responding to ORF3A, replicase, or nucleoprotein, nucleocapsid specific peptides. So we've built a whole panel of these tetramers. A few of them are listed over here, but let me show you just a little bit more of how this works. One of the great things about this is that we can now fish out those very rare cells in the peripheral blood and watch what happens to them during various events. In this case, I'm showing you what happens over time during a breakthrough infection in people that have vaccine immunity that then gets recalled during a breakthrough infection. Engage on these tetramer positive SARS-CoV-2 specific cells. And what you see, and I'm going to show you a little more of this, is that by day 7, many of these circulating spike specific cells are highly activated. They upregulate the activation marker CD38 and begin to proliferate as measured by Ki67. If you track this longitudinally over time, and we're capturing people on the day of very first symptom and test positivity, we can actually track what happens in very tight kinetics for a human experiment here. And what you'll notice is that even by day 2, if you just look at the CD38 differences, you're starting to see that these antigen-specific T cells can be activated and report that exposure to antigen very sensitively within 2 days. The signal peaks following breakthrough infection around day 7 and then wanes back down. This is consistent with the clearance of virus during breakthrough infection. I'm not showing you some of that data. And you can just track these kinetics. One of the things that you may notice if you stare at this data, as long as Mark and I have, is that there's a different kinetics. And so whereas Ki67 has a very narrow peak, peaks very tightly between day 5 and day 10, with really the peak around day 7. CD38 starts earlier and actually has an extended duration, and that's true for different markers depending on which marker you analyze. So we can get sort of different windows of reporting of what's happened in the host using T cells as sort of a sensor for that activity. So what Mark did is he built a large panel of tetramers, not just for SARS-CoV-2, but he also built tetramers for EBV, VZV, CMV, 3 persisting herpes infections, as well as a control influenza virus, and use this panel of tetramers to start to investigate what happens in long COVID. We can resolve all of these in a single flow cytometry panel because of the double labeling. These different populations basically show up along the diagonal depending on how you do your gating. So the different colors here just represent all those different tetramer positive populations. So I'm not going to belabor a lot of the things you can learn from this. There's a lot more data based on the high dimensional cytometry about the different flavors of T cell differentiation responding to influenza virus, a persisting herpes virus, or SARS-CoV-2. But let me just show you what we've done in the long COVID patients. And we have 3 cohorts here. And so thanks to PolyBio, we've made great connections with Akiko's group and Mike Peluso and Steve Hendricks and Steve Deeks and Tim Hendrick to get access to really, really important cohorts of long COVID and importantly, the convalescent controls that go along with them. So what happens when we start to look at those tetramer positive cells in acute, convalescent, and long COVID patients? So as I showed you a couple slides ago, during acute infection, those SARS-CoV-2 specific cells are highly activated, here using CD38 as the main marker. Once people are convalescent, that is, they've controlled the infection, no long COVID, they're healthy, those SARS-CoV-2 specific cells are not activated. So all of these are SARS-CoV-2 specific in these 3 panels on the right. But in long COVID, at least in this example, about a quarter of those spike specific cells are now activated. In fact, if you look across these 3 cohorts that I introduced on the last slide, we see about 25% of long COVID patients have significant spike-specific T cell activation compared to 5% or less of our convalescent controls. Now, in acute infection, this is much higher, as you might expect, but still it's telling us that about a quarter of our long COVID patients have clearly detectable activation of spike-specific CD8 T cells. I'm going to show you a little more data in a minute, but this suggests that there's antigen still persisting that's capable of stimulating these T cells. If you look at EBV-specific T cells, you see exactly what Akiko described during acute infection. We see that EBV-specific T cells are highly activated. That's consistent with the original Jim Heath data and other data from Akiko and many others now. But during acute infection, there's dysregulation of EBV latency, virus reemerging from latency and actually boosting some immune responses. In our convalescent patients, you see a lot less of this, despite the fact that herpes viruses persist and undergo rounds of reactivation from latency. There's relatively little activation of EBV-specific T cells. In long COVID, again, we see about 15 to 20% of our long COVID patients have activation of EBV-specific cells in the steady state. And so that's significantly higher than our convalescent controls or other healthy individuals. If you look across the different viruses we examined for EBV, you see it's about 15% of our long COVID patients. You see about the same frequency when you examine varicella-zoster virus-specific T cells. Importantly, though, CMV-specific T cells are not different between long COVID, convalescent, and acute infection. So this is specific in some way for EBV and varicella-zoster. You also have flu over on the right as another control. This is not simply bystander activation of any memory T cells. The flu-specific T cells are incredibly quiet, even more quiet than CMV, which is about what you'd expect. But it really suggests that there's some dysregulation of the ability to contain latency for EBV and very likely varicella-zoster virus. So where are we going from here? So this tells us we think that there's antigen persisting for SARS-CoV-2 and probably some dysregulation of containing EBV and VZV. We'd like to see whether these cells can tell us anything more about where and how this is occurring. So what Mark has done is he's built a single-cell RNA-seq approach where we can actually label the cells with those surface tetramers, so a barcode-enabled antigen mapping or a BEAM experiment. So we're surface labeling with the tetramers, then performing single-cell RNA-seq with TCR-seq to get transcriptome, TCR diversity, and surface phenotype on these cells. So we'll be able to map all these different specificities over time. The system works incredibly well. This is just a pilot experiment showing that there's great specificity of these reagents in the single cell landscape. So we see this nice diagonal with 2 different BEAM reagents labeling the same peptide from spike or CMV, flu, or EBV. Our initial pilot experiment told us what we sort of knew from flow cytometry, but that is that these cells that are specific for CMV, EBV, spike, and flu exist in different transcriptional states. And so that's consistent with forming very nice central memory for flu, more effector memory or TEMRA-like cells for CMV, and some other populations for EBV and spike. So we're very hopeful these experiments should be completed in the next probably 4 to 6 weeks as we get the sequencing done, and a little bit longer then to analyze these data. So we think these data tell us that there's evidence of SARS-CoV-2 specific antigen persisting in the host, whether it's replicating virus or viral antigen, we don't know. There's not a great correlation between the Simoa assay for circulating spike protein and this T cell activation. We're still trying to understand what that means. But it does tell us that somewhere between maybe 20% and 40% of individuals with long COVID have T cells that are telling us that they're still seeing the antigen. There's similar evidence for T cells sensing reactivation of EBV and VZV, maybe a little bit lower, same ballpark. And in fact, there's partial overlap. Not everybody that has CMV detection has SARS-CoV-2 detection and vice versa, but there's a good degree of overlap. Also, this confirms some of the earlier data on dysregulation of EBV, at least during acute infection. So we can detect at least one response with elevated activation in about 40% of long COVID samples, and really very low in convalescent, probably even less than that now that we have more data. So this suggests that there's SARS-CoV-2 virus or viral protein persistence that can be detected by T cells. Other persisting viruses may be awakened in long COVID, and we haven't looked beyond the ones that I've mentioned yet. And that T cells and probably other immune cells can function as potent biosensors, telling us information about underlying immune health, that we can harness this approach to actually learn about persisting viruses, perhaps persisting inflammation in the context of antigen stimulation. So I'll stop there. This was all done by Mark, who's done really an amazing job with this project, really set up on a foundation of things that Mark and Rishi and Socrates did during the end of the pandemic. We've had great collaborations with lots of folks on the call and are very, very thankful for the support from PolyBio. So I'll stop there. Thank you.
Thank you, John. It really is incredible what you can understand about T cells when you use methods like those in your lab. So it's really exciting to see the work come along.
Thanks.
I'll introduce our next speaker. It's Dr. Michael Peluso. He's an assistant professor of medicine at UCSF in the Division of HIV, Infectious Diseases, and Global Medicine, and he'll be talking about targeting the SARS-CoV-2 reservoir in long COVID clinical trials and link expansion to MECFS.
Hey, everybody. Really happy to be here as always. I'm going to talk about some program updates and then this sort of consortium-wide effort that we've been working on to express our views on the optimal design of these trials. So I want to begin by acknowledging the link team at UCSF and PolyBio for their continued support. Our program in San Francisco opened 4.5 years ago in the earliest days of the pandemic, and we've been in continuous operation since that time. We've enrolled over 1,200 individuals, conducted over 6,000 visits, have banked tens of thousands of specimens, and have shared over 30,000 biospecimens with collaborators at this point. Embedded within Link, we have an acute program, a tissue program, an imaging program, a trials program, and as Amy mentioned, with support from PolyBio, we're going to be expanding to other infection-associated chronic conditions, which I'll talk briefly about. I wanted to just call out a couple of our publications from the last couple of months. So in collaboration with our acute COVID cohort, we published our findings that during acute COVID, those who are destined to develop long COVID months later actually have a higher amount of virus that can be recovered from their nose in the first couple of weeks, and are more likely to be positive for longer during the first couple of weeks of infection. So we spend a lot of time thinking about and studying post-acute antigen persistence, but I think that what is going on during acute infection and whether that can be altered to sort of change the longer-term course of someone is a big blind spot for research in this field, and really needs to be a priority over the next year. The other publication that we contributed to at the last symposium, I talked about our work with David Wald's lab looking at antigen persistence in the post-acute phase, where we were able to clearly detect ongoing antigen persistence in blood for up to 14 months post-COVID. We're now working on an analysis that extends that out beyond 2 years, and we're still detecting it. But we, as part of this collaboration with the National Recover Program, provided the results from that other study for a combined analysis with other cohorts, all using the same assay that came out last month and basically showed a statistical relationship between post-acute antigen persistence and the presence of long COVID symptoms. This was especially pronounced among those with cardiopulmonary, neurocognitive, and musculoskeletal symptoms. I think that this is the first of, multiple studies that we will need to really pin this down, but very excited to have been able to contribute to this. I think it's a really important finding that'll shape the field. So in terms of quick program updates, our tissue program is going strong. This is supported by PolyBio. We've done over 80 gut biopsies in the last year. We've done 18 lymph node fine needle aspirations. We've done over 25 LPs with CSF collection over the last couple of months, and actually 11 bone marrow biopsies so far. So we're actually about twice the pace that we expected to be at for those. These samples are going far and wide. We're working on a lot of them at UCSF, but also sharing with Michela's group at Penn, with Akiko at Yale, and with others. And then you'll hear later from Nadia Rohn about our plan to expand the tissue program to also include the female reproductive tract, which will launch next month. With support from PolyBio, we're also expanding to other infection-associated chronic conditions. So what our hope is to be able to make direct comparisons with data and specimens collected in the same way between these different conditions. Initially, we're going to start with post-COVID ME/CFS and non-COVID ME/CFS, but we're hoping over the next year to also expand this to other IACCs. We're going to begin with sort of what we do best in LINC, which is deep clinical phenotyping with rigorous biospecimen collection. We'll share these samples across the consortium. And then we're hoping a few months after we get started with this to expand to our tissue collection program and our imaging program to do these in people with other infection-associated conditions to directly compare the pathophysiologic mechanisms that we're talking about. So this is being reviewed by the IRB now, and hopefully will be approved quickly and be able to launch early next year. And we have a perspective piece kind of laying out our thoughts on this that's coming out in Science Translational Medicine next week. And then the clinical trials program is also going strong. So we've completed enrollment for our monoclonal study. Hopefully we'll have some initial results to report in the next couple of months. We've completed enrollment for our Ensitrelvir study, which was done in collaboration with Shionogi. The IRB is under review for the IL-15 agonist study that we talked about at the last symposium. And then we're also participating in this NIH-funded study of baricitinib. So a lot of momentum on the clinical trials program, and we're excited that that's still going. So in the second half of my talk, I want to talk about some considerations for trials targeting the reservoir. And this is from an effort that was led by Amy and myself, involving pretty much every member of the consortium and even beyond the consortium over the last couple of months. That's currently in revision, so hopefully we'll be able to share final publication soon. But I wanted to lay out sort of how we're thinking about all of this, because I think it's really important to sort of formalize the important considerations. So when we're talking about the viral persistence and the reservoir, each of us could be talking about slightly different things. And I think we need to think about all of the sort of formats in which the reservoir could exist. So the first format in which it could exist is that there's simply persistence of viral RNA that is sort of like defunct, but maybe still prompting an immune system response. And that's shown at the top here. And so, studies that sort of identify the presence of RNA, but don't identify active replication of that RNA could be consistent with that sort of form of persistence. The second form of persistence is that the virus can be being translated into viral proteins without necessarily propagating an active infection. And so there are certain observations that are potentially consistent with that. For example, the identification of spike persistence in blood or in exosomes. And then the third form in which the virus could persist is in active infection, which would in some ways be the simplest form, where there is, perhaps a low-level ongoing replication, potentially with infection of new target cells. And there's evidence that's consistent with that, including the identification of double-stranded RNA, which several groups, including ours, have found in, blood and platelets and tissue. And so that's a third possibility. And the reason that this is important is because the reservoir could be persisting in any of these forms or in multiple of these forms. And when we think about how to target it, we'd be thinking about different types of interventions. So for active replication, drugs like antivirals should work if they're getting to the places they need to go. For this middle form where there's ongoing translation, but you really need to clear infected cells and clear proteins, maybe monoclonals would work better. And for the first form, perhaps immunotherapy would work the best. And so we're thinking a lot now about targeting these different forms of persistence and whether, in fact, we actually need combinations of these types of treatments to do that most effectively. The last point, or last few points I want to make are specific considerations for the conduct of these trials. And this is a figure that is currently under review. And I just want to call out a few of these issues that we've been thinking really hard about over the last few months. So the first is with regard to eligibility determination for these trials. It would be great if we could screen people into these studies with a measurable, with a biomarker that says, this person does have this mechanism of persistence, and that's why we're going to include them. We're not quite there yet. The first round of trials really enrolled all comers with broadly defined long COVID. I think based on some of the emerging data, we might see that the second round of trials is going to be honing in and trying to recruit a more sort of homogeneous set of people with, perhaps specific symptoms. And I think that that is a valid approach, especially in the current landscape of eligibility determination. The second issue I wanted to briefly mention was tissue penetration and sort of pharmacokinetics, which has not been a big topic in this field, but I think is something we're really thinking very carefully about in our trials. And so, for example, in our monoclonal trial, we're actually going to be doing detailed pharmacokinetic modeling of the decay of the antibody in the people who got treated. And we'll also be able to look for the antibody in the tissue specimens that we've collected in that study. The third issue is related to duration and timing of treatment. So all of the studies that will report out this year, including the Stanford study, used really, fairly short durations of antivirals, about 2 weeks, no more than 3 weeks. If you look at the animal models, including cat models of persistent coronaviruses, they often require months of treatment. And so I really think we need to be thinking carefully about generating toxicology data that will allow us to do longer courses of treatment in people. On-study reinfections are a huge problem that the field needs to grapple with. We were fortunate in our monoclonal study that most of the reinfections happened after the primary endpoint. In our antiviral study, we're seeing people getting reinfected within 30 days of the study, which is a big challenge that we are all going to need to grapple with as we're thinking about measuring antigen persistence in these trials. And then the last point I want to make is it's been so encouraging on this call so far to see how far we have come in the last 2 years in measuring viral persistence. But what we really need to do now is test all of these different measurements side by side to figure out how they vary, how the tests vary between each other, how they vary between people, and even how they vary over time within people. And I think that this is really going to be a key over the next couple of years for figuring out how to proceed with these measurables in our trials. So I'm going to end there. Want to again acknowledge our team and PolyBio for funding and look forward to the Q&A. Thanks.
Great. Thanks, Michael. And I agree so much about that need to test the different diagnostics side by side. That's something we're really going to push to try to make happen this year. All right, next guest, our speaker is Dr. Tim Henrich. He's a professor in the Division of Experimental Medicine at UCSF. He'll be talking about molecular imaging in long COVID plus tissue biopsy program updates.
Okay, thank you so much. And thank you, Michael, for introducing the LINK cohort and all the excellent work that's been going on, not just here at UCSF, but all our collaborations and really in due to PolyBio support for making all of this happen. So really grateful for this as well. So I'll be giving a little bit more details in some of the molecular imaging in long COVID and tissue biopsy program updates within the LINK cohort. And again, this is our pathogenesis team. Again, we collaborate widely across, but we certainly have a core group here that has been working very hard to understand the mechanisms that are driving long COVID and the immune profiling and inflammation that we see that results. Just so that you know, our main goal and focus right now is essentially to stamp out viral persistence in the setting of tissues, whether this is SARS-CoV-2 or EBV or VZV reactivation has also been mentioned. So I think, first of all, I think what we have been learning, and I think that all of the talks have so far touched upon, is that long COVID is a tissue-based process. That this is not a peripheral blood disease, but this is something that is spread throughout the body in various deep anatomical compartments, which can be very difficult to sample, especially in the setting of treatment schemes, et cetera. So we obviously need novel methods to identify abnormal immune responses, inflammation, viral host interactions on a cellular and tissue-based level. And so we've taken the macro and micro approach, so macro to micro, looking across non-invasive nuclear imaging using immunopet strategies to look at viral persistence, immune responses, et cetera, but also looking at digital spatial profiling within tissues itself to understand what these viral host interactions are and how they change in the setting of treatment. So just to recap what we showed last time back in spring, which is now in print, as you can see on the left, this is actually on the cover is actually SARS persistence and gut samples from the LINK cohort. And this was using both a combination of immunopet imaging to look at T cell activation throughout the body. And just to show you, here's a pre-COVID participant with some background in the liver and some glandular tissues, et cetera. But that's our background noise. But when we look, a few months after acute infection, we can see that there's a dramatic uptake across many tissues. And then even 663 days later, we see persistence in many of our participants, suggesting that not only is there a long COVID association, but there's a post-COVID association with long-term immune activation and dysfunction across the body. And so we seem to have entered a new post-COVID set point, at least from what we can see from some of these molecular imaging techniques. And that's important to take note of when using pre-COVID controls as well in the setting of long COVID. So what we're doing now, we've greatly expanded on this initial project. We are now trying to fully understand the relationships between long COVID and people that are fully recovered. So we've almost performed 75 images on 75 individuals at this time, which is one of the largest PET studies ever here at UCSF. That's actually a lot of effort to go in to get people into a scanner, to inject them with radioactive material, to do all screening and things like that. But we've included folks with various long COVID clinical phenotypes through Michael's work to really fine tune and clinical phenotype, as we heard earlier as well, is very important in interpreting some of this data. And we've also expanded imaging, obviously, to people with full recovery, because it's now nearly impossible to get uninfected controls. And obviously, as Michael Plus also just mentioned, it's challenging to do these types of studies with so many waves of reinfection, and those have slowed things down and made it more complicated, but certainly we can power through. So we've also really been focusing on performing longitudinal imaging in greater than over 30 people right now experiencing long COVID prior to and following intervention. So Michael talked about the Aryan monoclonal study and our oral Infectious study, as well as others. And so far, we have managed to image about half the cohort before and then after their interventional therapy. So we're going to get a sense of what dynamic processes might change and do a deeper dive than just looking at peripheral blood measures. We've also been collecting saliva at the same time and looking both in blood and saliva for correlates of imaging with peripheral blood markers. Again, as folks have talked about earlier today, that there are starting to become more and more biomarkers that are showing increased inflammation, immune activation, and immune sensing over time. And I can say that right now, 25% of our cohort, which is far out from acute infection, actually has detectable EBV, in some levels very high EBV shedding in the saliva. So very interesting as well. We're still trying to make and understand what this means. As we showed before, we've seen SARS-CoV-2 RNA persist for years after initial infection. This is before all waves of reinfection in the kind of smaller but cleaner cohort. And we also saw double-stranded RNA suggesting either some viral life cycling or at least endosomes with double-stranded RNA that are persisting over time. And they seem to be in these clustered regions within the gut tissue in the lamina propria, which we're calling coroniomas at this point, although we're open for interpretation and other suggestions. But we're also now looking at not just SARS-CoV-2, but what the reaction and what the relationship, is with other viruses like EBV reactivation, for example, in the tissue, how this correlates in space, in situ in the gut sample. To talk a little bit more, as Michael alluded to earlier, we've processed now almost 90 gut samples from our participants. And again, we're also focusing very closely on treatment responses in this clinical trial. So looking, for example, we've done 18 pre and 17 post in the Outsmart Arian antibody trial. I think one of the posts we couldn't make sense of because they had acute infection right before. So unfortunately, but just as a real life example. And then in our Synfil trial, we're at a goal of 20 pre and post paired gut biopsies along with our imaging as well. So we're doing a deep macro and micro dive before and after therapy. Just to get a sense of what we've been seeing, and this is all preliminary data, but here's our coronioloma, our double-stranded RNA cluster in the gut. When we use high-dimensional digital spatial profiling to look at host viral responses within each individual cell across space, in this, we can actually see that there is actually quite a different makeup or milieu of each of these cells within people with and without long COVID in the tissue. Now, this is just preliminary data, so this could change, obviously, as we look at more samples. But what we're seeing is very interesting, that if you look at macrophages within gut tissue in those that have detectable SARS-CoV-2 versus non-long COVID without detectable and pre-COVID samples, what we actually can see is upregulation of specific genes that may actually modulate or dampen a cytotoxic, either NK cell or cytotoxic T cell response. But at the same time, it's also triggering, type 1 interferon and other inflammation. So the body is sensing virus that's present. It's inflamed, but it's also preventing it from being cleared. It seems to be regulating and modifying that natural killer and cytotoxic T cell response. For example, here, if we see in a long COVID participant where we have CD8 T cells on a UMAP on the upper left, we see almost no granzyme B production, for example, even though we can find copious virus in this tissue. So again, preliminary work, but it gives a sense of what we're doing now in space across all these tissues. So just want to say that we actually have been seeing this in the peripheral blood as well. This is recent data that we generated looking at NK cell phenotyping using a high-dimensional phenotypic spectral flow assay. And what we're seeing is that all the numbers of total NK cells, these CD56, CD3 negative NK cells, are similar across cohorts. We see actually a significant smaller or lower proportion of these CD56 dim CD16 positive NK cells, which are thought to be more mature and more cytotoxic. These are the doers in the NK cell repertoire. And we're seeing fewer of these doers or cytotoxic phenotype in people with long COVID. And we actually see a correlation in figure H here, where we have fewer frequency or proportion of these cells with a greater number of long COVID symptoms in the peripheral blood. We also see this reduction in these mature cytotoxic phenotypes of NK cells in various different long COVID phenotypes as well. Interesting enough, adaptive NK cell responses, which are akin to like memory responses in NK cells, don't seem to be up or different in people with long COVID, but it certainly is in the setting of CMV antibody antigen positive. And we know that NK cells seem to develop adaptive responses to CMV, but it does not seem to be the case with SARS-CoV-2. So it's possible that there's an insufficient natural killer cell response in the setting of long COVID that is leading to some of the viral persistence that we're seeing in tissue. This obviously is leading to our IL-15 trial we talked about. If we can boost NK cell response, if we can boost CTL responses, perhaps we can clear these viruses from these tissues directly. And so that's nearing approval stage at this point. But I want to say that, we also saw a lot of uptake in the bone marrow on our T cell activation panel, which is very interesting. And so we became very interested in the bone marrow. And as Michael also touched on, we've now biopsied 11 people, and we're doing a very, very deep dive now using multiple different components of the bone marrow. So aspirate, we're looking at core biopsies as well for our digital spatial profiling, and also looking across different multi-omic approaches to understand what's happening in terms of viral persistence, host immune response, hematopoietic function, inflammation, also megakaryocytes and protoplatelets, essentially these kind of platelet precursors, and to see if they're also harboring SARS-CoV-2 directly within the bone marrow. And then lastly, we've now made significant strides in developing PET imaging studies to directly look at viral persistence within the body. So we have finished preclinical testing of both a radio-labeled sotrovimab, which is an anti-spike antibody from pre-Omicron era, as well as an Arium antibody. Unfortunately, the company is no longer. But we have seen that looking at dosimetry, that we actually get a very clean, very low background noise, for example, in the sotrovimab antibody about 48 hours after imaging, suggesting that we may be able to pick up subtle areas of viral protein persistence across tissues in the human. So we hope to have our IND submitted within the month or so, and we will rapidly move forward to imaging our cohorts with this technology. So that's the update I wanted to give. And again, just thank everybody through the LINK program and PolyBio, Recover, and all the other sources of funding that have supported this work. So thanks so much.
Thanks, Tim. Very great work. Seeing your images. What I love about imaging is how you get really cool images, both from the scanning and actually just the biopsy. So it's so cool to just see your data and how
it's always better to see something, in my opinion.
Yeah, no, seeing is believing. Right? Yeah, it's true. So yeah. All right. Well, perfect then. Our next speaker is Ethan Civic. She's an immunologist and associate research scientist in the Flavel Lab at the Yale School of Medicine, and she'll be talking about a humanized mouse model of SARS-CoV-2 RNA persistence.
Can you hear me? I'm not sure.
Yes.
Oh, wonderful. I'll share my slides. So I've actually recently been promoted to assistant professorship at Yale. But we are continuing to dissect disease mechanisms of long COVID. And as we have heard from many people, we're focusing on the persistent virus and viral reservoirs. And the work that I'm going to present today is a very collaborative effort of multiple labs at Yale, including the Flavel Lab, Iwasaki Lab, Violent Lab, and Pile Lab. So the way we have been studying SARS-CoV-2 infection and viral reservoirs is utilizing humanized mice. These mice have a human immune system, and they're generated by transplantation of human hematopoietic stem and progenitor cells into various strains of immunocompromised mice. And the goal here over time is to generate a comprehensive immune system comprising of lymphocytes and myeloid cells. And the model that we are using, which we call Mr. G6 family of mice, provide these unique factors, human cytokines, at their physiological level to allow development of monocytes and macrophages. From these, MCSF allows full development of monocytes. GM-CSF allows development of alveolar macrophages, and targeting the GCSF axis has enabled us to work on human neutrophils. So we have been using these mice to characterize immune reservoirs of COVID-19. We first focus on the pathological macrophage response and have shown that the infiltration and the phenotype of macrophages that are entering the lungs in response to COVID are very similar between humans and humanized mice, the Mr. G6 system of mice. We see infiltration of inflammatory monocytes, which then differentiate into monocyte-derived macrophages. And there is accumulation of CD163 pathological macrophages in both patients and mice. We use these mice then to characterize the mechanisms of pathology and identify that SARS-CoV-2 indeed infects human macrophages. There is viral replication happening in these human macrophages, although there is very little virus, actual virions being made and produced. This viral replication leads to NLRP3 inflammasome activation, then secretion of inflammasome-mediated cytokines, such as IL-1 and IL-18, which further contribute to inflammation, and chemokines such as CXCL10. And interestingly, CXCL10 expression was preferential and unique to infected macrophages. These macrophages then died through pyroptosis, which is an inflammatory form of cell death, and leads to severe pathology. And this infection of macrophages under response is persistent throughout the disease, and we see it as early as 4 days post-infection, but is preserved until a month after infection. And this persistence that we see, viral infection, replication, and inflammasome activation is also seen in humans. When we looked at human autopsy lungs, we were surprised to find that almost half of the macrophages in those lungs had either spike or RNA-dependent RNA polymerase of the virus in them, suggesting that they are indeed infected. So we have been working on macrophages, and as we continue to work on our work, we were interested in whether other human myeloid cells could contribute to this hyperinflammatory loop. And we focused on human neutrophils, given that we have the means to study these human neutrophils in vivo in mice. We first infected humanized mice that bear human neutrophils and compared the SARS-CoV-2 pathology to mice that lack human neutrophils. And as you can see here, both at 4 days post-infection and 14 days post-infection, mice that have human neutrophils had worsened pathology and had a worse histopathological score. We wanted to understand how neutrophils contribute to this, so we looked at neutrophil effector functions and identified that human neutrophils are positive for MPO and citrullinated histone and DNA complexes, which are known as NETs. Netoses or NETs are extracellular traps when neutrophils in a very organized manner secrete the contents of their nucleus, histones, and DNA. This observation was only present for human neutrophils, as mouse neutrophils didn't respond to SARS-CoV-2 infection in a similar manner. This wasn't because mouse neutrophils did not or cannot undergo netosis, because if you take these mouse neutrophils and stimulate them ex vivo with a potent stimulator of netosis, PMA, we could achieve netosis in these mouse neutrophils. So this was a unique feature of human neutrophils. And this has been described in COVID-19 severity and pathology in blood, finding NETs that are MPO and DNA complexes correlated with disease severity. And there were more NETs in non-survivors compared to survivors, regardless of their other parameters. Next, we wanted to see if we can block netosis, and netosis is a very regulated process, and its role as an antimicrobial is very well described for bacterial infections, but we know that it is also induced in response to viruses, although their protective role in that scenario is not that clear. So first, we wanted to see if neutrophils can be infected and whether this infection can cause or initiate the netosis program. For this, we isolated neutrophils from blood of human donors and infected them with SARS-CoV-2 in vitro and assessed either their netosis potential, but also presence of virus in these cells and replication. To assess replication and presence of virus, we used a reporter strain of SARS-CoV-2 known as MNG. And when this virus, which is a cDNA clone of the original SARS-CoV-2, infects a cell, it turns that cell green. And you can see a very clear signal here in human neutrophils. When we treat these human neutrophils with Paxlovid, we can reduce this MNG signal, suggesting that blocking viral replication blocks MNG signal, which is also quantified here. Another way of looking at it is measuring subgenomic RNA in human neutrophils, which is or in any cell type, which is a measure of viral replication. And in vivo cells, which we use as control, we can see a very clear subgenomic band. And this band is also present in human neutrophils as early as 4 hours post-infection. This is lacking in uninfected neutrophils, suggesting that human neutrophils are indeed infected with SARS-CoV-2. Next, we wanted to see if in this in vitro scenario, infection of SARS-CoV-2 could induce NETosis, and it does indeed. You're looking at uninfected levels versus infected levels of NETs in these neutrophils. And when we treat these neutrophils with Paxlovid again, we can reduce the NET positive neutrophils. But we can also block NETosis either using PAD inhibitors, which is the enzyme necessary for citrinylation, or serine protease inhibitors, which block various enzymes involved in NETosis pathway. So we wanted to see if this is true for in vivo as well, and we used serine protease inhibitor, Pafablog, which has been used in other in vivo contexts, and treated mice with Pafablog, and we have observed a similar observation in vivo when NETosis is in vivo. When Pafablog was used, we had fewer neutrophils that were NET positive. But more importantly, when we blocked NETosis in these mice, we could also ameliorate the lung pathology and in a way bring it down to the levels that we see in mice that lack neutrophils, human neutrophils. So overall, we think myeloid cells are important reservoirs of SARS-CoV-2. We've shown it in macrophages, but it's true in neutrophils as well. In macrophages, virus can enter the cell through a variety of mechanisms. It could be mediated through antibodies, ACE2 or CD169. And viral replication initiates a very inflammatory form of cell death, which communicates and amplifies the signal that there is infection. Although it might be helpful in the early stages of the disease, it is pathological when it's persistent. And we do see a similar observation, and so far we have identified that ACE2 is a player in neutrophils. Neutrophils express high levels of human ACE2 and can mediate viral entry. And this, again, initiates a viral replication program and ends with a very inflammatory form of that. For macrophages, it's pyroptosis, and for neutrophils, it's NETosis. And we're trying to identify right now if there are other ways the virus can enter the cells and whether neutrophils play a role in patient as well. So I've already mentioned it. This has been a very collaborative effort involving multiple labs at Yale, but I would like to thank my former mentor, Richard Flavel, for all the support. And I would like to thank PolyBio for their continued support for our work. Thank you.
Great, thank you. And I look forward to introducing our next speaker, Mike Van Elzeker, because I know he'll also be talking about NETs quickly. So that will be interesting. Mike is an assistant professor in the Department of Neuroimmunology at Harvard Medical School, and he'll be talking about understanding long COVID and ME/CFS brain fog from cells to circuits.
So thanks very much for having me on. I appreciate it. And thanks to all the previous speakers. So, I work in a neuroimaging lab at the Harvard MIT Martino Center. And our lab kind of shares the PolyBio vision, of course, of trying to combine multiple measurements so that we can get a little bit closer to the root cause of abnormalities and symptoms and difficulties that patients face. So we have an ongoing neuroimaging program, but along with that, we collect a lot of other things. And I wanted to give a little bit of a tour of that today and maybe talk a little bit about some data that we have. So here's some current work in the lab. Almost everything we do is both long COVID and people that have the ME/CFS diagnosis from before 2019. We also work with some other patient groups as well. So we do a dual MRI PET neuroimaging study for neuroinflammation in both long COVID and pre-COVID ME/CFS. And we're also starting a PolyBio supported full body MRI PET scan for fibrin accumulation in organs. We want to see, especially of people with ongoing dyspnea, trouble breathing, whether they show signs of fibrin accumulation along with microclots in their blood. So in these two imaging studies particularly, what we do is before they come in, before our participants come in, both cases and healthy controls, for the 2 nights prior, they wear an overnight sleep monitoring device so we can get some objective sleep data. And then before they come into the scanner, we have them do pupillometry, which I'll talk about in a little bit, a quick flash of light to measure pupil responses, as well as giving us blood and saliva. That way we can sort of a little bit better understand what's happening in the neuroimaging or the full body imaging. Another study that we have going in long COVID and ME/CFS, but we just got a really generous donation to study craniocervical instability, which in some cases is infection onset, in some cases it's accident onset and things like that. But what we use is a MRI safe vagus nerve stimulator for the ear. And so we can actually sort of probe the vagus nerve function to see whether or not that system has been sensitized in these patients. And we can also measure cerebral spinal fluid flow, velocity and volume as people, and it's measurable even as they lie flat on their back. And you can measure the sort of cerebral spinal fluid flow to see if that's maybe something that's happening in these patients as well. We also have a study ongoing that uses regular MRI and EEG for cognitive dysfunction. And this, again, we take blood and do a bunch of other measurements before they even get into the scanner. And we do this overnight sleep quality recording with a sleep image wearable device that allows us to record for the 2 nights before they come in for the scan, sleep quality, measures of sleep apnea, things like that, which seems to be disrupted in these patient groups, as well as pupillometry, which is a measure of autonomic function, neuroautonomic function, as well as intracranial pressure. And then we do multiple forms of blood plasma analysis and multiple forms of whole blood analysis, as well as multiple forms of saliva analysis. And I wanted to walk through those last three a little bit to kind of show you how we take advantage of the consortium that we've built with PolyBio. So these are all these are among the things that we do. So what you see up top is the tubes that we collect saliva and blood into, and then down below are the different aliquots that we produce. And so we measure SARS-CoV-2 RNA with Johan Van Weyenburg in Belgium. We get a saliva microbiome and inflammatory profile from Marcella Fieri at the J. Craig Venter Institute. We look at live neutrophil tests with Daniel Remia next door to us. And just to show you what Dr. Shafiq was talking about just before me, this is an example of participant plasma being added to neutrophils, and you watch as the neutrophils release their neutrophil extracellular traps. This is actually DNA that you're seeing being released. And here's an example of neutrophils in a healthy person swimming. You sort of barely see any swimming at all. And then if we look at a patient, in this case with pre-COVID ME/CFS, you can see their neutrophils are much more activated. So this is that neutrophil activation that we were talking about in previous talks. So we also do microclot quantification, and we're trying to develop with Dr. Kieran Deep Gill and Dr. Daniel Remia a microclot quantification techniques where we can sort of get a value of healthy controls and compare long COVID patients as well as other chronic illnesses. And then we also measure a bunch of immune, vascular, endocrine measures, matrix metalloproteases, cytokines, et cetera. We do a spike protein assay with Dr. David Walt here at Harvard Medical School. And then Dr. Jean Tan at the J. Craig Venter Institute will be testing antibody function and history. And then we also have several tubes banked for future analyses and data crunching. So I wanted to talk just a little bit about the symptom of so-called brain fog. That was originally a patient term some people think now that it's a little bit dismissive, but basically trouble concentrating, difficulty with cognition. So we had a hypothesis about why this may be happening based on some previous work that happened before COVID, basically a meta-analysis that measured inflammation in just people undergoing neuroimaging per se. And this is a meta-analysis of 24 studies with 457 participants where they measured inflammation in the blood, or they deliberately challenged with inflammation to find out what the circuitry is for the brain's response to inflammation, which is part of why you feel sick. So we noticed that one at the top up here is a region called the anterior mid-cingulate. And we thought that was really interesting because the anterior mid-cingulate is really important for what we call cognitive control. So it's the ability, and this, what you're seeing here is this activated during a task called the multi-source interference task. And cognitive control is really important for being able to focus while there's distraction. And that's something that patients have really talked about. So the combination of it being a hub of control of cognition and being really sensitive to inflammation led us to think that maybe this is part of why people experience so-called brain fog. So what I'm showing you here is long COVID, 43 healthy controls versus 12 long COVID patients. In this case, the healthy controls are pre-COVID. And what I'm going to do is superimpose the task that we collect in the same scan session. We get both neuroinflammation and task. This right here is the anterior mid-cingulate. And then what you'll see in blue is activation during that task. So you can see that it really overlaps. So we think that the inflammation in that one particular brain region that's also in charge of cognitive control is probably part of the reason that people have such trouble maintaining focus and concentration when they're sick. And here's some pilot data from pre-COVID ME/CFS that also shows in that same region activation of neuroinflammation right here. And then again, if you can see the blue, that shows activation during that task. So in this case, we think the long COVID patients and the pre-COVID ME/CFS patients have similar symptoms for similar reasons. So regarding that sleep quality portion of our study, this is output from the sleep study that we do with people before they come into the scanner. What this system does is it collects something called a sleep quality index, which is a measure of several forms of, essentially measuring sleep quality, sleep stability, fragmentation, periodicity, measures of sleep quality. And you can see both the long COVID and the pre-COVID ME/CFS patients show a pretty big decline relative to the healthy controls. And that dashed line is what you'd expect a healthy control to be. And in this case, since we do 2 nights and sometimes it doesn't work, we've got 15 samples from 10 healthy controls, 30 nights from 17 chronic fatigue syndrome or ME/CFS patients, and then 53 nights from 25 long COVID patients. And then here, what I'm showing you is pupillometry, where there's a quick burst of light that measures pupil contraction. And in this case, you can see the long COVID people have a smaller diameter of their pupils before the provocation relative to healthy controls, and the ME/CFS patients don't show that difference. And here, the long COVID folks have a stronger constriction after the stimulation, which is significantly reduced relative to both the ME/CFS and the healthy controls. In this case, showing that the ME/CFS from before 2019 show a slowed pupillary response, which is really what you'd expect in something a little bit more like concussion. So this is an example where the symptoms look a little bit different. So I'd like to just thank my current lab. Here we are standing next to the PCR machine that we use to test ourselves all the time. And thanks to everybody.
Thanks, Mike. Really good overview of a lot going on in with your group. Cool. So our next speaker is Peter Broden. He's the Garfield Weston Chair of Neonatal and Professor of Immunology at Imperial College of London and a professor in the Department of Women's and Children's Health at the Karolinska Institute in Sweden. And he'll be talking about sex hormone regulation of human immune systems.
Thanks so much, Amy. I hope you see the slides well. I'm going to move forward until you protest. So my team is really grateful to PolyBio for supporting a project that we're looking for genetic determinants of long COVID. And what we're doing in this project, just to give you an update, because this is not going to be the focus of my talk today, is to do whole genome sequencing. And we've currently sequenced 110 patients, which are really well characterized, very severe patients with long COVID, all having objective measures of disease with various forms of organ dysfunction, etc. And we're currently looking for genetic variants that can explain why they have what we think is impaired antiviral immunity, leading to viral persistence as our preferred hypothesis underlying the condition. And so I hope to give you an update in our next meeting on some results to this end. But today I'm going to focus, as you said, on sex hormones. And the reason for this and why this is relevant, I hope will become apparent. We know for a very long time that infection severity tends to be more or greater in males versus females. Vaccine responses tend to be stronger in females as opposed to males. We also know that side effects to vaccines tend to be more frequently reported in females, suggesting an overall stronger response. While autoimmune diseases predominantly affect females more than males, with some extreme examples as Sjögren syndrome and SLE being 9-to-1 female to male ratios. And just to illustrate this with some of the lessons learned during the COVID pandemic, we know across all countries prior to vaccines that case fatality rate was higher in males than females. We now know quite a bit about why this is, and the timely type 1 interferon response is a real determinant of severity in acute COVID, and typically stronger in females than in males, protecting them from more severe acute disease. In the context of failed or delayed type 1 interferon response, we know that a pro-inflammatory response driven by TNF, IL-6, is seen and sometimes referred to as a cytokine storm, leading to severe disease and death, predominantly in males during the pandemic. Now, when it comes to long COVID, there are numerous publications from various groups and different countries that have shown that the risk of developing long COVID symptoms is higher in females than in males. And there seems to be a greater risk in reproductive age. So, women in adolescence up till menopause and so on. But the question is why? And with all these sex differences, the biggest challenge has been to decompose the different contributions from the X and Y chromosomes and the hormones, because both differ when comparing males and females. And so that led me and some of my team members to design a study already back in 2016, where we decided to follow individuals that undergo gender-affirming hormone therapy. And we recently reported the first study out of a few where we have studied individuals born assigned female sex at birth and then receiving testosterone therapy as a gender-affirming hormone treatment, during which we follow the adaptation of their immune system to this changing hormone environment, and this is obvious, but while the genetics do not change. So the hormone levels change in these individuals in this way, where testosterone is provided and increases to male reference range at the first study visit 3 months after initiation. And after that, or during that time, we see a reduction in estradiol and progesterone, and the menstrual cycle stops in these individuals. And when we look broadly at the immune response in the immune system using whole blood transcriptomics, we see that there are 2 large-scale changes that happen at the transcriptional level. And the first thing is that a group of genes, all associated with type 1 interferons, decrease in these individuals after testosterone therapy, while a group of genes associated with pro-inflammatory responses, IL-6, TNF, etc., is increased. So it's as if the immune system basically rebalances itself or recalibrates itself based on hormone treatment. And a part of that could be explained by changes in cell composition, although these changes were rather small. But one key cell type that decrease in blood is PDCs, or plasmacytoid dendritic cells, which is a key producer of type 1 interferons, and it is decreasing after testosterone therapy. However, it's not only a numbers game, because the function of these cells is also decreasing or reduced. So when we stimulate cells in vitro with either at baseline or after 3 months of testosterone, we see that the PDCs produce less type 1 interferon related genes after R848 stimulation, a viral mimic, you can say. And when summarizing this, we see that this change is the most significant change. It's also seen at the protein level in these cultures. And also in other cell types, it's not only a PDC change, because if we look at monocytes, which also produce these interferon-associated genes after R848 stimulation, we see a reduction after testosterone. And interestingly, in these monocytes, we also see the reverse pattern for TNF responses. And when you stimulate these monocytes, not with a viral mimic R848, but actually with LPS, which is a product of gram-negative bacteria, and that's a principal inducer of TNF, you really see this enhanced response after testosterone therapy, suggesting that these two branches in our immune system, the type 1 interferon response and the TNF, are cross-regulated features that are balanced against each other. And this balance is calibrated by sex hormones. And it's intriguing to think about whether these are direct effects or indirect effects. But when looking at the hormone receptor expression in these cells, so AR here is the androgen receptor sensing testosterone, and ESR receptors sense estradiol. And when you look at the sort of baseline transcriptional expression of these genes, plasmacytoid dendritic cells really stand out as the real sensor of sex hormones in human immune systems, both in males and females, I should point out. So it's almost as if these cells are sensors of sex hormones and then modulate the immune system accordingly. So this cross-regulation, I think, was actually known previously, but I don't think we've paid enough attention to it. And the reason I say that it's known, because when clinicians like myself give patients anti-TNF for arthritis or Crohn's disease, etc., we know that a subgroup of patients, when you block the TNF pathway, will actually upregulate interferon and develop a lupus-like disease, which supports this notion of cross-regulation. But from an evolutionary point of view, it's tempting to speculate why these two branches might be balanced by sex hormones. And this leads me to speculate around, based on the framework of life history theory, and based on the notion that males and females make different investments into reproduction and growth, for example, it's tempting to think that anything that potentiates or increases the chances of successful reproduction will be boosted in a female, specifically in reproductive age. And in light of that, we know that TNF levels are associated with poor outcomes in IVF treatments. It's associated with pregnancy complications and poorer outcomes both for child and the baby. And also, we know that type 1 interferons is a really important beneficial factor in pregnancy, specifically in protecting against vertically transmitted viruses, and to my knowledge has no deleterious effect on implantation or pregnancy development. But on the male side, you might wonder, but why would males want to do the opposite, boost TNF and dampen type 1 interferons? And again, going back to life history theory, we speculate that the increased investments in males made towards muscle growth and strength might give us a clue, because as TNF, every time we perform strenuous exercise, TNF is released locally in the muscle tissue by macrophages and plays a really important role in muscle repair and growth. And on the contrary, in individuals with dermatomyositis, which is one of these diseases really driven, an autoimmune disease driven by type 1 interferons, we know that there's a direct effect of interferons on muscle fibers leading to muscle wasting. So suggesting that potentially it's beneficial for a male to boost TNF and dampen type 1 interferons in light of these differences. And so this work has been published, and if you find this interesting, I would urge you to check this out. I do think this work, although not directly studying patients with post-infectious diseases, given the sex hormone bias in post-COVID, long COVID, and given some previous reports by others, such as the Iwasaki lab and others showing hormone differences in long COVID patients, I think this is a research line that should be pursued to understand these conditions better. And with this, I really want to thank my phenomenal team members who have done this work. In blue are the main drivers of this work and collaborators, and the funders, and obviously PolyBio for supporting our work. So thank you.
Thank you, Peter. It's great for you to set the stage and just discuss the intersection between sex hormones, immunity, and infection. I do think that's going to be a huge area across many chronic conditions, with long COVID being a good example. So thank you. All right, the next speaker is Lael Jonker. She's an associate professor of pediatric pulmonology at Harvard Medical School, and she'll be talking about a pediatric program long COVID update.
Great. Thank you so much, Amy. And thanks to PolyBio for including specifically pediatric long COVID and the work that you're supporting. So I'm really grateful. And, you know, just to sort of highlight how understudied pediatric long COVID is, here's the number of publications that come out for long COVID in general, the vast majority of which are focused on adults. And then if you look at the number of publications that come out for and I used a couple different terms to try to capture pediatric long COVID. You can see it's really a fraction of the amount of research that's being done. And most of these are reviews and then a few sort of clinical studies, as well, several clinical studies as well. But that is really a detriment to kids who are suffering from long COVID. And it's really important to do this research because we need to know what long COVID looks like in kids. We need to know how kids are responding. Is there evidence of inflammation of long COVID, you know, that we're seeing in adults and kids as well? And then what can we do, most importantly, for kids with long COVID? And so I would encourage all of the speakers on this symposium to include samples from children with long COVID and really highlight that you're including samples from children so that people can understand and sort of accept long COVID in children as well. So what are the signs and symptoms of long COVID, of children with long COVID? So there was a great study that came out. I was not part of this, but this was led by Rachel Gross of the Recover Pediatrics group. And they surveyed over 5,000 children between March 2022 and December 2023. And they were basically looking at symptoms, and they grouped people as to whether or not they had had COVID or they had never been infected. And they confirmed by looking at nucleocapsid antibodies. And some of those who had been infected, it's been, over 500 days. So it didn't matter how close or far they were. And the symptoms were compared between the two groups. And I'm going to show you that importantly, so they differentiated into two different groups because the symptoms did seem to separate in school-age children, ages 6 to 11, and then teens. Here's the school-age children. And you can see one is the comparator group, so never infected. And you can see the wide range of symptoms that are increased following COVID. And these aren't kids who are self-identifying as having long COVID. They were just previously infected with COVID or SARS-CoV-2, and now we're just asking these general symptoms. And so to highlight a few, those who had never been infected, about 5% were reporting daytime fatigue. as compared to almost 13% of those who had been infected. There was also an increase in feeling lightheaded or dizzy, stomach pain, especially in this younger group, nausea or vomiting, muscle pain, body pain, headaches, and sort of worrisome, very worryingly, significantly increased risk, refusal of going to school. So this is impacting their social and emotional and intellectual learning. And so then looking at kids, similarly, daytime sleepiness, difficulty sleeping, changes in loss of taste or smell, again, muscle and back, neck pain, headaches, difficulty focusing. So a lot of these symptoms that, again, they weren't self-identifying as long COVID. They'd been previously infected. And so there's an increased risk of these symptoms. So that's to say that most likely many of these individuals do have long COVID. We're just not necessarily recognizing it. There's also a few studies that have looked at physiologic signs of long COVID in children. Again, sort of just justifying that long COVID is a physiologic disease that it can affect kids. There's orthostatic intolerance in about 71% of kids with long COVID. There's autonomic cardiac function abnormalities that can be detected on EKG in cohorts of children with long COVID as compared to controls. And there can be some abnormalities, some sort of subtle abnormalities on cardiopulmonary testing. Not surprisingly, like in the adult, there's no clinical labs that show one way or another if somebody has long COVID or not. So it's still challenging, and these are not necessarily tests that we would do routinely, although you could certainly do orthostatic testing in clinics. But, you know, so we were given this sort of increased understanding and recognition of COVID. We actually wanted to ask pediatric providers if, you know, what made them think about long COVID in kids. And so we surveyed, you know, over 100 pediatric providers within the US nationwide. And we asked them, and sort of what you can see is none of these graphs could go. We asked about what primary symptoms, what primary exam findings, or were there any diagnostic tests you would do to look for long COVID, specifically blood tests, or would you refer? And you can see across all of these charts, we never had to go above 60 because there was a wide range and there wasn't one thing, not surprisingly, to help people understand it or sort of to label somebody as having long COVID. And so there's lots of work to be done. There were some sort of interpretations by providers that I think is backed by the research that we've been talking about. But, you know, it also implies that there are potentially some biases in recognizing long COVID in kids. And so, you know, we asked if people were more or less likely to diagnose long COVID based on these various demographic criteria. And interest in, I guess, the teenagers, adolescents were more likely to be diagnosed with long COVID than younger kids. Although I think I just showed that young kids can have symptoms of long COVID. Maybe we're just not recognizing the right ones. Females are more likely to diagnose in females than males. Going back to Peter's talk, but again, I think that that's, you know, you have to keep an open mind. Interestingly, there were some race and ethnic differences. And then whether or not somebody had COVID vaccines also helped or sort of made them think more or less about diagnosing with COVID. But this was about a year ago that we asked this, so this might be changing. And then also there were clinical scenarios that made people more likely to think about it. So if they weren't going to school, if there was some new sort of secondary diagnosis, if they were risk factors for severe COVID, or if they had family members, and if the family was asking about long COVID. So if the family had already thought about it, they're more likely to diagnose. So to switch gears slightly, so, you know, clinically recognizing long COVID in kids is one thing. Now, the question is, are there similarities with what we're talking about today with how kids, what's driving long COVID in kids? Is there an underlying inflammation? Is there antigenemia? You know, what is going on? And so we're, you know, I can tell you about one of the projects that we're doing, or at least a part of one of the projects. We are looking at neutrophil profiling. Many of the speakers have talked about how neutrophils are quite activated in long COVID. And what we're doing is we're actually comparing long COVID with healthy controls, but also with MIS-C, or children who had multi-system inflammatory syndrome in children, which was when children, weeks to months after they'd had acute infection would develop high fevers, severe GI pain, and end up in the hospital, sometimes in the ICU, for this hyperinflammatory syndrome. And what we're learning is actually they seem to be along the same spectrum, where MIS-C is the most severe version of this post-acute sequelae of COVID, and then long COVID is more of an indolent inflammatory response. And so we're looking, comparing, and we have a cohort that we're still building, and we're doing all of this testing here, but we're really focusing on neutrophil profiling. And just to give you one sense of what we're doing here, actually, this is in collaboration with what Mike had just shown, looking at the neutrophils. He had a nice video showing neutrophils activated. This, however, is spontaneous activation. So these are just neutrophils that are isolated and then placed on a chip, and we watch them. We don't stimulate them in any way. And what you can see is, compared to healthy controls, kids with long COVID have increased netosis, or neutrophil extracellular trap formation. And those with spike detected in the blood, those children with long COVID with spike have even higher neutrophil activation, pretty much to the same level as what you see in multi-system inflammatory syndrome in children. So their neutrophils are very activated. And this is looking at extracellular DNA that is a marker of netosis. And so then we started to wonder, why, what's driving these neutrophils? We do know that immune complexes in general activate netosis, but when we have spike in the blood and it's bound to antibodies, you generate these immune complexes and it activates NETs. And that's what we're seeing here. And it seems to do it in a dose-related manner. So the fewer immune complexes you have, the fewer neutrophils that are activated. And maybe that has something to do with, the differences that we're seeing in MIS-C, where there was a sort of severe inflammatory response, versus long COVID, which is more indolent. But that's speculation. We also have shown that when looking at cytokines in long COVID as compared to MIS-C, compared with healthy controls, long COVID also kids with long COVID also have increased cytokines at baseline. So not to the same degree as MIS-C. That was really cytokine storm situation, but they're more activated than what you're seeing in healthy controls. And so some of the other pediatric long COVID translational work that we're doing, we're doing spike antigen detection, and that's through the Walt lab. We're doing more complete neutrophil functional profiling, looking at some other assays, as Mike had talked about with Daniel Mia's lab at Mass General. We're also doing RNA profiling, bulk and single cell. Some microclot quantification, endothelial activation, cytokines I showed a little bit, and then planning to do some expanded antibody profiling as well. And this is really important because we want to compare with MIS-C to see if we can understand the spectrum of post-acute sequelae of COVID more, see what signals we can continue to learn from. With MIS-C, we want to compare to and contrast with adults to see if there are any age-related differences. Working with kids is really nice because they don't have those comorbid and morbid inflammatory conditions that also can rev up the immune system. And it's important to do this work because it provides a rationale for inclusion in biologically targeted clinical trials, which gets to my last point, is what can we do for children and adolescents with long COVID? Here's a summary. I just went out to clinicaltrials.gov last night and pulled all of the interventional trials for children who have long COVID. And you can see there's only 10 that are recruiting. And if you look down the list, there's only one drug study, and that is liraglutide, which is one that we're running. And I'm going to show you one slide on after, and we'll have more data by Dr. Fasano later in the day. But there's tons of studies in adults, and there's only, 14 here that kids are eligible for, excluding the ones that are already completed. And several of them are device or behavioral. Most of them are device or behavioral treatments. And so, which are important. And, but I think we need to include kids. And I don't think that there is a rationale not to. There's no treatment for long COVID. So I would just push everybody to go below 18 years of age, talk to the FDA. I would argue 12 and older. Here is our lorazepam study. We're enrolling 48 patients. It's at Mass General. It is a pediatric study, ages with a lower age of 7 years. We are expanding up to age 50, and that's to try to push our enrollment faster because we want to move faster to patients. The thing is we have to detect spike in the blood, and that's something we do as a screening process. You'll hear more about this study later, so for time, I will move on and just end with a summary that children can be affected by long COVID, and it is likely under-recognized. Inflammatory changes are seen in pediatric long COVID, and so far this seems to be similar to adults. And so that being said, we must include children in clinical advances, and there's no reason to wait years until we find an effective treatment and then roll it out to kids. So with that, thank you so much, and please reach out to me if you're interested in this long COVID study looking at lorazepam. And just thanks to all my collaborators, and thanks very much to PolyBio for including me in the talk.
Great, Leo. I agree that it is so important to keep doing research and treatment trials for pediatric long COVID, so thank you for setting that precedent. All right, our next speaker is Marcelo Huot. He's an associate professor in the Genomic Medicine and Infectious Disease Division at the J. Craig Venner Institute, and he'll be talking about mucosal systemic molecular signatures in long COVID. Yep.
Okay, well, thank you so much, Amy, and thank you all the speakers. This team is just so inspiring. And it's incredible to just share our data, communicate our science, especially in an era where there's misconceptions of science. And it's incredible to be here united, working together towards the clinical translation towards long COVID and chronic diseases. My lab is focused in JCVI La Jolla, and we are focused on chronic conditions. And I have a little bit of a different background because I have a PhD in immunology, but also a DDS, and I work in patients doing oral surgery. So it was always really common to me how the oral tissue harbors pathogens. And you can see the neutrophils communicating with the pathogens within the tissue. So viral persistence within the oral tissue has been something, pathogen persistence has been something very common to our field. And what I'm going to talk about today, it's sort of this oral and mucosal systemic relationships that we are learning. And a lot of it is work in progress, but it's evolving, and we are understanding much better what's going on. Also, I will show some of the tissue analysis that we are working on. I can skip this slide because we all talked about the background, but the idea is there's so many symptoms. There's a heterogeneity there in long COVID. And truly, this is a systemic condition. And what started as a local tissue then evolves to system-wide symptoms. But what we don't have is a biomarker to monitor clinically. And we don't have non-invasive testing like we had for antigen testing for acute disease. So there's a lack of reliable biomarkers that we can monitor, but also understand treatments and therapeutic interventions. My laboratory focuses on neutrophil biology. We're also very interested in mucosal immunology, so we utilize saliva and oral tissues and blood to investigate how neutrophil works. And we look into tissue and we ask the questions of host microbial interactions and how pathogens evade the immune system and the initial barrier to then create a lesion within the tissue. A lot of work that we do is related to proteomics right now and tissue analysis, which I'll show you later at the end of the presentation. But the idea that the technology in shotgun proteomics has evolved where you can do a protein digestion into your tubes and have very specific types of membranes that will filter out the peptides that you need if you're doing peptidomics or full proteomics. you can control where you're getting next after a mass spectrometry. So it gives you a possibility to understand not just the proteins, but also other aspects and post-translation modifications. And we received the funding to create this human salivary proteome, which is a database in which we investigate how saliva is in health and disease. And we have the highest number of salivary proteomes data sets. It's available to the public. So the advocates here and the audience, you can use the website and you can actually don't need to have any background in informatics, and you can use some of the data sets and do your own analysis, your own questions for subject by subject, but also by population. For example, our COVID studies population. So this is a salivary proteome database. When we started with this, we were really interested on neutrophils and inflammatory components because that has always been the goal of my lab. And we did shotgun proteomics in saliva and plasma. And we evaluated antibody responses as well. But clearly, what we found was that in the plasma, we had an upregulation of fibrinogen and many other markers, including myeloperoxidase, ELANE, and some other markers for neutrophil extracellular traps. So we knew that this not only is a immune dysfunction, but there's also a dysregulated vascular dysfunction there. But in this case, it wasn't long COVID. We were using convalescent patients. It was clear to us that that we would find later, from 2020, 2021, a persistent disease that would continue because these were really similar to other conditions. For example, type 2 diabetes, you find very similar type of proteomic responses. But here you can see in COVID subjects, the fibrinogen alpha increase and multiple neutrophil markers significantly increase in saliva and plasma. And we asked the question, okay, but this is not really long COVID, but because there were patients that were up to 90 days in our study, we stratified by severity. And as you stratify patients from no symptom to more severe, mild, moderate to severe, you see this sort of dose response on some of these markers. And we did, in this paper for this immune persistence, this regulation, immune and vascular, we also look into markers, and clearly fibrinogen was one of the top markers into the plasma, and neutrophil netosis-related markers were present in saliva. We also found fibrinogen in saliva. So we utilize normalization of proteomics. A lot of proteomics are done very differently, how you prepare the samples, how you run the analysis, do you use some label or no labeled. So we have to normalize our data to account for some of the difference that we see across many data sets into the salivary proteome. And we also have to start to think what type of features are we going to select so we can create, investigate the clusterings. So this classification allows the predictive modeling to happen all the way to a possible biomarker identification. A lot of the talks today mentioned the idea of biomarker, and more we have to think of multimodal markers. It can't be just from one assay. It doesn't seem like that's the way we're going to go in the field of long COVID. So that's sort of the model of biomarker that we're thinking right now. And then in collaboration with David Price and the Cardiff University, funded by PolyBio, we received the subjects in JCVI from David Price, and we were looking into the his long COVID population, and he shipped to us both cases and controls, but also plasma and saliva. So we are trying right now, we are working to replicate that initial study to see if some of these markers are aggravated or continue to happen in long COVID in this population. and it's still a work in progress. We started with evaluating a 45-plex panel for cytokines. And our goal is to understand some of these compartments and how inflammation is regulated into those fluids. In comparison of this T-cell responses, also inflammatory responses, and some of the neutrophil markers. Obviously, you won't be able to appreciate all the details of the data right now, but when you look at blood data and saliva data, you start to see IO8 and some of the clear markers for, I'm sorry, can you see my cursor?
Yes.
Okay. And you start to see IO8 and some other markers that are clearly really high for long COVID. And you start to realize that there are some clusters of patients that are healthy versus cytokines of patients with long COVID. And that's also, could be also the opposite. There are certain things really much higher in healthy and much lower in long COVID. And we are learning why. And there are some of these are anti-inflammatory cytokines that are much lower in long COVID subjects. The idea here is not to find always a linear correlation of the cytokines, but to start to dissect one by one and understand how is this impacting the mechanism. Although we were not able to find viral persistence into the blood of these samples, but we see that the plasma shows an incredible amount of information in terms of the pathogenesis. These patients do not behave similarly. This population, health versus disease, they're completely dysregulated. We have to go through in the molecular level and see those cytokines to understand what they are doing. Is this sort of it's a catch-22. Is it causing the disease or is it a consequence of what's already happening with the viral replication in the tissue? So the other component in proteins, right? So one level of proteins is evaluating cytokines. The other level of proteins is understanding the antibody response. A lot of us think of antibodies as a protective response. And we started to hypothesize that IgA actually could be pathogenic. And in certain sites, for example, in local tissue sites like the mouth, you might have more viral replication, might have higher an increase of IgA response to certain antigens. But lessons from kidney disease and some other autoimmune disease, excessive amount of IgA can also trigger some of the inflammatory response. But in blood, it wasn't surprising. We didn't see a huge amount of IgA response. We actually saw a decrease in comparison to the control. But in saliva, we see a robust increase of IgA response. For here, we're looking at S1, spike trimer, RBD, and nucleoprotein. So we start to understand that these patients have these markers for some either previous disease or active disease or some level of active replication. So we are now thinking and trying to use IgA isolated from these patients in next studies, and we're preparing a lab to do those investigations. We want to understand what's the function of these patients having this continuous IgA response. We are stratifying this data from Cardiff study. We know that there's a population that's severe and moderate and mild. And we know that a lot of the symptoms clusters into the neuronal system and circulatory system. Obviously, this is a system-wide disease, so there are multiple areas in the body that are affected. But what we want to do now, next, from this initial work that we completed, we want to look at the molecular. So, for example, the proteomics data, the IgA data, or the cytokines. How do they stratify according to these subgroups? And can we stratify according to the symptoms that we are finding? Before COVID, we were working the NETs and neutrophil extracellular traps. And we were working not just in sequencing what's outside of the NETs, but also in terms of the genes, but also the proteomes. we were very confident what were the core proteins for NETs, what's outside once they're produced. We were able to isolate NETs and sequence them. So myeloperoxidase, for example, Elaine, and some others are classic markers, and histones are classic markers of identifying when a neutrophil is releasing NETs. So we did the neutrophil proteomics, the undifferentiated neutrophil differentiated in the NETs. So now what we're trying to do in our lab is create a high throughput model where we can ask questions like I mentioned, can I use the IgA and from this subject and actually investigate? Can we investigate what's going on with the cytokines that are increasing the blood? So we are doing a cell culture model here, one of my students here in the lab, Gavin Lee, and the idea was to have undifferentiated cells with no PMA or PMA, which induce NETs. It's a chemical that's induced NETs in the lab. But then differentiated cells, seeing the baseline, and then understanding once you add this chemical, you have this netosis formation. The goal is not, the goal is to make high throughput experiments where we can ask many questions. So for example, here is how are the antigens impacting, SARS-CoV-2 antigens are impacting netosis, and we're finding that certain antigens versus others really impact, like RBD and S1. So
Marcelo, can
yes.
you wrap, we have one more minute to wrap up yours. Thank
Okay,
you.
yes, and we quantified these and we found a lot of that some of the spike proteins are the biggest inducers of NET, and we are learning the concentration. And this is really important for what we're going to do now with IgA specific to these antigens. So that's sort of our working model here. We think that IgA purified from oral tissues can actually impact the netome on the NETs and what type of protein. And we believe that this could be a lot of the future of the mechanistic of the pathology that we are studying. I'm just going to skip one slide here, but just to remind everybody, there's evidence for multimodal biomarker from all the stocks today. We just have to figure out how can we make these clinical and validated diagnostics, whether you're using the microbiome, IgA, netosis, or some of the other markers for T-cell. I think it's time for us to unify into finding out to put together these multimodal biomarkers. I want to acknowledge my team in the lab and the JCVI collaborators, Amy from PolyBio and Mike, and the team from Cardiff and Mount Sinai and the funding. Thank you.
Awesome. Thanks, Marcelo. That was packed. Really appreciate it. Okay. Our next speaker is Rajal Chan. He's an assistant professor in the Department of Neurology at UMass Chan Medical School, and he'll be talking about the impact of SARS-CoV-2 infection on Alzheimer's and neurodegenerative disease.
Thank you very much. Very excited to share our work here for doing SARS-CoV-2 infection and really figuring out Alzheimer's disease, which is something like in the forefront of what we do in our lab. So we do basically a lot of studies on neurodegenerative diseases, and Alzheimer's is just one of them. So this work, we thank PolyBio to really fund this work, and it's a collaboration between a few investigators here at UMass Chan Medical School, including me, Elaine, and Fiacra. All right, so let me give a real high-level view of how we think about viral infections as individuals or as patients. So of course we have short-term adverse outcomes. So these are the things when you get infected, you actually immediately see the symptoms and you feel it, and you know that you have to deal with it immediately because you have fever, cough, chills, shortness of breath, these things, and you are the virus infection is really in your mind at this point in time. But once you are okay and you clear the virus and you're fine, there are midterm adverse outcomes, and long COVID, I think, fits into this category, and you have brain fog, extreme tiredness, and so on and so forth. And this is the major reason why we really want to think about long COVID, right? But I'd like to step back and also try to convince people that there are also long-term adverse outcomes, and we are talking about maybe 10 years down the road or 20 years down the road, where you might get the infection so early on and you don't really think about it, but later down the road, you get things like cancer, autoimmune disease, and even dementia. So like cancer, we have the example of HPV causing stomach cancer. As for autoimmune disease, there's examples of Epstein-Barr viruses being tied to multiple sclerosis. And what I'm going to talk today is whether the viral infection early on can lead to late stage. I mean, after a long time, you get like dementia, like Alzheimer's disease. Okay, so let me give like a brief overview of what I think, or maybe what the field is thinking of Alzheimer's disease in relation to viral infection. So early on in life, at stage 1, you have common neurotropic viral infection. These are probably viruses that circulate around the environment, and one of the more common ones are herpes viruses. So we get infected by them, we clear it, we are okay, no problem, right? But as the virus gets infects the person and it goes to the brain and starts some sort of neuroinflammatory process, it activates specific biological process for the cells in the brain to try and fight off the virus. And usually, most of the time, we do very well in that. So the virus stays benign, you don't see it, you don't have symptoms. But in some people, over the course of the life, as the virus is infecting the brain, you produce something called amyloid beta, which is a pathological molecule. We think it's a pathological molecule, but it's also a molecule that's meant to help fight off the viral infection. So it's a good thing, but if it's dysregulated and a lot accumulates and we cannot do much about it, it starts to poison sort of the environment around it, and things die, and it triggers a cascade of events, including phosphorylated tau tangles, and leads to a later stage 4, number 0.4 here, neurodegeneration due to the pathology resulting in Alzheimer's disease. So the whole reason for this slide is to convince, or at least tell people that the viral infection can cause later downstream consequences. It's not apparent initially. And so here in our lab, what we do is we try to really study this kind of effect of how viral infection can cause Alzheimer's disease using cerebral organoid models. So cerebral organoid models are basically artificial, you can say artificial brain models that we use induced pluripotent stem cells derived from individuals to make a sort of mini model of the brain. And the process is pretty deliberate. So you put the iPSCs or the pluripotent stem cells together, and you try and induce its pathway towards the brain, and you embed it in some matrigel, which is the environment that sort of nourish this newly formed brain, and you start growing it in a incubator, and within 3-4 months it starts to develop. And when we actually profile this artificial system, it has a lot of different cell types of the brain, including stem cells, neurons, astrocytes, microglia, and so on and so forth. And once we have this artificial system, we can then dissociate them and perform infection assays. So we can expose these organoid cells to viral particles. And in this case, I'm showing you data for herpes virus infection that carries a green fluorescent protein. So when the virus infects the cells will turn green, and this is what we see. So how is all this related to Alzheimer's disease? So I'm going to show some data here for HSV-1 infection. And we actually infect our cells, our organoid cells, and use a technique called RNA sequencing to sequence the gene expression of the cells and compare that between infected versus uninfected. And we have a method to look at all these genes and these differences to find out whether they are associated with any of these disease or human traits. So this graph over here, I have like Alzheimer's disease, noted as AD, and maybe other diseases like ALS, multiple sclerosis, Parkinson, and so on and so forth. So you can see that when we do a comparison for the gene expression differences and Alzheimer's disease, we see there is a clear enrichment. So it's a gene set enrichment, and we see that for Alzheimer's disease, but we don't really see that for the other neurodegenerative disease. So the next question is to ask, what about SARS-CoV-2 infection? Do they also have a similar effect? And so we started off this project by exposing the same organized cells, but this time instead of HSV1, we do it with SARS-CoV-2, and we actually don't see much infection. So over here, I'm putting the red boxes. If there's infection, we'll see either the neocapsid protein being targeted or the spike protein being targeted by the antibodies, but we don't see that here. But when we do a quantitative PCR, which means to really look at the viral RNA, we do see it at MOI of 1 after 48 hours. So this result suggests that there's active replication of the virus, but the virus is inhibited by transcription or translation. So to really investigate that, we did RNA sequencing of the organoids infected with SARS-CoV-2. So we have basically a few time points. One is 6 hours and one is 24 hours, and we have 3 replicates for each of these time point with the equivalent controls. So over here on the left is the raw counts of the genes of the virus being counted during the RNA sequencing. So you can see from samples 613 to 615, they are uninfected. 616 to 618, they are infected after 6 hours. And so the ones after is the 24 hours. And on the right here, you can see that there's like, some viral gene expression changes, and between 24 hours and 6 hours, there is a slight drop after 24 hours compared to the 6 hours, but in general, the viral RNA is deaf for all the different genes in SARS-CoV-2. So now the question is to ask, comparing between the infected and the uninfected for the 6-hour condition and the 24-hour condition, what's actually occurring. So on the left here, I'm showing the 6-hour condition between infected and uninfected. You see a lot of different genes being differentially expressed. So the x-axis, the horizontal axis is the log fold change. So things on the right, they are higher expressed in the infected, and the things on the left are lower. But the point to show here is that when we compare the one on the right, which is 24 hours, we actually see not many changes. But one of the change here is a gene called GFAP, which is very interesting. It's also found in the 6-hour, and it's actually a marker for astrocytes. So this result suggests that the astrocytes in the organoids are the ones being infected, and they are proliferating, and then subsequently dying at 24 hours. Right, so what we think is going on? There's basically a program that even though when it's infected is growing in large numbers, they do undergo cell death as a way to curb the infection. And when we perform the same gene set enrichment analysis to actually view like whether that's associated with Alzheimer's disease or not, we actually don't see it. So in all the cases, whether it's a 6-hour or 24-hour, or we can also compare the conditions between 24-hour and 6-hour, and we also don't see it. But when we compare 24-hour and 6-hour, then some of the other traits and diseases are found to be enriched, which means that there's something else, but that's nothing to do with Alzheimer's disease. Okay, so this is my last slide, and we want to really think of what's going on. So some further research directions that we are actively working on is to ask, these astrocytes, they are infected, they have a natural way to curb this, infection. And so why do astrocytes have this power to curb the infection, and whether we can learn its biology and use it as some sort of therapeutic angle for treating like SARS-CoV-2 infection. The other thing is maybe the way that Alzheimer's disease is triggered is not because of an actual infection, but it's because of exposure to antigens like spike. And that actually alters the condition such that it becomes more susceptible to HSV1 or herpes virus infection, and then can induce Alzheimer's disease. Or that herpes viruses can go into latency within the brain, and maybe when SARS-CoV-2 infects it in this way, it will trigger the reactivation of the herpes viruses, then leading to Alzheimer's disease. And so these things are still things that we are thinking of that might be able to trigger Alzheimer's disease, but it's still an active area of research. So I'd like to thank everyone involved here at UMass Chan Medical School, Ben Redhead from ASU, and the folks from Harvard Medical School, and of course, for once again, PolyBio for funding this research. Thank you very much.
Thank you, Rigel. So important to understand potential effects of many viruses on the Alzheimer's disease process. So really great research. Then I'll introduce the next speaker, which is Zian Zheng. He's a professor of medicine in the Division of Cardiology at UCSF, and he'll be talking about the COVID post-SCD or post-mortem systematic investigation of sudden cardiac death study.
Good morning, everyone. Well, it's morning for me over here in San Francisco. So for those of you who are not familiar, I'm going to give you guys a little background of our study and why a electrophysiologist is on the long COVID study here. And so one of the big question marks in cardiology and sudden cardiac death is, despite the fact that it's the #1 killer by AHA statistics, more than the top 5 causes of death combined, the way we define sudden cardiac death has traditionally been a presumed definition. If someone collapses and dies suddenly, they're presumed to be cardiac. This is an example of that. A few years ago, or now 8 years ago, one of our Supreme Court justices died suddenly at a ranch in Texas. His death was called a sudden cardiac death without an autopsy, let alone the coroner even viewing the body. It was defined as a sudden cardiac death. And this is an example, but occurs hundreds of thousands of times a year in the US and millions of times in the world. So we really don't know what causes sudden cardiac death. In practice, what we see are sudden cardiac arrest survivors, the small fraction that make it to the hospital. And we presume all the other causes to be cardiac, but we don't really know. So this is the genesis for a collaboration I initiated with our medical examiner in San Francisco. Can we surveil all of these sudden cardiac deaths and define the actual underlying causes? San Francisco is a medium-sized city, 850,000, about double that during commute hours. It's very diverse, so allows us the opportunity to extrapolate our findings to many different ethnic groups. It has approximately 10 hospitals, and very importantly, by California state law, any out-of-hospital death gets reported to the medical examiner. So I just have to work with one person, the medical examiner, to not only track every sudden death, but also then capture them for further investigation. And so any ambulance, any emergency department, somebody who dies suddenly out of the hospital gets transported to the medical examiner. Now, the problem is most medical examiners and coroner's offices don't have the bandwidth to investigate all of these natural deaths, because their charge is to investigate unnatural deaths, such as traumas or homicides or suicides. So the natural deaths, as I said, are all presumed to be cardiac most of the time. And so we forged a collaboration where we look carefully at every single case. And luckily, San Francisco is not so big that we couldn't capture every single case. A few years ago, we published in Circulation. This is Dr. Moffitt, the amazing medical examiner collaborator that I work with, who agreed to my crazy idea to do an autopsy in every single case. And I'm not going to walk you through every single panel here, but we start with every single death reported to her office. And we take the ones that are defined as cardiac arrest by the paramedics. They're all presumed to be cardiac, and then she does an autopsy, and she achieved an incredible autopsy rate of 97%, with 3% of family members declining an autopsy. So this represents a complete picture of out-of-hospital deaths, people who are well 1 minute and then die suddenly the next minute. And so in this publication, these are the first 3 years of all of these sudden deaths. And one of our huge discoveries was, guess what? Only half the time are they arrhythmic, meaning these are cases that would have been saved by a defibrillator in red here. You have the myocardial infarction causes, the chronic coronary disease causes, the cardiomyopathies, hypertrophy, etc. And then over in green, a full 40% of cases were not even cardiac. And we're talking about overdoses, infection, pulmonary embolism, strokes, aortic dissection, etc. And so this has really changed the way we define sudden cardiac death. Now we define them as presumed sudden cardiac deaths if it hasn't been confirmed by autopsy. And so this has led to a number of discoveries in numerous publications. We coined the term sudden neurologic death. These are cases that were called cardiac deaths, but in fact were neurologic causes upon autopsy. We were the first to discover overdoses masquerading as sudden cardiac deaths. We're the first to define HIV as a huge risk factor for sudden cardiac death. And then occult device problem, pacemakers and defibrillators failing suddenly and causing sudden deaths. So how does this affect, or how has this set us up to do a study where we're looking for long COVID? So my team, in addition to collecting all these autopsy data, reach out to family members and have them see if they are willing to consent to donate all the tissues that are collected at autopsy to our biobank. And my team has been amazing in achieving approximately 40% consent rate. And so these tissues represent all of these underlying causes of sudden death, in addition to some trauma-controlled deaths, as you see here. Why is this important? Because this represents the inverse of what we've discovered in other human tissues in long COVID studies, which generally capture inpatient deaths. And inpatient deaths, I'll show you in a second, are very different than out of hospital deaths or ambulatory patients, which is where long COVID exists and where we want to find these answers to long COVID. So COVID post-SCD is a find the reservoir type of study. What we're doing is to look for the cause of the long COVID by examining these tissues across all these organ systems. And again, these are ambulatory persons. These are people who are well at home, at the grocery store, in the park, and they're well up to the time they die suddenly of a sudden arrhythmia. This gives us a chance to have a snapshot of a person walking around fine. What is going on with SARS-CoV-2 in those cases? We can look at viral persistence, immune dysregulation, interactions with chronic diseases such as diabetes, because we have the power of hundreds of cases, endothelial dysfunction, metabolic dysfunction, or cell-cell crosstalk. Now, again, I want to highlight the fact of the huge difference between out-of-hospital deaths. So here in sudden deaths, approximately 80% die suddenly after your cardiac arrest, and 18% make it to the hospital, 6% ultimately survive. And again, this is the population in whom most autopsy tissues are derived. This is the population in whom we're studying out of hospital. So, sorry, let me... So here, as I alluded to earlier, is an example of the limitations of specimens from hospital autopsies. This is a transcriptomic study, an expression study that we did of a myocardial biopsy from a person who survived their survived their acute traumatic injury to live in the ICU for about approximately a week. This is over on the right, and compared to a trauma death that died instantly at the scene. So when you die instantly at the scene, the transcriptome of that heart represents and reflects the steady state, the person walking around well 1 minute and then died suddenly. Those changes, post-death changes, aren't going to be reflected because expression arrests when you die. Now, if you live in the ICU, as you see here, that heart, an equivalent traumatic injury, that person lived for a week, very, very inflammatory, markers of inflammation were much increased in that case as compared to the instant trauma. So this shows you the potential limitation of making conclusions, for example, about inflammation in tissues derived from autopsy studies. So the COVID post-SCD study, we have the power, as I said, we started this study in 2011. So I have almost a decade of sudden deaths, of comparisons, of controls to compare to post-2020. And so what we're going to do then is examine tissues from post-2020, take advantage of this surveillance study to look at pre and post-COVID. And so we start with all of these autopsy specimens that Dr. Moffat's collected for us: brain, gut, heart, liver, lungs, kidney. And we're able to study not only SARS-CoV-2, but we're also going to be examining these tissues for many other viruses. We've fixed these tissues in FFPE blocks with contiguous sections, which allows us to do all the array of omics, RNAscope, geomics, xenium, et cetera. And then we also have more specific single organ autopsies that we can look at, for example, in the brain, many different areas within the brain to see if SARS-CoV-2 is heterogeneously distributed in the brain in long COVID. And so not only can we examine each of these tissues in a single person, but then we can examine them across different populations that are ambulatory in our study. And so as a teaser, this was an example of a post-2020, a person who had recovered from COVID and was well at home and then died suddenly of their acute MI. And we found SARS-CoV-2 spike RNA, which represents active expression of SARS-CoV-2 months after recovery, and had gone home. And so there's active expression of SARS-CoV-2 in brains of a person who is walking around at home. So what have we done so far? A lot of this is to set us up for all these systematic studies. What we've done is already processed 42 matched sudden deaths pre-2020, 208 biospecimens from all of those different organs I mentioned. And now we have, we're comparing them to 45 cases we've had so far post-2020 to give us a survey of everywhere from relatively recent infection to now, potentially 4 years after initial infection. And we have 220 biospecimens from post-2020 as well. So a total of 400 total biospecimens, 3 replicates each. And so we'll have the power to look at all these different interactions. And very importantly, we've started to do initial Q&A studies, and we've discovered EBV transcripts that we found in the gut, for example, in pre-2020. And so this is just an initial overview of our study, and we hope to share more in the months and years to come, and stay tuned. This is a reminder for me to acknowledge Dr. Moffat, Tim Henrich, who I work with closely with these molecular studies, and also, of course, to PolyBio for support of our study. Thank you very much.
Thank you, Zan. It's great that you can get access to so much tissue that we cannot otherwise get from biopsy. It's a really thorough study. So I'll introduce our next speaker, who is Gene Tan. He's an assistant professor in the infectious disease group at the J. Craig Venner Institute. And he will be talking about deep characterization of antiviral immune responses and long COVID pathogenesis.
Yeah, as Amy had mentioned, my name is Gene. I'm an assistant professor here. I'm a virologist and some vaccinologist, and my lab is interested in characterizing the antibody response and mechanisms of protection, and if not disease, pathogenesis. So this is, sorry, I'm having problems. All right, so I think I've shared this slide before, but putting it here again to remind us of the case control study that Dr. David Price and colleagues designed to identify risk factors associated with the development of long COVID. As participants of this study were collected at Cardiff University in the UK, and the study is composed of 2 groups, both of which were composed of people or participants previously infected with SARS-CoV-2, but where one eventually developed a set of symptomology that's associated with long COVID, while the other never developed any chronic disease, which we labeled as convalescent controls. So for today's talk, I'm providing some updates on our serological characterizations against SARS-CoV-2 and other viruses, as well as generating tools to interrogate the neutralizing antibody response to different regions of the spike protein and just kind of really figure out what's happening with the disparity between the neutralizing titers between people who have long COVID and not. So we finally finished performing the new assays of all the participants recruited for the study. This represents over 200 long COVID participants and around 120 convalescent controls. And this is only for the Wuhan ancestral strain. As we had reported last time, our preliminary observations held true, and we see the significant disparity in the neutralizing titers between participants who have long COVID and the ones who do not. So what is really jarring is the observation, if you're to look at on your right, is that the difference between these two are titers coming from the female participants, while there are really no significant difference between titers and males. So this is quite pretty jarring. So in order for us to explain the cause of the disparity in the assays, we thought it would be a good idea to construct novel antigens that can deconvolute the neutralizing titers or antibodies against the different domains of SARS-CoV-2. So we accomplished this by grafting SARS-CoV-2 domains that are major targets of neutralizing antibodies, such as the receptor bind domain, or RBD, or the amino-terminal domain, or NTD, onto a backbone of a spike that the general population has never really seen or exposed to. So you can see this, I'm not sure if you can see using, if you were to look at the upper left-hand corner. So the pink spike protein is SARS-CoV-2, and then the blue spike protein is from SARS-CoV-1. So we've never been really exposed to SARS-CoV-1, but we have been exposed to SARS-CoV-2. So in essence, if we make these, we accomplish this by grafting SARS-CoV-2 domains onto the backbone of SARS-CoV-1. And in essence, we make these chimeric proteins when you make, when you basically make these chimeric S proteins that has the RBD or the NTD SARS-CoV-2 in the background of a SARS-CoV-1 spike. Then when we essentially measure neat activity against RBD or NTD. So in the figure on the right, you will see that the NTD chimeric has the NTD of SARS-CoV-2 domain in pink, but has the body of SARS-CoV-1 in blue. So we then incorporate these spikes into our pseudovirus assays, neutralizing assay, and we perform the assays. But one thing that we had to think about is that one caveat to this assay is that we do know of cross-neutralizing antibodies that can neutralize both SARS-CoV-1 and SARS-CoV-2. So to account for that, we subtract those values against SARS-CoV-1, which is represented by the bottom figure, the graph. It's called long COVID cases TOR. And that would be the titers against SARS-CoV-1, which represent cross-neutralizing antibodies. And so we subtract those from our values for SARS-CoV-2, the chimeric protein, and therefore, and from there, we would have the value, the neutralizing value that's specific for the SARS-CoV-2 domain. So what we see here is our preliminary data. It's interesting. One is that on the left, you will see that there is basically no difference in the RBD-specific response between long COVID and convalescent control, suggesting that both groups can mount comparable neutralizing antibodies that target the RBD. However, we do see a drastic difference against neutralizing antibodies between against the NTD between people who have long COVID and those who do not. Where long COVID participants have lower neutralizing antibody titers against NTD. And if you were to further break that down, the NTD data down between males and females, you will see that while there is a trend in males, there is a significant difference in neut value in the female participants, where long COVID individuals have lower titers against the NTD, something that is reminiscent to what we saw in the neut data against the ancestral Wuhan or the full-length SARS-CoV-2 spike. This is still preliminary data, and we still need to validate our constructs using monoclonal antibodies and other chimeric constructs. But it's quite interesting to see that there is no difference in the RBD-specific responses, but there are some differences in the NTD response. And that might explain why there is a disparity between why long COVID participants have lower neutralizing antibodies against the SARS-CoV-2 in general. So to switch gears a bit, there are published reports that show that individuals who have progressed to long COVID tend to have higher binding titers to seasonal coronavirus. So what we thought was that since people are seeing antibodies that can bind to it, can we establish a neutralizing antibody assay that can measure this? So determined this was also true for this specific study. We developed this assay to measure those antibodies that can neutralize both seasonal coronaviruses. As you can see here on the right, long COVID individuals have slightly higher neutralizing activity against a seasonal beta coronavirus, OC43, but have comparable antibodies against alpha coronavirus, NL63. This suggests to us that perhaps there are some conserved epitopes within the beta coronaviruses that may not be beneficial. Oftentimes, when we talk about cross-neutralization or cross-neutralizing antibodies, we often think that it might be beneficial. But in this case, and what other people have seen is that there are higher neutralizing or higher binding, and for what we've shown right now, higher neutralizing antibody titers against a related seasonal beta coronaviruses, while there is none against a more distantly related alpha coronavirus. We still need to extend our work against other seasonal beta coronaviruses, such as HKU1 and other alpha coronaviruses, if what we see holds true. Let's see. So lastly, there's data to suggest that some human herpes might be reactivated during long COVID. We want to evaluate that by performing ELISAs, using the antibody response as a biomarker of past evidence of reactivation. I know this has been mentioned probably previously already. It's been published, but we want to see whether this holds true for the Cardiff study. So given that this study is a case control where participants were recruited based on disease phenotype, we anticipated there may not be much of a difference in the IgM shown on the top response to ABV, since the data collection is potentially past the activation period and peak IgM titer. So, but we did see an overall significant higher IgG response to EBV, the viral capsid antigen 18 in people who have long COVID, and most significantly found in the female population of our group. Again, since this is a case control study, current data suggest that either participants with long COVID had a past event of EBV reactivation. Due to the higher IgG response, we did not really see a significant increase in the IgM response, which is a little bit more reflective of a very recent reactivation of the virus, or that long COVID subjects have a higher prevalence of high IgG against EBV. So again, this is a better, I would say, interesting future approach would be to do a prospective study design whereby you would follow people with EBV, high EBV or low EBV IgG and see whether or not one or the other would have a higher chance of developing long COVID. And so again, but what's interesting is that we did not see a difference in the IgG response to another herpes virus, varicella zoster. What's interesting for the EBV is that EBV is a latent virus that targets B cells, and that could potentially influence how people who have long COVID or previously have higher IgG EBV, higher IgG response to EBV could have a dysregulated response to long COVID compared to those who do not. But against another herpes virus, there appears to be no significant difference between people who have long COVID or not, those who do not. So I'll end with our conclusions so far. Neutralizing activity was significantly lower in long COVID cases and notably driven by females. We generated novel chimeric S protein that allows us to identify SARS-CoV-2 spike domains targeted by neutralizing antibodies. There appears to be no distinct differences in the neutralizing antibody response to the receptor bind domain of SARS-CoV-2. However, long COVID participants appear to have a lower neutralizing antibody response against NTD, seen more distinctly in the female population of the group. Limitations, well, this is just preliminary, driven by the number of females. We don't have enough males and potentially might be as well. We will still need to finish the rest of the samples to validate our observation, and probably would have to generate other chimeric proteins to really validate what we see with our current model. Long COVID participants have higher neutralizing antibodies against related seasonal beta coronaviruses, OC43, but not against a more distant related alpha coronavirus, NL63. And I think this is pretty reproducible at this point in time, and seen by others, is that long COVID participants have higher IgG responses to EBV, but have comparable IgG responses to VZV or chickenpox. And so the real people to thank are the ones who are actually doing the work, and the funders. I'm very thankful for my own team. Jesus, who has been taking the lead on this project, has been phenomenal at doing a job at it. We had brilliant support and outstanding and brilliant support from Terry and Gigi and Noah. We also want to thank Chris's team, Marcelo's and Richard's team, JCVI. And, you know, thank you to and acknowledge Amy Pro and PolyBio for the funding that you provide to be able to do this. If not, we wouldn't have been able to do it. And last but not least is a big thank you to David Price and Helen Davis. Very thankful for your generosity for sharing your samples with us. Thank you.
Great. Thank you, Jean. Really comprehensive. Our next speaker, I'll introduce then. It's Chiara Gianarelli. She's an associate professor in the Department of Medicine and an associate professor in the Department of Pathology at NYU's Grossman School of Medicine. She'll be talking about deep characterization of SARS-CoV-2 cardiovascular reservoirs and plaque formation.
Sorry, little hiccup here. Okay, thank you, Amy. I will be talking about our work on cardiovascular complications of SARS-CoV-2 infection and COVID-19, really stemming from the very beginning of the pandemic, where we realized that among all the cardiovascular complications, that was one in particular that was emerging and in the acute phases of the disease, and now we're learning more and more, becoming complications in the long term in individuals who were infected during the first wave of the pandemic, that is myocardial infarction. And in particularly, we are focusing about the type of myocardial infarction that results from the accumulation of lipids and material into arterial vessels that progressively over time results into a narrowing of the lumen of the arteries that bring blood to the heart, and ultimately culminating into a very important complications that is myocardial infarction or heart attack. And what we are trying to understand is how SARS-CoV-2 infection impacts atherosclerosis into this progression, contributing to the acute, but particularly into the long-term ischemic cardiovascular complications that now we know are increased at 3 years of follow-up. We started our work working on coronary autopsy specimens with patients who died of COVID-19. And in these patients who presented to the hospital with infection, we found different type of pathologies into the coronaries, ranging from normal coronaries into more overt disease, fibroateromas, that is one of the stages of atherosclerosis. And all these patients had underlying atherosclerosis. They had hypertension, so high blood pressure. They were mostly overweight. Most of them had type 2 diabetes. So they were patients that were at very high risk for severe COVID-19. And some of them presented with cardiovascular manifestation consisting in stroke or a myocardial infarction.
Chiara, one thing, your slides aren't moving, so I think you need to share full screen, and then I think they'll go for it. No problem.
Okay, I think it's not. I need to restart this sharing because it was moving on my screen.
Okay.
And thank you for telling me.
Yeah, no problem.
Do you see them?
Yeah, now I see them. Perfect.
And do you see the full screen?
Yes, now I do.
Okay.
So that's great.
So this is what I was telling you about. This is just a template that shows all the clinical characteristics of these patients. So to summarize, we took these coronaries and we analyzed these coronaries using a combination of methodologies that allows the detection of the viral RNA in these tissue samples, and also artificial intelligence to reliably and unbiasedly quantify with very specific copy number accuracy the presence of viral RNA in these specimens. And using this approach, we were able to see that when we were looking at the vasculature that is annotated here, I hope you can see my mouse moving, and the perivasculature, where a lot of immune cells are also contained, we found that the virus was more present in the vasculature itself, in the arterial wall of these vessels, as opposed to the perivascular fat, which was very interesting finding. We looked into the cells that were containing this viral material, and we found that the majority of them were macrophages. Now, macrophages are a factor of the immune system. They are present in these lesions. They are present also in coronary arteries. And we saw that in this context of atherosclerosis, where macrophages contain more lipids that accumulate and they're trying to phagocyte, also contain more spike viral RNA, and also spike sense viral RNA. So a type of RNA that is only expressed by the virus when it's replicating, which was a very interesting finding. We also looked into differences between macrophages and foam cells that are all these lipid-rich macrophages, and we quantified the presence of viral RNA, and we observed that foam cells contain more of these, the virus, indicating that somehow these cells were more susceptible to infection. And when we try to understand why, we figured out that while macrophages were able to clear up the virus more efficiently, the virus was more persistent in foam cells, so these macrophages rich in lipids, that were less capable of producing an important antiviral response over time, that is interferon response. So at that point, we wanted to really ask the question, because these were autopsy specimens, whether SARS-CoV-2 can increase proteogenic inflammatory response in human coronary tissue. And we used this model where we can take samples from the operating room and we can culture them for a certain period of time in our lab. And we infected this tissue, and we observed the accumulation of different type of viral components of the SARS-CoV-2 viral genome in these infected tissues, as opposed to the non-infected one or mock-infected. And this tissue really was able to mount a very important inflammatory response that is obvious, of course, in response to an infection, and in this context is also speaking for pro-atherogenic cues that could contribute to aggravated atherosclerosis. Next, we ask, how can SARS-CoV-2 infect macrophages, or is this a mechanism that is related to the phagocytosis of the virus itself and not really an infection? And what we found is that, first of all, macrophages express more of this NRP1. NRP1 is a protein that is expressed, I'm sorry, these slides are moving strangely, is expressed by cells that are infectable and really facilitate the infection of SARS-CoV-2 and infectivity in different cells in our body. And this was published in early stages of the pandemic by two different groups. What we found is that neuropilin-1 is expressed more in foam cells than in macrophages. So this was a very interesting interesting finding, potentially explaining why foam cells were more infected in our system model. We found also more of these NRP1 positive macrophages within the coronary arteries that contain the virus. And here you can see the distribution of these cells in the human coronaries. And interesting, when we analyze macrophages and we silence the expression of NRP1 in macrophages and in foam cells, the infection of the virus was reduced to some extent in a significant manner, suggesting that NRP1 contributes to the infectivity of these cells. So based on these findings, we provided evidence that SARS-CoV-2 entry is increased in foam cells, and it's dependent, at least in part, on an NRP1 expression that is increased in foam cells that are cells present in atherosclerotic lesions. Foam cells are less capable of mounting interferon response and antiviral interferon response, so they are less capable of reducing viral clearance. And also they respond by producing important cytokines like interleukin-6 and IL-1 beta that are driving coronary artery disease and also heart attacks. So now we're trying to answer or follow up on this question, trying to really answer pertinent question related to the influence of SARS-CoV-2 in long-term consequences of COVID-19. And we want to know whether there is a SARS-CoV-2 vascular reservoir and what is the effect of this vascular reservoir on immune responses and cardiovascular complications throughout the atherosclerotic progression, and how the immune reprogramming that is potentially dictated by the virus plays a role in driving the increased risk of cardiovascular events in the long term. We have access to different cohorts that we started collecting before the pandemic. It's called the ATHEROIN study. This is an ongoing project that involves also the biorepository and very careful and annotated patients, where we collect not just blood, but also carotid specimens and coronaries in the case of individuals that require a heart transplant. And we are also able to collect from an upgrade of this, an updated study that is called AtheroCore, additional tissues like bone marrow and lymph nodes. So we will leverage these tissues for performing in-depth analysis to try to answer the question that I told you about in the previous slide, try to understand whether there is a difference in immune cell composition and function and presence of viral debris, whether proteins or SARS-CoV-2 RNA, before or after the pandemic. And also we will leverage on our ability of performing these ex vivo studies using human tissue, trying to understand how this tissue reacts to infection and clearance of the virus, as well as whether there is a different response of this tissue based on whether the tissue is from a patient who presents or meets the criteria for long COVID or not. So with this, I would like to thank PolyBio for supporting our research that is ongoing and just started actually this year. So I will be happy to share more updates in future meetings.
Great. Thank you, Chiara. That was an excellent overview. I'll introduce our next speaker now, who's Chris DuPont. He's a professor in genomic medicine, environment, and sustainability on synthetic biology, also at the J. Craig Venner Institute, and he'll be talking about an update on the tissue analysis pipeline from small fiber neuropathy to endometriosis.
Excellent. All right, so I know we're running behind, and I really need to keep this to under 10 minutes, so hold on to your hats. Here's our overall approach. So we have been working often in collaboration with Amy and PolyBio in lining up the availability of tissue samples from a variety of sites that are often unstudied and that we believe may actually act as reservoirs for pathogens from past infections. And so our overall approach is to collect these types of tissues. And at this point, some of the ones I'm going to talk about are lung and endometrial tissues from patients with long COVID and endometriosis, respectively, but also looking at intestinal pinch biopsies, skin punch biopsies, and surgical samples from patients getting CCI surgery, i.e. C1 ligament and nerve. And we then essentially try to establish a whole cell catalog for these tissues. And so this involves unbiased, untargeted shotgun metagenomics and metatranscriptomics. I'll talk about some of the ways that we deplete these samples of host material so we can enrich for the putative pathogens and microbes available. We also conduct single nuclei RNA sequencing to develop a cell catalog of the host cell types within these samples. And I'll show some of the emergent data from that. And finally, we, in some of these samples, are starting to conduct parallel spatial transcriptomics, which really needs the cell catalog from the unbiased metagenomics, metatranscriptomics, single nuclei RNA-seq. And then this is a later part of the pipeline. This feeds into a bunch of artificial intelligence learning modules, so machine learning with differential network abundance analysis to look at which interactions between microbes, host cells, and phenotypes of the host are really driving the differences in the relationships here. So why would we study the human microbiome and the virome? Well, since about 2003, 2005, it's been abundantly clear that the microbes that are living in and on our body are far more diverse than we thought. So microbiology and in terms of human health has had this laser focus on pathogens up until about 15, 20 years ago. And now we're beginning to discover that every body site within the human body that's an epithelial layer will host very specific microbes. So the epithelial layer on my skin has a different microbiome than the epithelial layer within my duodenum, for example. And so these are distinct across the human body, and they're influenced by environmental genetics. Prior to a lot of these projects spinning up, we had started putting together pipelines. Our team at JCVI way back and the mid-aughts had developed pipelines for taking individual sequences out of metagenomes and metatranscriptomes and reassembling them into an entire microbial genome. And this is akin to going into a big box store, grabbing every single puzzle on the shelf, flinging them in the air, and then be able to recreate a single puzzle without ever seeing the box beforehand. And so we've made all of these methods publicly available. What does this derive? So this is from samples from oral plaque. Each of these little circles in these constellation plots are reassembled high quality genomes. Each cluster of connected dots are species of bacteria with each of those circles, individual genomes. Some of these are incredibly personalized, so that one BC0 in the very dead center with about 65 points in it. It turns out this is a microbe that every single household that we measured in the study had a very unique form of this genome. Essentially, it was a personalized part of their oral microbiome. So these methods are in place. However, this was oral plaque, which is highly enriched in bacteria, not tissue samples. And a lot of those tissue samples that I mentioned previously, if you were to do unbiased sequencing without host depletion, you would have mostly 97 to 99% host DNA, and you would not be able to see the rest of it. Another point out of this plot is over on the right, and this is where we assemble metatranscriptomes. This is sequencing the transcripts. Post-host depletion, post-16S or ribosomal RNA depletion, and we're able to recover both DNA and RNA viruses using these approaches. A lot of this, we're in the very analysis stage of reassembling these for each of those tissue types that we were talking about, but one that we actually divvied out and we're starting to prepare a clinical observation paper, is that in numerous different patients who were undergoing CCI surgery, we have recovered entire Bavundomonas genomes from the C1 ligament in these patients. And it appears that each one is individual to the patients, that's personalized. Bavundomonas is a known common constituent of your intestinal microbiome. So a working theory right now is that these patients have essentially undergone microbiome translocation from a microbe-enriched environment, that is the intestinal environment, to what's thought to be a posibacterial environment, in this case, the ligature and the nervous system. But this is a very, very preliminary result. Watch this space. We're going to be pushing out clinical observation papers based on some of these results going forward. I mentioned previously that some of these tissue samples, if you were just to go in and sequence without any sort of depletion of host DNA, you will end up just sequencing the host. And so we've partnered with industrial partners to test, develop, and deploy methods for using large-scale CRISPR-Cas9 degradation of host DNA and RNA. These are guide RNA libraries of over 1 million guide RNAs in order to enrich for everything that is not the host. And some of these are ongoing, getting optimized for the different tissue types. One fun thing in this space is that we're also developing a panel specifically for blood profiling in acute Lyme disease as well. So some new things from the single cell pipeline. We were really interested. You've heard quite a bit about David Price's samples in terms of these lung studies. And we were very interested in coupling lung microbiome profiling with single nuclei RNA profiling in the lungs. And in an unfortunate setback, the commercially produced instrument that's supposed to be able to extract nuclei or cells from any tissue type failed miserably in this case. Fortunately, one of the developers of single-cell genomics and single-cell transcriptomics from the laboratory side, Mark Novotny, has been working at JCVI since 2007. And so he's developed a homebrew method that we've now deployed for a balance. These are 16 samples, and we have another 6. So we now have 22 samples balanced case control, where we've been able to recover substantial numbers of nuclei relative for each of these samples, despite the samples being incredibly low biomass. Transbronchial biopsies are very, very tiny. If you were to go to some of the lung single cell studies from the past, you will see that many of those samples are actually, mouse head sized chunks of lung. Whereas here we're looking at something that you might break off from an old lead pencil. On a side note, I have mentioned single nuclei versus single cell RNA-seq, and I wanted to return to that. If you were to just hit cells with a disaggregating enzyme mix, many of these cells, because they're interdigitated, will not actually separate. And it's very easy for those cells to end up getting double counted, and those end up being data chucked in the past. So Mark, again, he's an absolute wizard. He will essentially slice the outer cells, float the nuclei out, and then using high specificity FACS, sort individual nuclei out of these samples. And these plots are very useful. This is a multiple gating mechanism method. And you can see that huge amounts of debris would come out of these samples if we weren't doing exactly this type of FACS sorting prior to feeding them into the traditional 10x platform. In another case, we've actually, we're just finishing up the final sequencing, and it'll push us 32 samples for endometriosis. We've completed a cell catalog where we've sorted endometrious single nuclei according to their transcriptional profiles. That's over on the left, and identified marker genes, which will allow us to go in and look at the distribution of these cell types later on and see if they are different in endometriosis versus non-endometriosis samples. And as promised, that's 2 seconds under 10 minutes. I want to thank everybody on this team, as well as the funders. Thank you.
Nice. Good job, Chris. That was a great overview and perfectly timed. Our next speaker is Ben Reedhead, Associate Professor at the Arizona State University Neurodegenerative Disease Research Center, and he'll be talking about validation of a cytomegalovirus-based biomarker for Alzheimer's disease clinical trials.
Terrific. Thanks so much. Thanks so much, Amy, for the kind invitation, and thanks everyone for their time and attention. Yeah, as Amy mentioned, I'm excited to share this work on validation of a cytomegalovirus-based biomarker for Alzheimer's disease clinical trials. And the more technical jargon-filled scientific story that underpins this is called Alzheimer's-associated CD83 positive microglia linked with increased immunoglobulin G4 and human cytomegalovirus in the gut, vagal nerve, and brain. But my favorite version of the talk title is There and Back Again, A Herpes Tale. And so for anyone that's familiar with with the story of the protagonist wandering around Middle Earth, starting at one end and going down to the other end and then ending up back where they started from, it was kind of like that for us with this story where, I'm an Alzheimer's disease neurodegeneration guy, and we were doing a single nucleus RNA-seq study of the Alzheimer's affected brain. And then in trying to understand the context of this particular immune cell type of microglia, that kind of led us down to looking at the transverse colon, and then looking in the cerebrospinal fluid, and then looking in the vagal nerve, and then back eventually to the brain. So there was kind of a meandering quality to the study I want to share with you today, which it's a complex study to share in 10 minutes. So I'm going to have to skip through some detail, but hopefully I can give you a sense of what the story is. And it's a story that we've kind of built up in two vignettes that are described in a couple of papers, one published a couple of months ago and another one coming out in a couple of weeks. And the first piece in this story is what is an Alzheimer's-associated CD83 positive microglia that we see as being linked with a curious increase in immunoglobulin IgG4 production in the transverse colon, which, this is an unusual set of things to connect together, but let me show you how it came about. And so for any fuller detail, I'd like to refer you to our paper, which came out in Nature Communications a couple of months back, and it really represents a lot of work from ourselves at ASU and many of our partner institutions who are invaluable in doing this. And it's kind of a big data network modeling type approach applied to single nucleus RNA-seq from a set of 101 Alzheimer's disease affected and aged control subjects. They had really rich characterizations, and we did single nucleus RNA-seq, and we ended up paying attention to a particular microglial subtype that we identified as being overrepresented amongst Alzheimer's disease patients. So about 25 to 45% of Alzheimer's disease patients had some of these microglia that we're referring to here as CD83 positive microglia. Just as a bit of a shorthand for these microglia. And as we were getting to know them, trying to understand them, as is our bias, we did a lot of network modeling type approaches. And I'll skip through it mostly. I've got to skip through the detail here, but essentially they look to be a senescent-like microglial subtype. We were trying to understand, does it tell us anything interesting about these patients? Are they different in some informative way? We got a bit of a clue when we were looking at their neuropathology, and we saw that Alzheimer's patients with CD83 microglia, they had more neuropathology, they had more neurofibrillary tangles, and they had more neuritic plaques. They didn't have more diffuse plaques, but beyond that, we couldn't really see a difference. They weren't different in terms of their age, sex, ApoE4 carriage. They weren't different in terms of their disease comorbidities. We looked at many different covariates, trying to understand if it is clinically or neuropathologically informative. Where we got a little bit of a breakthrough was when we were thinking about CD83, in its broader context within the immune system. This is a marker that often will characterize an activated antigen presenting cell. So we started to wonder whether or not maybe it's representing some kind of immunological or microbial perturbation. And on somewhat of a hunch, we really got our breakthrough when we generated some proteomics from transverse colon tissue samples from these same set of subjects. So, the transverse colon being the site of the highest microbial diversity in the body, And then we compared CD83 positive Alzheimer's patients with CD83 negative. And what this really pointed us to was this very interesting protein, IgHG4, which forms the heavy chain of the IgG4 antibody type. And so for anyone that's not familiar, this is the smallest component of the IgG pool. In healthy subjects, it's maybe 5% of your total. And it's functionally an interesting antibody because it's considered an anti-inflammatory, in terms of its kind of net effect. It's somewhat anti-inflammatory. That is, it'll bind to an antigen, but then it doesn't initiate phagocytosis or complement activation. And so that's kind of where this story was left in that Nature Communications study that I referenced earlier. And what's coming up next is where I think things are getting really interesting, where we picked up from that point and then said, like, what is it that's driving this IgG4 in the transverse colon? And what we think is happening is that there's some kind of chronic CMV or cytomegalovirus infection in the gut, which is acting as kind of a risk factor for eventual passage into the brain through the vagal nerve and perhaps other paths as well. And so this will be described in a forthcoming paper in Alzheimer's and Dementia. And we did a bunch of immunohistochemistry on tissue samples from these same subjects that we had the single nucleus RNA-seq for. And what we saw was actually like really striking IgG4 presence in the gut of Alzheimer's patients with CD83 positive microglia in their brain. So the IgG4 really was there. And then that got us to the point trying to understand, like, what does that IgG4 represent? Are they specific? The hope is with antibodies that they're raised to see some specific antigen or group of antigens. Or it could alternatively be some kind of non-specific increase. We weren't sure what, but we applied this really cool technology called serum epitope repertoire analysis, which is where you take a biosample. In this case, we were looking at cerebrospinal fluid, and you pull down the antibody repertoire, and it gives you insight into what sorts of antigens that antibody repertoire is capable of responding to. And what we saw when we compared Alzheimer's patients with CD83 positive microglia to Alzheimer's patients with CD83 negative microglia, and focusing in on the IgG4 specific component, what we saw was a really clear signal pointing to cytomegalovirus, which was really interesting to us. And we wondered maybe CMV, might be infecting the transverse colon, which is where the IgG4, was first observed. And so we went back to these tissue sections and we stained for several different CMV antigens, and we saw actually very, very immunoreactive transverse colon tissue samples only really amongst the CD83 positive Alzheimer's subjects. So this is a very common virus. 80% of us will have seropositivity generally, but what we observed was that most people don't have it detectable in their gut, at least not using this assay. But almost everyone with CD83 microglia had it detectable in their gut. And so we wondered, could it be that the the IgG4 or the CMV might actually be getting up into the brain and activating these microglia in this particular way? So we looked in superior frontal gyrus tissue samples from the same subjects. Remember, superior frontal gyrus is where we did single nucleus RNA-seq at the beginning that put us on the path of CD83 microglia. So kind of coming full circle back to that same brain region. And what we saw was actually very consistent immunoreactivity against late and early antigens for CMV in the brain of these CD83 positive subjects. And I've shown representative micrographs here, but we actually looked at a lot of different tissue samples, so we could work out the statistics of these associations. And essentially what we see is like very tight coupling between CD83 presence in the brain and both CMV and IgG4 presence in the brain, almost at a kind of a one-to-one coupling. And so we wondered whether or not, we wondered about the vagus nerve, which of course is kind of the main source of innervation connecting the gut to the brain. We all wondered whether or not that could potentially be a route of passage for some perhaps chronic gut infection, making its way up into the brain. And so we were able to get hold of vagal nerve tissue samples from these same subjects. And what we saw actually was, again, very consistent immunoreactivity against IgG4 and cytomegalovirus in the vagal nerve of subjects with these CD83 positive microglia, but we didn't see it in CD83 negative microglia Alzheimer's patients. So again, it was kind of partitioning quite neatly. I have to skip forward, but we were pleased to be able to replicate these findings in an additional independent cohort run out of Rush Alzheimer's Disease Center in Chicago, and using additional viral detection methods, using like an RNA-based in situ approach. One last piece I want to leave you with is terrific brain organoid infection studies that were run out of UMass Amherst by Rigel Chan and Elaine Lim, where essentially we're able to show when CMV gets into these brain cells, the more virus gets in, the higher the amounts of A-beta-42 are produced and the higher phosphorylated tau. So this is giving us paths to understand the ways in which this virus might be able to cause havoc in once it makes it into the brain. So this brings us to what we think is a really interesting potential translational direction, because we're seeing the presence of both the immediate early and the late antigen for CMV. That implies really an active replicative CMV infection, and so we wonder if it might actually be responsive to some already available antivirals. And so we're thinking about trying to gear up for some kind of clinical trial of antivirals in CD83 positive Alzheimer's subjects. But as a stepping stone to get there, we need a good biomarker. We need to be able to identify this in patients, that come into the clinic. So can we find a blood-based biomarker that can tag CD83 positive microglia and CMV in the brain in a way that disentangles it from more general exposure to CMV? So almost all of us will have been exposed to CMV and have IgG antibodies against CMV, but we're actually trying to hone in on a much more particular section of the population that gets exposed to CMV, and we need to find a biomarker for that. And so one of the really promising directions we think is using this epitope repertoire analysis, where in some initial data that we've generated, we've identified some CMV epitopes which actually turn out to be quite predictive for being able to classify subjects as CD83 positive or not. Giving particularly a set of 3 CMV epitopes, which gives us quite good classification accuracy. But really our big priority now is to try and validate this potential biomarker in an additional independent cohort of 200 serum samples. And this is where PolyBio has really stepped up. This epitope repertoire analysis is expensive, so it's really only through PolyBio's support that we're able to push forward in validating this biomarker, which would be a necessary step to any kind of clinical trial where which might be looking at antivirals or other kind of microbially themed or microbially targeted interventions. And we think there are some great options there that I need to skip through just in the interest of time. But just to leave you with the conclusions, we think that CMV infection in the gut appears to be a precondition for eventual passage to the brain with apparent impacts on AD pathobiology. Really what's been critical for our success here has been access to matched samples across the body, so that we can really tease out these peripheral impacts on a brain disease. And we were really excited about all sorts of translational opportunities in the therapy and also the biomarker space. So with that, it's really a privilege to be able to present on behalf of terrific friends and collaborators, and very grateful to PolyBio for their support of this work. So thank you.
Thank you, Ben. That's an amazing overview. And if there's anything we can do to build towards these trials, sometimes of affordable existing herpes virus antivirals, for example, that's just an incredibly huge priority in Alzheimer's disease. So really exciting. And with that, let me introduce our next speaker. It's Matt Frank. He's a senior scientist in the Department of Integrative Physiology at the University of Colorado Boulder, and he'll be talking about a multiple hit model of long COVID.
Hey there, Amy. How are you?
I'm good. Amen.
Oh, good. Good to see you. Thank you for the invitation. So today I'll be talking about a multiple hit model of long COVID. So there's evidence that there's neuroinflammation in long COVID. And of course, if you have neuroinflammation, you can have neuroinflammatory mediators. These, of course, can draw many drive many of the symptoms in long COVID, such as fatigue and brain fog and what have you. So our interest has been in understanding the neuroinflammatory properties of SARS-CoV-2 antigens, in particular the S1 subunit. And so it's been shown it's shed from SARS-CoV-2. It can enter the circulation and persist for months or longer. It can cross the blood-brain barrier and enter the brain parenchyma, and that is key. They directly produce neuroinflammatory cytokines such as IL-1, TNF, and interferon gamma in immune cells such as microglia. And then finally, many of these cytokines outside long COVID have been shown to be sufficient to induce many of the symptoms seen in long COVID, in particular fatigue, effects on memory and cognition. And so we've been interested in developing a two-hit model of long COVID. And so let's just work through the priming dynamics here. And so the first hit involves, for us, looking at the spike S1 subunit. And what you end up getting is this initial neuroinflammatory response, which then resolves, but this leads to these protracted neuroimmune alterations, which is considered priming. These neuroimmune alterations could be persistent changes in receptor expression or signaling pathways. But essentially, these changes can persist for quite a long time, such that immune cells in the brain can then become sensitized to subsequent inflammatory challenges, which we term a second hit. This could be viral, bacterial infection, psychological stress, any kind of physical stressor. And thus, if you're then exposed to a second hit, this will produce a potentiated neuroinflammatory process or response, and of course, the physiological and behavioral sequelae of that response. And so our experimental approach is essentially to inject S1 into the central nervous system. This is considered the first hit. We give it into the central nervous system because we want to obviate any peripheral effects. And also, since we already know that S1 crosses into the brain parenchyma, we simply give it into the CNS. We then wait 7 days. This allows priming to occur. That is, we see changes in signaling pathways in microglia that occur over the 7-day period. And also at this point at 7 days, the neuroinflammatory response to S1 has resolved. And then at 7 days, we then have a second hit. That is, we inject a bacterial mimetic into the peritoneal cavity. We then wait 4 or 2 days, and we looked at an array of outcomes, such as neuroinflammation, sickness behavior, physiological responses, and microglial activation. And so I just want to summarize first some of the data we found published in Brain Behavior Immunity over the summer. And essentially what we found is that prior S1 potentiates the effects of bacterial mimetic. And that involved increased neuroinflammation, such as IL-1 beta, sickness behavior. We saw decreased activity, drinking and self-grooming. We saw increased physiological responses to the bacterial mimetic. That is increased fever and heart rate, as well as increased microglial activation, that is increased expression of IL-1 beta expression. And so what I want to focus on today is what is the mechanism that may be driving this priming phenomena that we see with S1? And so I want to focus on glucocorticoids. And so this paper that came out by Klein in 2023 showed that serum cortisol levels are remarkably reduced in the serum of long COVID patients. Simply, we see we see healthy controls, convalescent controls, and that the two cohorts of long COVID patients have reduced cortisol levels. And so this really grabbed our attention because, for many of you probably already know, is that one of the main features of glucocorticoids is that they can be immunosuppressant or anti-inflammatory, although that's not exclusive. And so we basically started thinking, okay, maybe glucocorticoids, if they're lower in long COVID patients, you may get a disinhibition of neuroimmune processes. And so glucocorticoids, they largely serve as a brake on the immune system, that they're anti-inflammatory, although they can be modulatory in other ways. And so when glucocorticoids bind the GR alpha receptor, you can get induction of anti-inflammatory genes as well as reduction of inflammatory genes. And so, in light of these properties of glucocorticoids, we then thought, well, maybe a priming mechanism could occur through reduction of glucocorticoids in the brain by S1. And so we conducted a study where we injected S1, again, into the central nervous system. And so 7 days later, we looked at glucocorticoid levels in various brain regions, such as the hypothalamus, frontal cortex, and hippocampus. And we found that indeed, glucocorticoid levels were profoundly reduced in all 3 brain regions. It's important to note that S1 is not detectable at any time point after 24 hours post-injection. And so also we found that serum S1 levels were also reduced by S1 given into the central nervous system. These data suggest that S1 may be dysregulating the hypothalamic pituitary adrenal axis. And so the idea being that, maybe we're getting alterations in ACTH or CRH. Ultimately, this may be leading to reduced production and release of corticosteroids from the adrenal cortex, which then would lead to less glucocorticoids in the central nervous system. And of course, glucocorticoids themselves have profound effects throughout the body, pretty much any organ system and cell type. The glucocorticoid receptor is ubiquitous. And so, we see changes. In particular, I think it's interesting that you see changes in glucose homeostasis, and of course, in the nervous system as far as cognition. And so the possibility could be that in long COVID, if you have a hypocortisolism in some patients, it could be that low glucocorticoids could be driving some of the symptoms of long COVID, in addition to being a mechanism of priming. And so the general idea here is that S1 at some point shed leads to lower brain glucocorticoids. These might then disinhibit brain immune cells such as microglia, thereby priming these cells to subsequent inflammatory insults. And of course, these subsequent insults could be viral, bacterial, it could be stress. Essentially, the idea is that by disinhibiting these cells, you're essentially sensitizing brain immune cells or making the brain vulnerable to subsequent inflammatory insults. And thus, if an individual is then exposed to, let's say, a subsequent viral infection after having been primed by S1 exposure, you can then, of course, get increased neuroinflammatory processes. This then can lead to fatigue, cognitive problems. And so this is an ongoing project for us, and we're currently exploring how these reduced brain glucocorticoids may be affecting glucose homeostasis in the brain. And so on that note, I'd like to acknowledge Tel Kelly, Robert Thompson, Shelby Hopkins, and Angelina Kuzma in Monica Fleschner's lab in integrative physiology, and then, of course, Jason Ball and Linda Watkins' lab in the Department of Neuroscience, and the generous support of PolyBio. Thank you.
Thank you, Matt. It was really good work focused on mechanism, which is great.
Thanks.
Cool. All right, our next speaker is Max Quian from the J. Craig Venner Institute, and he'll be talking about long COVID endotype identification.
Thank you, Amy. So let me share my screen. Can you see my screen?
Yes.
Great. Okay, so my topic will be focusing on how can we use machine learning to identify the patient groups from the long COVID cohorts. And this is a teamwork. I'm grateful for PolyBio support. And also our data sets come from UCSF as well as Mount Sinai. And Beverly Prone at JCVI did most of the programming and data analysis. I really appreciate everyone. And so I think everyone in this room understands the complexity and importance of the long COVID symptoms. So we are not the first group who are trying to divide the patients into multiple groups and subgroups so that we can study the mechanism better. And however, when I review the previous work, so they actually focus on EHR data, which is great. However, you require too many data sets to be able to do this grouping. And we are trying to figure out whether we can actually do this based on the questionnaires. So I think from that perspective, we are actually the first group who are doing this. And this is actually pretty challenging because it's a catch-22. So before knowing the feature sets, we don't know how to define the patient groups. And if we don't know the patient groups, how can we extract the patient group specific features? So we try to use those like very simple approaches such as a PCR analysis and try to group them. And we found it doesn't work. So we have to use some fancy approach called the topic modeling. The topic modeling means actually, so we are trying to do this nonlinear transformation of the patient matrix feature table. And then we will try to identify clusters from these advanced transformation results. In that method was previously invented to process those text data from like identify those topic groups from those like news articles to decide whether this article is about entertainment, sports, or politics. However, recently people further improved to use this kind of deep Poisson factor modeling to make it really working well to analyze those like questionnaires data as well as the text data. So there are major 4 steps in our approach. First, we have to determine the best number of topics, which are the important transform features to describe the patient groups. And then second, we're going to cluster these new metrics to output the patient class. And then we further interpret the results by trying to map the cluster to those preconditions and all other variables to see whether we are identifying those groups correctly or not. And so I think previous people already show how this can be done at a high level. So when you have a patient symptom matrix, you convert into a topic space. And then based on the topics, you construct a new matrix called a patient topic matrix, as you show here. And then now the data becomes quantitative. So you can actually use those quantitative values to do your data cluster analysis to identify those subgroups and subphenotypes. However, so our approach is actually really trying to improve this analysis to fit our data sets. And we got data from like both UCSF and Sinai. So, but last time I already presented the UCSF. So this time I'm going to focus on Sinai data sets. And so with their help, we actually were able to convert those into 2 like metrics. One is focusing on preconditions and the other focus on the symptoms. So we have about 615 patients from the Sinai regarding these symptoms and conditions. Okay, and regarding how to identify the best number of features. So here's the coherence and likelihood. Coherence help us to make each cluster really cohesive. So the more topics we use, so the less cohesive that cluster will be. And but for likelihood, so the more topics we use, so we will have a better way to separate those patients. So we have to find a balance between those 2 criteria. So the way we did is we try to combine them together. Then we can identify a good range of those top topics to describe, so between 18 to 27. So which is a really good range to describe our current data set. And based on these results, we can see, so we have those topics matching to the symptoms. And when we evaluate these things, so we can notice in here, so for example, we can see those very specific symptoms such as nausea, vomiting, and digestion, they actually will group together. And also there are some other groups who also can be matched very well to the predefined categories, neurological, cognitive, and also those muscular and GI. All those groups work pretty well. And based on those topics, we further cluster those patients. And we already tried to apply different methods to see which method could actually give us the better results. And based on those benchmarking results, we picked like 3 methods to apply to our cohort. And so we finally identified 16 patient clusters from this 600 patient cohort. And we ranked the different K values using the standardized scores, and then we can actually see these results look really well based on the, not only based on the scores, but also based on the UMAP visualization. Those 2 results were actually produced separately and independently, but they actually match very well when we visualize the clusters identified by topic scores and clusters you could see on the UMAP space. So we kind of convinced ourselves, okay, this is pretty good clustering results. And then we also can visualize each individual cluster together with their symptom signatures. So I'll show you the 15 clusters here. So the female is colored in red and male cluster in blue. So you can see most of the clusters consist more female than male. And it also, their individual patient pattern look very consistent within each cluster. This is a good sign because, as I mentioned, so if you don't know the good feature set, you don't know how to identify the cluster. But however, if you already identify a good set of symptoms to describe your subgroup, then you will see this kind of very consistent patterns. So this is beautiful. And okay, of course, we can interpret this using heat map too. So first thing I can see here, okay, there's definitely a severe group here. This group of patients reported almost every symptom. And however, there's also another group who did not report many symptoms, which is here. And when I look at those like top 6 symptoms per group, so I realized, okay, they are pretty distinct between the classes. However, there's still some overlapping. So I'm thinking, so maybe we can further merge those classes into endotypes. And this allows us to actually do another round of hierarchical grouping, which finally identify A endotypes in the group as I show here. So the first one is here, second one is this region, and the third one is the severe region. And that's it. And now it's time to see what those individual endotypes are. So I would, because we don't have enough time, I'll just go through a few of them to show you those interesting patterns. So endotype number 2, this is a typical cluster focus on like neurological and cardiovascular problems. So you can see the patterns are very consistent. And actually, they actually reported asthma and their preconditions. So let me remind you, so we actually did not include the precondition in our analysis. So this is an independent assessment. So when we see the precondition can actually match to a cluster, that actually tells us this cluster was actually pretty well identified. And the second one is showing, so another endotype, this group focus on like, has more sensory hormone and GI problems. And when we look at their preconditions, they actually report those different like bowel symptoms and also the autoimmune diseases. And they also consist most female than male. So this is very typical, like the thing we saw in our results. For endotype 4, so this is kind of eating disorder group, interestingly. So they have those kind of signatures. And for this one, it's actually cognitive and neurological problems. So we can see they are all consistent with this cluster. However, we can see the big difference between this group and the baseline, which is dotted line here. And finally, this one, we can actually see, okay, neurological and musculoskeletal, and they actually have depressions more than the, okay, in summary, so we actually completed this method development very well, and we have a very good pipeline to be used. If you have input data, we actually can do this for you. And we identify 16 clusters and 8 endotypes within this finite data group. And most of them actually consider more female than male. And we are trying to interpret the results by matching this to the pre-existing protein categories. And we also mapping to the preconditions. We showed the very good mapping to interpret those results. And we visualize to track the consistency, and we correlate the patterns with other variables to show the correctness of the results. And next step, so I will need to validate this using assay data. So I'm trying to collaborate with Emory Group to use their meta results to do this. And also we need to focus on those very useful patterns to compare case versus control, and also different like severity groups between male and female to show the patterns. And yeah, that's all. Thank you so much.
Great, Max. These endotypes will really help clarify just patterns for clinical care and maybe even recruitment into studies and clinical trials. So it's really helpful. Our next speaker is Nadia Rowan. She's a professor of urology at UCSF and a senior investigator at the Gladstone Institutes. She'll be presenting potential SARS-CoV-2 persistence and T-cell activity in the female reproductive tract.
All right, so thank you, Amy, for this opportunity to present. Today I will be sharing a study that PolyBio recently funded to study SARS-CoV-2 persistence and T-cell activity within the female reproductive tract. Okay, so over the past couple of years, we have spent significant efforts really trying to compare immune responses between individuals with versus without long COVID. And this was really enabled by our closely working together with a cohort called LYNPH, which you heard about earlier today. It stands for Long-Term Impact of Infection with Novel Coronavirus. It's based at UCSF, where my lab also is, and it's headed by Michael Peluso, Timothy Henrik, and Steve Deeks. And this cohort was really established at the very beginning of the pandemic, and to date has recruited many individuals with and without long COVID. And so just as an example, we published a study earlier this year where we deeply compared immune responses between individuals with versus without long COVID using a variety of omics approaches. And the overall punchline is that we found many immunological perturbations in the people with long COVID, particularly within the T-cell compartment. But one limitation to this study is that we exclusively looked at blood specimens, so PBMCs and plasma and sera, whereas as many of you know, and as we've heard much about today, the mechanisms driving long COVID are really thought to originate within tissues. And one of the main leading hypotheses as to what may be an important driver of long COVID from tissues is that there may be a persistent reservoir of SARS-CoV-2 infected cells. And many of you are familiar with this paper that was published by Michael Peluso and Timothy Henrich. Sorry, something just popped up. By Michael Peluso and Timothy Henrich earlier this year, where they reported that they could detect SARS-CoV-2 RNA within the guts of people with long COVID. This was years after initial infection, suggesting that there is potentially a reservoir within the gut. Now, the gut is a somewhat unique tissue. It's an immunotolerant tissue. It needs to tolerate food antigens, microbiome, but there's another very important immunotolerant tissue as well, and that is the female reproductive tract. It needs to tolerate different other types of antigens, such as sperm antigens and allogeneic fetus. It also has its own microbiome. But we were really interested in studying the female reproductive tract because it, like the gut, is a site of immune tolerance. There's another important reason why we were really interested in studying the female reproductive tract, and that is because of the sex differences in long COVID. And so you already heard a little bit about this earlier today from Peter Broden, but it's well recognized that women, particularly premenopausal women, are more likely than men to experience long COVID. And just as an example, within the UCSF Link cohort, 50% of our participants are female, but 78% of those with severe long COVID are women. And there are a number of things that could drive these differences. One is that it's well known that there are sex differences in immune responses. For example, women mount stronger innate and adaptive immune responses to both infection and vaccines, but at the same time are generally more prone to autoimmunity. And there are a number of potential drivers of these sex differences in immunity. And the two I'll kind of point out are their chromosomal differences. So individuals that are assigned female at birth typically have 2 X chromosomes. And there's something called X chromosome inactivation that will inactivate one of the X chromosomes, but it's often incomplete, meaning that you end up sometimes expressing higher levels of these X chromosome encoded genes. And one of these are TLR7, which is involved in type 1 interferon signaling. There are also hormonal effects as well, in particular sex steroids, including estrogen, progesterone, testosterone. And just calling out testosterone, there's data that testosterone itself can decrease type 1 interferon responses, including to TLR7. In a nice preprint study published recently by Akiko Iwasaki's group, testosterone was associated with protection against long COVID, specifically in cis women. And so for the PolyBio funded study, what we are doing is we are focusing on women and we are focusing on the female reproductive tract. And the overall design of the study is relatively straightforward. So we recruit 5 women with long COVID, and these are specifically pre-menopausal women because we wanted to focus on this group because they are at higher risk for long COVID. And we also compare them to 5 matched premenopausal control individuals. And from these individuals, we are obtaining biopsies from both the female reproductive tract and the gut. And to do this, we are leveraging an infrastructure that we had set up for our HIV studies. So this HIV study is called TRUST. It was started a while ago, but essentially we've recruited ART-suppressed women with HIV to come in to donate within a single study visit 3 biopsies from the gut, so ileum, right colon, and rectosigmoid, as well as 3 biopsies from the female reproductive tract, so endometrium, endocervix, and ectocervix. And we've done a number of different things with these types of specimens. For example, back in 2022, we published a study where we did in-depth immunophenotyping by CyTOF on these different specimens, and we can find differences in the phenotypes of immune cells, including T cells, from these different sites. And more recently, in some unpublished data, we are leveraging single-cell sequencing platforms to really deeply compare the immunological differences between immune cells from these different sites. And so essentially what we are doing now for the PolyBio funded study is to leverage that infrastructure and to get these multiple biopsies from these different sites together with paired blood. And we're performing 3 major types of assays with these specimens. And I'll touch upon each of these in turn. And so the first of these is to dig deeply into T cell responses against both SARS-CoV-2 as well as other viruses implicated in driving long COVID, in particular EBV and CMV. And just a word about the main assay that we're using. So we're leveraging one of the main assays that we use in this published study, which is a CyTOF-based assay for assessing viral-specific T cell responses. And it was using this assay that we found major disturbances in the T cell compartment in individuals with long COVID. And so the assay is essentially a CyTOF-based assay where we take a control samples where we don't add peptides. We have a very clean background, as you can see here. Whereas when you add overlapping peptides spanning SARS-CoV-2 proteins, you can see only in someone that had recovered from SARS-CoV-2 infection do you see a distinct population of cells inducing a specific cytokine. In this case, I'm showing interferon gamma, but we have many other cytokines and cytolytic effectors within our panel as well. So I show this because this is typically what we see. This is what we've seen in the context of mild COVID, severe COVID, people with long COVID. Typically, maximally, we will get less than 0.1% of the cells that are responding. And I show this because in some preliminary analyses, we've made a very striking finding that when we looked at the endometrium of people that have recovered from COVID, that we saw a much more robust T cell response. So in this particular endometrial sample, about 4% of all the CD8 positive T cells from the endometrium were really specifically responding to spike. Now, this is not in the context of long COVID. This was for a different study, but it really highlights that the endometrium might be a unique tissue in that it harbors a high frequencies of these viral specific T cells. And perhaps even more strikingly in this particular specimen, this was from a woman that had recovered from mild SARS-CoV-2 5 months previously. So it was not active infection. It was already during the convalescent phase, and yet you still have these really high frequencies of SARS-CoV-2 specific T cells. And so we're really interested in trying to understand what might be unique about the endometrium and might this have some implications for long COVID. So that's the first study to deeply look at T cell responses against SARS-CoV-2, EBV, and CMV. But we're also implementing 2 other assays. The first of these is single-cell RNA-seq analyses, including for assessing for the presence of persistent virus. And this leverages some of our HIV studies where we've been developing single-cell sequencing-based platforms to analyze PFA-fixed cells by single-cell RNA-seq under conditions where we can actually find virally infected cells. And so we've developed this for HIV by designing various probes against relatively conserved regions of the HIV provirus highlighted here. And this is just validating the system, showing that when you take uninfected cells, you have very minimal background. But here we've taken HIV infected cells, and we can see a distinct population of cells that harbor HIV RNA. And the nice thing about this platform is we can look not only at viral RNA, but we can also look at viral protein. And in the case of HIV, the protein is something called P24. And so here on the right-hand side, we're looking at the HIV RNA positive cells specifically, and we're looking for those that have protein, have HIV protein versus those that do not. And the ones that have protein are shown within this box here. The different colors actually correspond to the different probes that were bound, but we have ones that are protein positive as well as ones that are simply RNA positive, but not protein positive. And so we want to apply the same kind of platform to look for SARS-CoV-2 RNA positive cells and to look amongst those cells whether they are SARS-CoV-2 protein positive or not. And finally, the last assay that we will be performing is something called MIBI. It's spatial profiling at the protein level. It's essentially CyTOF in situ. So it's also borrowing from our HIV studies because we have been developing MIBI platforms to look deeply at persistent HIV within tissue compartments, including the gut. And essentially, we can look at about 40 parameters simultaneously. We can quantitate the numbers of 40 different types of proteins. This is just an example in the context of HIV, where we can find reservoir cells that are infected with HIV based on HIV protein expression. Here's one, here's one. And this is actually all in the context of ART suppression. So this is an actual reservoir of virally infected cells. We can look at what's unique about those reservoir cells versus their uninfected counterparts. That's a bit here on the right. And most importantly, we can also look within the microenvironment of those actual reservoir cells to see whether there's something unique about that microenvironment that allows these persistently infected cells to persist. Because you would imagine the immune system, in theory, should eliminate it, but for some reason it does not. So we're really interested in understanding why that's the case, now in the context of long COVID for this study. And so this study actually just began, so we haven't collected specimens yet, but we have begun recruiting, and we hope to recruit the first participant in December, and hopefully at the next meeting, we'll have some interesting data to report. I just want to thank the folks that are involved. So from my group, these three folks, Marta, Mackenzie, and Kaylin, are the ones that are involved in the project. I want to say that this PolyBio funded study is a collaborative effort between myself, Michael Peluso, and Phyllis Tien and our teams. I want to thank the participants who made this study possible, and of course, PolyBio for their generous support. And that's
awesome. Thank you, Nadia. It's so great that you're collecting the endometrial samples and doing such intensive forms of really advanced analysis. Cool. Well, our next speaker is Alessio Fasano. He's the W. Allen Walker Chair in Pediatric Gastroenterology and Nutrition and the Director of the Mucosal Immunology and Biology Research Center at Harvard Medical School. He'll be talking about liraglutide in the treatment of SARS-CoV-2 spike protein translocation in leaky gut.
Thank you, Amy. And I have a lot to cover in 10 minutes. I will do my best. And I will do this on behalf also Lil Jonker and our team at the Mucosal Immunology and Biology Research Center. Some background just already mentioned before that, like in adults, the SARS-CoV-2 clearance, it's something that it's changing our understanding, in terms of the length of kids that, have, are infectable. I mean, it's relatively measured in days, but when you look at the possible reservoirs, including the GI tract, that this has been mentioned before, both in adults and kids, this GI tract can be a reservoir for months, if not years. So, and this has been proved and confirmed in many, many studies. What is also interesting, and we published that in conjunction of the fact that SARS-CoV-2 harbors its biological niche in there are evidence that viral particles, including spike and S1 and the nucleocapsid proteins, may be found in circulation, and they leak from the gut into the systemic circulation, given that the variance is not detectable, systemically, yet, the evidence of the presence of the SARS-CoV-2 in the GI tract and the presence of, viral particles in the circulation makes the possibility that is indeed the source of these particles, the GI tract. There is also one more evidence that point in that direction, that kids with MIS-C, that is this multi-organ inflammatory syndrome in children, they have immunoglobulins reflecting ongoing antigen exposure that are above of all the class, IgM, IgG, and IgA. I mean, this is not a piece of the puzzle, if you wish, that suggested indeed the presence of this virus in the GI tract, when you are genetically predisposed, can lead to passage of these particles systemically, and this will induce this cytokine storm that leads to this complication that now we consider part of the PASC spectrum. There was some precedent suggesting that this could be the case. As you know, at the beginning, MIS-C was compared. Actually, some people believe that it was very similar to Kawasaki syndrome, and that to be not the case, but the mechanisms were very similar. We already published that this likeness of particles coming from the GI tract in the Kawasaki syndrome was due to a molecule that we discovered a while ago, responsible for increased permeability in the GI tract. It's called zonulin. There is a family of molecules, to be more precise. That is a pathway in which the paracellular pathway is regulated to increase antigen trafficking from the gut lumen into the circulation. And sure enough, like we identified and published already for Kawasaki syndrome, also kids with MIS-C, they have increased zonulin levels in their bloodstream. There's also in this spectrum MIS-C long COVID past, so to speak, the evidence that again multiple studies now, as I told you, show this that the reservoir of the SARS-CoV-2 is not peculiar of kids with MIS-C, but PASC in general. I can list for more than a year. There are studies that suggest that every unit increase in zonin is equivalent to a 44% increase of risk to develop PASC. And then there are spike protein detected in the blood in adults with PASC, like as we did in kids with MIS-C. And all this in the context of the fact that this molecule, zonin, been targeted for possible, manipulation of antigen trafficking and therefore leakness of these molecules by using a specific peptide inhibitor that is called larazodide. They've been in clinical trials for other indication. And we had the opportunity in the midst of the COVID pandemic to have compassionate use of this larazodide in kids with MIS-C. And these were, open labeled compassionate use of a handful of kids, just showing that, giving this, molecule will increase the clearance of spike and therefore the time of recovery. With that said, and object of our discussion today, we decided to do this the way that's supposed to be done. We went to the FDA, we got approved for a double-blind placebo control, study design that you see here that, it was, implemented for, a 3 weeks surveillance and then a follow-up, at many months distance to see if indeed any possible beneficial effect in terms of efficacy was going to be sustainable over time. And of course, as always in these studies, safety as well was a concern that we look at. And speaking of which, you see here the patients population that end up to be, allocated to larazodide arm or the placebo arm, and there were not pretty much adverse events to be reported. There were differences between the two groups, and no major, severe adverse events were reported. I want you to note the relatively small sample size, and the reason why is despite they were trying to recruit more kids, we luckily for the pandemic situation, MIS-C became less frequent, and therefore we were forced to finish the study before it reached the intended target of 20 kids as agreed with the FDA. Despite that we were not able to really reach that goal, we surprisingly, I have to say, with a handful of patients that we eventually involved in this study, had the possibility to fulfill the primary outcome that was the time of recovery, the GI symptoms, that was much faster in the larazodide group, as you see here on the top, compared to the placebo group, while the two groups became similar after 2-3 weeks. So bottom line, it seems like that, the administration of larazodide speed up the time of, recovery from GI symptoms. And you see this in different shape and form here that can be quantified in the bars in the here or a heat map that you see, of course, at day one, the darker, the more intense are the symptoms, and the red, again, is the larazodide, the green, gray, is the placebo, and you see, visually how the larazodide group, recover earlier, and then they were all, comparable. There was also something else that was interesting, and it was the survival curve of spike clearance. And you see that, day 7, day 14, and day 21, this clear difference that was statistically different between the larazodide group and the placebo group, with the clearance of the larazodide group of the spike protein to be much faster than in the placebo group, to the point in which in the placebo group there were still 30% of kids that had tested positive for the spike protein, where none of them eventually had the spike in the bloodstream in the larazodide group. And again, this clearance was associated also to a series of inflammatory parameters that, again, I can summarize here because there was a direct correlation between the spike protein and, some pro-inflammatory cytokines that you see down here, interferon gamma and IL-8. And also there was a an inverse correlation between the amount of spike in the bloodstream and the GI symptoms. So the more spike you have, the more GI symptoms, the less spike, the less GI symptoms. So all this to say that, again, there was a correlation between the GI symptom and the inflammation with the spike levels. And because the primary outcome was indeed improvement of the symptoms and, as a secondary, the clearance of spike that we always believed, and now we are confirming in double-blind fashion, to be the driving force for the symptoms and the inflammatory markers, that, eventually this was confirmed. I'm going to finish up also with something else. As I told you, we look also at the long-term, outcome. And this was, quantified as a percent of patients report recovery to usual health. In other words, the typical activity, sport activity, and so on and so forth. And also to not feel tired or to feel, eventually, with any other health issue. And you see there that clearly in the Larazotide group, we have, a much, fast and sustained recovery of the kids compared to the one in the placebo group. So to conclude, what we saw preliminary in the open label approval concept studies that we did when we did this trial during the midst of the pandemic under compassionate use has been confirmed now with a double-blind placebo-controlled trial, and now we're in the midst of doing a similar trial in long COVID with PolyBio support that we of course are extremely grateful, and that's pretty much it.
Great, Alessio. It's really great that that trial met its primary endpoint and that you're also doing it in long COVID. So exciting to see that develop. All right. Our next speaker is Sarah Cherry. She's a professor of pathology and laboratory medicine at the Perelman School of Medicine at the University of Pennsylvania, and she'll be talking about long COVID, defining the viral RNA reservoir in the gastrointestinal tract.
Okay, thank you very much for having me, and I look forward to telling you today about some of our recent work trying to understand whether or not there's a clear reservoir in long COVID patients and which patients they might be, and if so, how do we think about the biology of the infection in the GI tract, and are there specific therapeutic options that we should consider in that context? So just as a reminder, during the acute infection, there's an increase in viral load within the respiratory tract, and either the treatment with antivirals or your own immune system will drop the virus down post-infection and ultimately result in recovery for most individuals. And so to model these early phases of the infection in the respiratory tract, we've been using a system where we can take primary cells out of the respiratory tract from humans and grow them in these air liquid interface cultures, which really phenocopy the respiratory tract of humans. And we can infect these with SARS-CoV-2, and we can characterize that infection paradigm. So if we infect these with SARS-CoV-2 that expresses GFP, we can see that we see a lot of infection by day 3 post-infection and peaks around day 7 and then sort of goes away. And we can also look at this by taking the supernatants off of these cells where the virus is released and quantifying infection using qPCR, where we see the highest levels of infection around day 3 or day 7, and it goes down to below a pretty low level over time. And then if we take these supernatants, if there's live virus remaining, we can only detect live virus up until around day 10. So the peak is very early, and then the virus goes away, kind of like modeling the respiratory tract in humans. And so we all know that there's a subset of individuals who have symptoms post-COVID, and there's estimates of about, up to about 10% of individuals. And so in thinking about what's happening there, many of us have considered that one of the drivers is a reservoir within the GI tract for a lot of reasons that many people today have talked about, including data from biopsies, autopsies, and stool samples. And so we've been trying to think about, can we use some of these more sophisticated models to model that infection? And so we use these 3D human intestinal models that form structures that are similar to the human GI tract. And again, asked if we can model SARS-CoV-2 infection in these models. So again, we're infecting from the apical side and looking at viral infection. So again, we can see SARS-CoV-2 infection of these models, and we actually see low infection on day 3, and it goes up over time. Again, sampling from the apical side of the cultures, we can see actually that the infection goes up over time and seems to be persistent for longer than it was in the respiratory model. And indeed, if we take titers from these different time points, what we've seen is actually the peak virus load is later at 10 days post-infection, and the cultures never clear the infection. We can detect infection for as long as we keep the cultures alive, suggesting that there's a fundamental difference between the bronchial cultures and these intestinal cultures, as the kinetics are different. The bronchial cultures clear the infection, whereas in the intestinal cultures, we see persistence, which may be modeling some aspects of long COVID. So the question is then, are there differences in drug sensitivities of the infections in these two different models? And so again, in the respiratory model, we can infect either with the ancestral strain or one of the more current Omicron strains called BA.1. And then we can treat these cultures on the other side as if you're orally taking these drugs with either Paxlovid or nirmatrelvir or molnupiravir. These are two drugs that have been approved in different contexts. And so what you can see here is by treating with either of these drugs, we can significantly block infection by 10,000-fold with either of these viral strains. And so then what happens if we do the same infection in the GI tract? And so you can see it's strikingly different. While nirmatrelvir or Paxlovid is active against the two strains in the intestinal cultures, molnupiravir has no activity, suggesting that there's fundamental differences in how these cells are dealing with these drugs to block infection. So not only are the kinetics different, but so are the drug sensitivities. And so this led us to also ask questions about whether or not the response to infection is similar or different between these different contexts. So I showed you the kinetics are different, the sensitivities are different. What about the cellular responses to infection? And so to do this, we infected these cultures and collected the RNA from the cells and performed RNA sequencing. And so this is from the respiratory model. We see at 3 days post-infection, about the peak of infection, we see about 50 genes are changed. And then we did the infection in the intestinal model, again, around the peak of infection, around day 6. And you can see a larger number of cells, a larger number of genes are misregulated. So are they similar or are they different? And so this is looking at the induced genes of the lungs by the induced genes in the gut. And what you can see here in the green are the genes that are induced in both model systems. And so this subset of genes are co-regulated. But what about the genes in general? So another way to look at that is to take all the genes that are induced in the lung and look at them here, and then ask, well, how did those behave in the GI model? And you can see, again, like I showed you in that dot plot, most of the genes that are induced in the lung are also induced in the gut. And if you look at them and categorize them by pathways, many of these pathways are associated with antiviral responses and interferon responses, consistent with the literature that's been published about the lung biology. So in some ways, they have similar responses. But what if we look the other way around and say, well, if we look at the genes that are induced in the gut, how are they behaving in the lung? And what you can see here is there's a lot of genes that are induced in the gut that are not induced in the lung, although these small number were the ones that are co-regulated. And if we ask, well, what's happening here? What are the categories or pathways that are being induced? They're quite different. So they include actually now a large number of inflammatory pathways that were not being induced in the respiratory tract, suggesting that there are differences in how the cells are responding to the infection, depending on if it's the respiratory tract or the GI tract. So this then led to the question, well, can we treat with immunomodulators or anti-inflammatories to block the infections? And again, how are they similar or different between these different models? So as we and others have published previously, if you treat with interferon that activates this antiviral pathway, you can see significant reduction of infection in the lungs, and we can also see that in the gut, so consistent with the fact that these pathways can be activated and restrictive. But there's other thoughts about blocking interferon signaling, for example, with JAK inhibitors. So what happens in these models if we then block interferon signaling in the context of infection? And so if we look in the lung, there's a modest, but some increase in infection, although it's not very striking. In contrast, if we look in the gut, there's substantial increases in viral infection if you block JAK signaling, suggesting that either interferon signaling or cytokine signaling is actually restraining infection in this context and potentially not something that one would want to use if there was, in fact, a reservoir in the gut. Then the question about these inflammatory and cytokine pathways that we now observed are induced in the gut. The question is, would blocking these pathways have an effect on viral replication or the inflammatory state that's created by this infection? And so we've treated with a drug called IKK16, which blocks NF-kappa B signaling. And again, we see a modest or little change in infection in the lung. And again, this has a more striking but modest effect in the GI tract on infection. And so again, we should think hard about whether or not we want to block these pathways and whether or not we should look at these other pathways that are induced that may impact infection in different ways. And so we're currently testing these other pathways to see if they can impact infection. So what we're trying to characterize is how SARS-CoV-2, if there was a reservoir, would be altering the GI tract to define the changes in cellular activity, whether or not there's inflammation and the consequences of the cytokines induced, and are these changes causing systemic responses? We found a number of cytokines that seem to be induced and secreted by these tissues, and are these now feeding back on the immune system, leading to the inflammatory state that we can observe under some conditions, and these are ongoing studies. And again, we're trying to figure out which antivirals are active in the gut, which modalities, how long should you treat, what combination, and should we be thinking about anti-inflammatories as combination therapeutics in this context? And then, of course, as we define other reservoirs, we can use these same approaches to understand the biology in those reservoirs and whether or not they're similar or different to what we've observed in the GI tract and the respiratory tract. Another project that we're working on that I don't have to talk about is trying to understand and model whether or not there is a reservoir using mouse models, non-human primates, and looking in long COVID stool to trying to understand if there's selection or drift in the types of viruses that may be maintained in the reservoir, and if we can understand aspects of that biology that may inform therapeutic treatment options. With that, I just acknowledge members of my lab, in particular Benjamin Gabriel, who perform most of these studies, as well as the screening core collaborators in the pathology department, as well as my collaborators, Mayan Levy, Christoph Dice, and Benjamin Abramoff, who contributed to some of these studies. Thank you.
Great. Thank you, Sarah. Such really relevant data to the clinical trials that we need to keep running in the long COVID space. So thank you. All right. Next up is me. I'm going to present. So I am Amy Prowell, president of PolyBio Research Foundation, but I also serve as the scientific director of the Cohen Center for Recovery from Complex Chronic Illness, or CORE. It's a clinic at Mount Sinai where I work with David Petrino and a really amazing team to innovate care for patients with long COVID, ME/CFS, tick-borne, vector-borne illness, and other complex chronic conditions. And that's relevant to this talk because I'm going to talk about a clinical trial that we're getting underway at CORE right now. And it's a clinical trial of low-dose rapamycin in patients with long COVID. So what my goal is to give you an overview of why we think it makes sense to trial low-dose rapamycin in long COVID, and then just a quick overview of the trial design itself. So first, what is rapamycin? Well, rapamycin, one second here, let me just move this. Rapamycin was isolated from the Rapa Nui bacterium Streptomyces hygroscopicus. This happened during a 1960s expedition to Easter Island, which is known by the inhabitants as Rapa Nui, hence rapamycin, when the compound was discovered there. It was this expedition of scientists with the goal of identifying natural products from plants or soil with possible therapeutic potential. And when these scientists were on the island, they found that the local indigenous people who often walked barefoot didn't seem to contract tetanus. And so that made them think there was something in the soil that might be providing protection. They isolated that. This was this metabolite of Streptomyces hygroscopicus. and it seemed to possess antibacterial and antifungal properties at that period in time. So actually rapamycin's original sort of indication was in an antimicrobial capacity. After that, then many teams tried to figure out, well, what is this molecule doing in the human body? And it strongly impacted the activity of a specific protein kinase, so much so that they actually named this kinase the mammalian target of rapamycin, or mTOR. And it's a kinase that controls functions such as cellular metabolism, autophagy, cell proliferation, homeostatic pathways. In fact, it has, a signaling part of signaling that controls many pathways. So really, rapamycin can have different mechanisms of action at different doses, which really makes it even more interesting for use as a therapeutic. Now, normally, and this is one thing, several teams realized that it could reduce the activity of certain immune cells when taken in a high dose, like a high daily dose. So its most common use is actually used in organ transplant rejection as an immunosuppressive drug. But key to our long COVID trial is that it's increasingly being used or trialed in a low, often once weekly dose for the treatment of other conditions. And in that capacity, it actually seems, at least in some trials and in some studies, to have effects that may actually support parts of the immune system. Now, it's also being used in lifespan extension, which is really interesting. And there's a lot of research on this. In other words, just extending how long, particularly, especially an animal in a study might live. So this is a group that published in Nature in 2009, and they found that rapamycin extended the lifespan of mice by 9 to 14%. And there's other studies in worms, in yeast models. This is particularly interesting to me. I got put in touch as I became increasingly interested in low-dose rapamycin with Matt Kaberlin, who's in that photo. And his group is running a low-dose rapamycin trial in dogs to promote lifespan extension, potentially in dogs. It's called the Test of Rapamycin in Aging Dogs, or Triad Cohort. And there's some really cute dogs in this trial, and I actually became truly interested as I started to read data on lifespan extension with rapamycin. But when you talk about extending lifespan, one of the things that we study at PolyBio that we're so interested in is the impact of infection on aging processes, diseases of aging. So, for example, you heard from Ben Reedad earlier talk about cytomegalovirus contributing to Alzheimer's disease or a certain phenotype of Alzheimer's disease. So we wanted to consider that further. Here's another study that shows that it's one of my favorite studies, and there's many connecting viruses to aging. This is a team that did a network analysis that uncovered dozens of viruses that encode proteins experimentally demonstrated to interact with proteins from pathways associated with human aging, including cellular senescence, which is a cell's loss of power over its division and growth. Now, these age disorder viruses identified by this analysis included most of the herpes viruses, many of the papilloma viruses, and not shockingly to this group, SARS-CoV-2. So, we also work with this group at Harvard Medical School. For example, Will Eimers on our PolyBio Scientific Advisory Board. For years now, they've been studying how, at least in a neuron in a dish model and in mice, the amyloid beta plaque at the heart of Alzheimer's disease can act as an antimicrobial peptide that forms in response to pathogens in tissue. For example, in the case of this study, the herpes viruses. So began to think, well, is there something about rapamycin that could be controlling infection or supporting the immune response? And that's probably part of why it could expand lifespan. And then Matt Kaberlin did point me to these papers which suggested, hey, this could be possible. These are trials done by John Manick's group. And what she did in this one is take elderly adults and give them 6-week treatments with 2 rapalogs. So these are analogs of rapamycin, also mTOR inhibitors with similar mechanisms of action. And a couple of things happened in the group that was given the rapamycin, these rapalogs. After just 6 weeks of rapalog treatment, there was a lower rate of infection for over a year in the group that was treated. This included respiratory infections, lower rate of those, and also, for example, lower rates of urinary tract infections. Also, they gave participants the influenza vaccine at the end of the rapamycin dosing period, and there was improvement in response to the influenza vaccine in the rapamycin group, suggesting a more robust immune response generally. And what really caught my attention was that they found increased interferon-induced antiviral gene expression in the adults who are taking rapamycin compared to controls. And interferons are the first line of defense against viruses. They induce the expression of hundreds of antiviral genes that inhibit the actions of many different viruses. So here is table 2 from that trial, and you can see these different upregulated interferon genes in the participants taking rapamycin. And I thought, wow, that's really interesting, because as we've been hearing, SARS-CoV-2 persistence is implicated in long COVID. The reactivation of other viruses like Epstein-Barr virus is implicated in long COVID. If we could better control or upregulate interferon signaling, that could help, I don't know, all of those issues. So I also looked at this second study done by Joan Manick's team. In this case, they used another rapalog in elderly adults. At the end of the rapalog dosing period, again, they found that influenza vaccine response was boosted by about 20%. This time, they actually also measured T cell function, though, and they found that RAD-001, this rapalog, reduced the percentage of CD4 and CD8 T cell lymphocytes expressing PD-1, which is a marker of T cell exhaustion, and we've heard about that later today. In fact, Nadia Rowan, who you heard from, and Michael Peluso, they mentioned this paper. This paper did find that SARS-CoV-2 specific CD8 T cells from individuals with long COVID preferentially express the exhaustion marker PD-1 and CLA4. So some signs of T cell exhaustion, including by one or two other teams, at least in the long COVID space. So that's interesting. Then this is just another encouraging trial done by Matt Kaberlin's team in the context of just SARS-CoV-2 and rapamycin use. What they found is that continuous use of rapamycin throughout the SARS-CoV-2 infection period, which is basically life, because we're all exposed to SARS-CoV-2. Well, what happened is that patients who were taking rapamycin reported more mild cases of COVID, and none were hospitalized, whereas patients in the control group reported more severe or moderate cases of COVID, and there were some hospitalizations. And rapamycin users also did not report any cases of long COVID, despite being an average of 7.5 years older than the non-users. So with these potential effects in mind, we decided that, hey, let's give rapamycin a shot in long COVID, and let's also, measure some of these parameters of the immune response in participants on the trial to see if any of these hypotheses of how the drug could work really hold. So what we did is we designed a double-blinded, randomized, placebo-controlled trial of 80 long COVID participants. It's a pilot trial. The dosage we settled on, and this was after meetings with several different teams working on rapamycin. There's a clinical trial, for example, in periodontitis. We work with that group. There's another group working on it actually for improving in vitro fertilization outcomes in women. We arrived at a 6 milligram once a week dose. And actually, patients will ramp up to that dose. They'll actually start at 2 milligrams for 2 weeks, 4 milligrams for the next 2 weeks, and then 6 milligrams for the remaining 2 months of the study period. We're doing that because we know that long COVID patients can be sensitive to medication, so we kind of want to give them that opportunity to build up to taking the drug in order to minimize side effects or potential dropout. And importantly, before intervention, at week 12 of the study and at week 24 of the study, we're going to be collecting blood samples and sending them to Akiko Iwasaki, who I'm so excited will be doing RNA sequencing to measure interferon immune activity, also CyTOF to measure T cell exhaustion and other T cell parameters in whole blood from the participants. Plus, we'll be doing a metabolic health monitoring panel, lipids, insulin, hemoglobin, other metrics, and to the capacity that we can squeeze it into the budget, metrics of SARS-CoV-2 persistence or other viral protein measures. In saliva, we'll be doing at-home hormone tracking in sub-participants, especially in women in the study, and we'll be doing endopat as measures of endothelial dysfunction. And that's a main overview. Our goal is that, again, better understanding who might respond and their biological correlates in this study might help us design a larger trial of a more tightly phenotyped group of patients, or allow us to potentially add an arm of pre-COVID ME/CFS or tick-borne vector-borne illness patients into the trial, but after we've first gotten a read on how well this dose is tolerated and some potential side effects and top types of characteristics of responders. So thank you. All right, and with that, I'll introduce our last speaker, who is my colleague, David Petrino at CORE. He's the Director of Rehabilitation for the Mount Sinai Health System and the Nash Family Director of CORE.
Hi, everyone. It is an absolute pleasure to be here. And Amy, thank you so much for inviting me to speak today. Since I'm the last speaker, I'm going to try and be real fast and keep us to time. But I'm really excited to be talking about a new clinical trial. We have a ton of clinical trials going on at the core over 2024 and 2025, which is really exciting because these are all drugs and devices that I think have a real chance of moving the needle on some serious symptoms. But today I'm just going to talk about one of our trials, which is a collaboration with a company that is called Humanity Neurotech. I wanted to talk about this one in particular because, I feel as though often when we put out, information about clinical trials, the community has tried so much already that they say, well, I've tried that, it didn't work for me, or, whatever the case may be. This one, I promise no one has tried because it is brand new and we're doing a first-in-human trial of it. So I wanted to share it with the community and just get some of the details out there. So to start out, I just wanted to point out that even when we first started monitoring long COVID symptoms, we started to see that cognitive impairment is one of the most common and disabling symptoms in long COVID. So roughly 60% or a little bit over 60% of people with long COVID were not just reporting cognitive impairment, because as we know, the reporting of cognitive impairment is much, much higher, but they were reporting cognitive impairment that was significant enough to affect day-to-day function. And the reason that this should really concern us is because the scale that we used in this 2022 paper to measure how much it was affecting day-to-day function was a scale that is typically used for folks who are experiencing brain injury. So what we were seeing was mild, moderate, and even severe impairment in people with long COVID that was aligned with the same level of impairment that we would see in people with a traumatic brain injury. So we know that cognitive impairment that comes along with long COVID is severe, it's serious, it's common. We also know this is, work that we published with Akiko Iwasaki and Michela showing that even mild acute cases of COVID lead to neuroinflammation, release of cytokines, release of chemokines that cause damage to microglia and neurons. So we know
David,
that
one thing, your screen has like 2 gray boxes on it.
Oh, where?
On the top and the side.
I can't see them.
The one you almost made it go away right now. Wait, no, it came back. I don't know. It's not the end of the world, probably, but
is it gone now?
No. Well, sort of. Yes, one went away. I don't know why.
Okay.
Right at the top, like where the title of your slide is, there's kind of one. Maybe just hit that area. See if it goes away. There. It kind of went to the side at least.
Yeah. Okay. So that's weird. It's the Zoom. It's the Zoom taskbar that's getting in the way.
Okay.
Okay.
All right,
I don't know how to minimize the taskbar, but anyway, let me.
Yeah, just keep going. Okay.
Just keep going.
I think so.
Sorry, everybody. In addition to our work, Mike Van Weyenburg has done some really beautiful stuff showing that folks with long COVID compared with healthy controls also, show signs of significant neuroinflammation, even on imaging. So we know that neuroinflammation is a driver of cognitive impairment in long COVID. And, there are therapeutics that target neuroinflammation, but the track record for some of these drugs has been spotty at best. so some people respond, low dose naltrexone is one of these drugs that the mechanism of action for low dose naltrexone is thought to be reducing neuroinflammation. As many people on this seminar might, attest, it works for some people, it doesn't work for others. We know that some of the drugs that we want to address neuroinflammation don't cross the blood-brain barrier. Some have terrible side effects, either acutely or if you're taking them for too long, like steroids. So in general, it would be, we have drugs, but the drugs are imperfect and they can be sort of tricky to navigate. Along this line, I just want to point out that neuroinflammation has a relationship to mitochondrial function in the neurons as well. And it's not a straightforward relationship. It's not if neuroinflammation goes up, mitochondrial dysfunction goes up. There's this very interesting cycle and relationship though. And so the reason that this relationship, I'm calling this relationship out, is because there is the ability to manipulate mitochondrial function in neurons in a way that will affect neuroinflammation. And that is the crux of the device that we're going to be talking about today, because turns out when you apply low energy magnetic fields across neurons, you can drive a phenomenon known as mitohormesis, which is this phenomenon where it's kind of a Goldilocks concept, where if you stress your mitochondria just enough to push up reactive oxygen species just a little, what you'll do is cause a significant anti-inflammatory effect on the neurons. So neuroinflammation will decrease as a result of this small stimulus that initially stressed the mitochondria. To that end, we've been working with this company, Humanity Neurotech, to gear up for a first-in-human trial of their device that provides a low-energy magnetic field to neurons in the brain. The company has already completed, animal studies to show not only safety but efficacy in the animal models of neuroinflammation, where the results were actually quite striking in these animal models that were receiving the low-level magnetic therapy. And so to that end, the study that we'll be starting in January 2025 is quite a simple one, and we're really excited to kick it off. We will be taking a baseline assessment that will involve a cognitive assessment, patient-reported outcomes, and we'll also be taking some blood biomarkers of neuroinflammation. Then people will be sent home with the device, and you can see what the device looks like here in the sort of top right of screen. Hopefully the boxes aren't blocking it, but it's a little headset that has a console, and people will be asked to use the device on a daily basis. And after 4 weeks of at-home intervention, we'll redo all of our assessments to see how people are doing. And then we'll do a follow-up at the end of the study 4 weeks after device usage is stopped, to see if the results of the device are persistent after the end of the trial. This will be a placebo-controlled clinical trial, so there will be placebo devices out there and there will be real devices out there. And this is something that we're really, really excited about. So I'll pause there and just thank everyone for their time, and thank PolyBio once again for supporting all of this amazing work and giving me the opportunity to chat. So thank you, everyone. And I think probably we're moving into questions.
Yes. I think that we could do maybe about 5 or so minutes of questions. By the way, as was obvious to everyone, we ended up running through the question and answer periods with each hour. That was because people, actually had so much information, honestly, to present that it was hard to keep it within 10 minutes, which I guess is a good problem to have. I think there's a couple more questions, so we try to answer questions in the chat instead. I think, let's see, we have one person who said, David, you could take this one. Metformin has been considered for aging. How useful or risky is it for long COVID, ME/CFS, and other illnesses?
Yeah, look, metformin is one of those drugs that some people respond well to and other people struggle with. One note of caution I will provide in terms of metformin is most folks with long COVID, as we're dose finding with metformin, will typically tell you that they have fairly severe GI symptoms as they're trying to figure out what's going on. That's not everybody, but it is a large percentage, and that seems to be less common in the healthy population that's using it for longevity. So it can be kind of a metabolically and physically stressful drug for some people with long COVID, whereas others have, because of the way that it works and the way that it helps with, production of energy, others have said this is really helpful and has, given them a little bit more energy and a little bit more resilience to PEM.
Yeah, and there was a trial in acute COVID which showed that use of metformin during acute COVID seemed to did prevent progression to long COVID to some extent. And that's interesting, which does bring up another topic in the long COVID space that we're really interested in also working with to move forward at PolyBio this year, which is that in terms of treating in long COVID, there are the treatments that you can do once someone is already sick with long COVID, and it's really important that we do a lot of those trials. But obviously, what we do during acute infection to prevent progression to long COVID is also really important. So especially, for example, with Michael Peluso's team and the Link group, we're beginning a new framework for clinical trials of drugs during acute COVID that might prevent progression to long COVID. And so that's one more consideration is use of the drug in both long COVID or acute COVID in terms of just trying to improve people's, responses. Yeah, let me, there's a question that someone asked. I think it's, it's a good question. They say, presentations were talking about varicella zoster virus, the herpes virus, reactivation in long COVID. And they've been trying to figure out how to word this. In some cases, patients with elevated varicella zoster specific T cells, are they displaying the shingles rash, or is this evidence of possible reactivation without the rash? And I think that, maybe Nadia, you could grab that question. I think that viruses, or she was on here. I think, let's see, who's on here that would do, that knows like a virus background on this one? Let's see, I think, or Johan. Okay. Johan, if someone has varicella zoster, it doesn't have to reactivate in a classic shingles manifestation, right? They could also probably have reactivation that would just have a number of other, impacts.
Yeah, just straight from top of my head. Mark has more experience with it, so it's not always the case, but I hope to give you an answer in a few months, because we're actually testing reactivation at the molecular level in all our long COVID patients. We're rescreening the cohorts with a pan-herpes panel. So VZV, EBV, CMV, all of them. So I hope to give you a detailed answer in a few months.
Exactly. Okay, good. That's perfect. We'll have more data. All right. I think most of the other questions were largely answered in the chat. So for there, I think I'll go ahead and just wrap up the symposium. I want to thank everyone who spoke today. Really appreciate the talks. It's great to see the collaboration among teams. I also want to thank all the patients who participate in our studies and trials. Thank you so much for donating your samples, for coming in for the study visits, for helping us to do these studies and trials, to be able to study long COVID and related complex chronic conditions. Last, I do want to thank our donors. Every time that you'll see someone thank PolyBio for funding, what that means is that donors actually believed in us And donors such as Vitalik Buterin and his funds of Balvi and Canro, or the Silver Givings Foundation, or the Wallace Foundation, or other donors have believed in our vision, our collaborative vision of growing this field of infection-associated and complex chronic disease, moving these technologies forward and moving these collaborative networks forward. And they've given funds to us to be able to give out. So we appreciate those donations tremendously and the support for all of the research that's been happening. Thank you very much. And we will see you at the next symposium in 6 months.